अच्छा भाई सुनो, ये चैट बॉक्स में डालना है ना क्वेश्चन सारे।
हाँ, तू वो यूट्यूब लाइव लाइव चैट से कॉपी करना और यहाँ पे पेस्ट कर देना, ठीक है।
हाँ हाँ ठीक ठीक है भाई।
Hello, can you hear me?
Yes, ma'am, you're audible.
Okay, okay. Thank you.
Good afternoon, all.
Hello, good afternoon.
Thank you, Medha. Thank you, Rishabh. And I can see Barsha, Jessica, Shubham. Okay, thank you. So Daniel has joined.
A very good afternoon to our respected guest speaker, professors, seniors, and all my dear viewers. It's a moment of great privilege and honor for me to extend a hearty welcome to all of you to the second day of Modern Trends in Microbiology, Chapter 18, a 3-day international webinar under DVT Star College scheme organized by the Department of Microbiology, St. Xavier's College, Autonomous, Kolkata. Today is an eventful day where we will get the opportunity to hear an engrossing lecture from our guest speaker, Dr. Daniel Ryan, see some fascinating presentations made by our very own students, and then watch a vibrant cultural program. Myself, Ohuna Nundi, along with Jessica Michael, will be MC for the first technical session. Over to you, Jessica.
Thank you, Ahana. Now I would like to request Dr. Kasturi Sarkar, the Associate Professor of Department of Microbiology, St. Xavier's College, Autonomous, Kolkata, to kindly introduce our guest speaker, Dr. Daniel Ryan from the Helmholtz Institute for RNA-based infection research. He will be enlightening us on the topic, RNA landscape of a dominant gut commensal bacteroids thetidaizomicron. Over to you, ma'am.
Thank you. Thank you, students. Am I audible?
Yes, ma'am.
Okay. A warm welcome to all of you attending the program of second day, Modern Trends in Microbiology 2021. And it is my pleasure to introduce Dr. Daniel Ryan to you all. Department of Microbiology, St. Xavier's College started its journey as an honors department with a group of enthusiastic students in 2005. And Daniel Ryan was one of them. My journey in St. Xavier's College as a faculty member also started almost at the same time. Hence, it is really a proud moment for me to introduce the young enthusiastic boy of the day to you, who is now a postdoc at Helmholtz Institute for RNA-based infection research, Würzburg, Germany. So welcome, Daniel.
Thank you very much, ma'am.
After completing graduation from St. Xavier's College, he did his master's and PhD from School of Biotechnology, KIIT University, Orissa, India. And the title of his PhD thesis was Role of Small RNAs in the Acid Tolerance Response and Virulence of Salmonella typhimuriae. After that, he joined St. John's Research Institute, Bangalore, Karnataka, as a research assistant, where he standardized a PCR method to detect HIV-1 from dried blood spots, which provide a safer and easier way to transport samples. He also standardized a method to extract Salmonella genomic DNA from human blood clots and a multiplex PCR to detect Salmonella typhi and paratyphi. Currently, he is a postdoc at Post-Pathogen Microbiota Interactions, Helmholtz Institute for RNA-based infection research, Würzburg, Germany, and involved in RNA biology of bacteroids theta iota micron. Is it right, Daniel?
It's close, ma'am.
Yeah.
I will
Because this
tell
is
you
the
the
first
story
time.
behind the name. Yeah.
Yeah. He's interested in the identification and characterization of non-coding RNAs in members of the gut microbiota as well as pathogens. Dr. Ryan has published some very good research papers in peer-reviewed journals. One such paper titled "A High-Resolution Transcriptome Map Identifies Small RNA Regulation of Metabolism in the Gut Microbe Bacteroids theta iota micron."
That was
And
perfect.
yes, and that paper has been selected as HZI paper of the month and has been published in August 2020 in Nature Communications. Today, we will be talking on the RNA landscape of a dominant dominant gut commensal that bacteroids theta iota micron. And I'm sure it will be an interesting session, and we all will be truly enlightened about some of the modern research trends in microbiology. We'll learn many new aspects of this microorganism. And over to you, Daniel. Once again, welcome to St. Xavier's.
Thank you very much, ma'am. Yeah, it's a pleasure to be here. It's been 13 years since I saw you all also last. So it's really a pleasure to be invited back and to come as a speaker. It's a pity that I couldn't come in person, but with the pandemic and travel as of now, it's at least I can deliver the lecture online. So yes, I will get started.
After a long time.
Yes, that's true. All right, I just share my screen. 1 second.
Yes, sir, you're requested to start your screen sharing and you can start.
I'm doing that.
However, I have a small announcement to make to the audience that during sir's lecture, if you have any questions regarding the lecture, you are free to write in the chat box of the YouTube, which at the end of sir's lecture will be addressed to sir and he will be answering to some of those questions. And you are also requested to kindly follow sir's lecture pretty well, because on the third day of Modern Trends in Microbiology, you will be given a feedback form, which will be consisting of questions from all the 3 days lectures. So upon filling up of which only you will be awarded with the e-certificates. So audience, I request you all to kindly pay attention to sir's lecture. Over to you, sir. I won't take much of your time. You can start with your lecture, sir.
Thanks a lot. Is my screen visible?
Yes, sir.
All right. Okay, so let's kick in. So the topic that I would like to talk to everyone about is titled "The RNA Landscape of a Dominant Gut Commensal, Bacteroides theta iota omicron." In short, B theta. So that's the way I'm going to be referring to this organism, because it's a mouthful, I know. I'm working currently as a postdoc in the Westermann lab, where we work on host pathogen microbiota interaction. So therefore, looking at dynamics of how bacteria interact with each other and with the host epithelial cells in our gut. With regard to that, I will spend a little bit of time introducing you to the microbiota and Bacteroides theta specifically, and then jump into a little bit about non-coding RNA regulators in bacteria, just so that everyone is on the same page. And then I will go into the major work that I would like to talk about, which is the RNA landscape of this organism, B theta, followed by summarizing the conclusions, and then looking at some interesting projects that we are working on currently in the lab. So to start with, what is the gut microbiota? As most of us probably already know, the gut microbiota is the ensemble or the collection of microbes that inhabit primarily our large intestine. Why the large intestine? Because the small intestine is relatively harsh. You have antimicrobial peptides, high concentrations of bile. You have low pH, so acidic pH, as some of the acids from the stomach move into the small intestine. And therefore, you also have high oxygen concentration. And therefore, you have quite a harsh environment where it's a bit tougher for microbes to, let's say, find a niche. And so as you move further on to the large intestine, a lot of these conditions subside. So the oxygen concentrations sort of reduce. As you need to keep in mind that most of the organisms that grow in the gut are anaerobic in nature. Therefore, oxygen is toxic to them. So therefore, taking into account all of these factors, so low pH and antimicrobial peptides, bile, salt concentrations, etc., the large intestine is a conducive home or a niche. It forms a niche that is suitable to the growth of most of the microbes of our gut. And as a result, you have a flourishing, thriving community of microbes that constitute the gut microbiota. With regard to development of the microbiota, it starts to develop immediately following birth. And for the first 2 to 3 years of life of the individual, it undergoes quite a bit of fluctuations, which is a normal part of, let's say, the development of the microbiota, following which the population remains relatively constant throughout life. However, this population, let's say, can be influenced and is influenced by a huge number of factors. For example, at birth or in the neonate, the population, let's say, would be influenced by the kind of birth, whether it's a vaginal birth or a C-section, for instance, whether the baby is milk-fed or fed some other bottled milk, for example, or breastfed. Throughout life of the adult, the microbiota composition can be influenced by a person's diet, whether they have taken antibiotics, whether they have taken particular drugs, whether they've had some injuries or stress in the gut. And therefore, while being relatively constant as a whole, you do have minor fluctuations in the composition of the microbiota. However, the microbiota, as we all know, is highly beneficial to the host, to us specifically. And some of the functions that it mediates include helping our immune systems to develop, helping us consume or utilize certain polysaccharides, let's say plant-derived carbohydrates that we ourselves cannot digest efficiently, but the bacteria in the microbiota are able to digest these polysaccharides and feed us, supplement our energy through these metabolic pathways. We also have certain vitamins that, for example, vitamin K, that are produced by members of the gut microbiota, and hence it's one of our major sources of vitamin, obtaining vitamin K. So the gut microbiota as a whole is vital to our host, our well-being. And of late, there is also a lot of research, and I'm sure a lot of you would know that the effects of the gut microbiota can be felt all the way up to the brain. As you see, there's a lot of papers and a lot of research going into looking at the gut-brain axis, is what it is known as right now. So a disbalance to the microbiota, or if the microbiota is influenced in our gut by maybe, let's say, someone takes antibiotics, this imbalance is known as dysbiosis. And dysbiosis, or this imbalance, can result in a lot of functions being hampered in our physiology. So you could have problems with, let's say, obesity, or you could develop diabetes. So there's not, I wouldn't say these are direct results, but there's a lot of correlation that can be drawn with having an imbalanced microbiota and certain diseases, for example, diabetes, obesity, colorectal cancer, etc. All right. Just to give you an idea, so there are two dominant bacterial phyla. So at the bacterial level, let's say there are two dominant bacterial phyla that constitute our human gut microbiota. And these are the gram-negative Bacteroidetes and the gram-positive Firmicutes. As you see in the graph over here in this figure, this shows you the composition of the gut microbiota across healthy individuals across the world, so from around the world. And as you see, two phyla are quite dominant throughout, which are the gram-negative Bacteroidetes and the gram-positive Firmicutes. Within the gram-negative Bacteroidetes, it's Bacteroides, the Bacteroides genus that predominate. And this brings me to my next slide, which is our gut model organism of choice. And what we are working on at the moment is Bacteroides theta, iota, omicron. So looking at this first picture, it should tell you something about where the name is derived from. So the Greek, the letters theta, iota, and omicron are derived from the shape of the bacterial cells as seen under a microscope. So this is one of the first drawings of B theta when observed under a microscope by Di Stasso et al. This is one of the first papers in 1912. And you see the three shapes quite clearly visible with the theta, the iota, and the omicron. So the round sort of coccoid shapes. I will show you, I will explain to you the figures, and then I'll tell you why this organism is interesting for us. The figure below is a relatively new electron micrograph that shows you the B theta cell with an extensive capsule layer. And so this capsule in Bacteroides at least is phase variable, which means that it can switch its capsule. So there are 8 different types or 8 different loci where these capsules are encoded, and it can switch and alternate between different types and therefore protect itself from our immune system. Okay, and one of the final figures that I wanted to show you that gives you an idea of where and how B theta has its niche in our colon or our large intestine. Here you have the intestinal villus. So this is zoomed in highly, and you have B theta embedded in the mucus of the intestine. And this is basically its niche, or where it basically loves to stay. So this organism is of interest to us. Why? Because number one, while several organisms of the gut we are able to culture them, we cannot genetically manipulate them. However, since the last, let's say 2 decades or so, people have been working with Bacteroides theta being a dominant member of the gut and trying to develop tools to be able to genetically manipulate it so that we can play around with its genome, remove genes, put in genes, and be able to study it. And therefore, the tools are available for us to be able to investigate the functions of different genes in B theta, how they contribute to establishment in the intestine, what are its interactions. So they can all be studied using the tools that are already available. Just to give you a little bit in numbers, so the B theta has a genome length of 6.26 MB, and it has a single plasmid, at least the strain that we work with, which is a strain called VPI 5482. It has a single plasmid of 33 kb in length, and the plasmid is mainly involved in environmental sensing. Okay, so B theta and the gut. Let's just go into a little bit of the dynamics and what does B theta do in our gut. So as you see in this figure on the left-hand side, the green, let's say green rod-shaped bacteria are B theta in this case, for all intents and purposes. And you have the gut epithelium lined by a layer of mucus, and you have an oxygen gradient that is higher towards the epithelium, and it moves and it becomes lower towards the lumen. So epithelial cells of the gut are constantly metabolizing, and they also release a little bit of oxygen, which is a defense mechanism, basically, in addition to antimicrobial peptides that are also produced by cells of the gut. In addition, there is a peristaltic flow, or a movement of the contents in the gut down towards the distal colon. And this flow also generates, let's say, a selective pressure on the organisms that need to colonize the gut. And therefore, B theta really needs to be specialized at being able to colonize the gut. And this is why it is an interesting, let's say, model organism, being a dominant commensal, which means that it is specialized at colonizing the gut. And one interesting way at which it does this is through its ability to utilize carbohydrates that are, let's say that we ourselves cannot degrade. So we cannot degrade a lot of the large, or we cannot utilize or metabolize a large number of plant-derived polysaccharides. However, B theta, with its 246 glycoside hydrolases, it can utilize carbohydrate sources that would be a waste, or which would just pass out into our feces if it weren't present. The machinery that B theta uses to do this are organized into what are known as polysaccharide utilization loci. So there are these groups of proteins that are present on the inner and outer leaflet of the gram-negative membrane. And what they do is they are able to bind to large polysaccharide molecules that are then cleaved by certain enzymes into smaller linear molecules that pass through the outer membrane, which are then further processed into smaller monosaccharides and disaccharides that can then be utilized by B theta. This architecture is sort of quintessential to the ability of bacteroidetes in general to be able to occupy this gut niche. Right. So that was just a brief introduction about B theta, or our organism of choice. And now, before we jump into the work, I want to tell you a little bit about regulatory RNAs in bacteria. So what are regulatory RNAs? So as the name suggests, they regulate. What do they regulate? They regulate mRNAs in particular, so post-transcriptional or translational regulators. And these regulatory RNAs do not generally code for any proteins. However, they play roles in regulating expression. What you see in the figure over here is an example, basically a canonical example of an intergenic small RNA, but this will help me illustrate how they work and what principles that these small RNAs work on. So they are generally, they fold to form relatively complex secondary structures that have stem loops within them that play roles in their stability as well as their function. And they also are characterized, at least the majority of the intergenic small RNAs. So intergenic would mean that they are encoded between coding genes. So they are not, let's say, they are not overlapping any genes that are already known. And so these small RNAs at their 3' ends have row independent terminators, which is a feature of the transcript and how their transcription is terminated. Their activity, on the other hand, is mediated through antisense binding. So these small RNAs are able to bind to their mRNA targets through complementary base pairing. And this complementary base pairing is mediated by a short, it depends, but in most cases by a relatively short sequence known as the seed sequence. So binding of these regulatory RNAs to their mRNA target then can mediate downstream functions. For example, if you have an mRNA gene, binding of the small RNA across the ribosome binding site can block binding of this ribosome, and therefore you don't have translation of the target gene. Or what they can also do is this complementary interaction can recruit what are known as nucleases, so RNases that bind to the small RNA and the mRNA target and degrade them. And therefore, again, you have expression being turned off. Another case, you could have, let's say, when an mRNA is transcribed, its ribosome binding site is blocked by a secondary structure. For example, there is a loop blocking its the access of the ribosome. Binding of a small RNA can then open up this loop and allow translation to occur, thereby activating a particular gene. So these regulatory small RNAs play roles in repression as well as activation of a wide variety of genes and are triggered by several environmental signals and stimuli, which can be stress mediated or nutrient mediated, whatever. There are several different signals. And to add, in many cases, these regulatory RNAs have been shown to require RNA chaperones. So like protein chaperones, which help fold proteins into functional conformations, RNA chaperones help the RNAs to interact. So they assist the interaction of a small RNA with an mRNA and thereby allow it to mediate its activity. However, in B theta, which is our organism of interest, we have not found any of these RNA binding proteins, at least global RNA binding proteins. And therefore, this would imply that either there are different modes of action of the small RNAs in members of the gut microbiota, or there is a totally different RNA binding protein that is not known and differs from the conventional canonical proteins of E. coli and Salmonella and other enterobacteria, for example. All right, so to give you just a short brief idea of the different classes of regulatory RNAs, I'm going to talk about three major ones. The first type are ribose switches. So what do ribose switches do? Let's say you have an mRNA. Ribose switches generally reside in the 5' untranslated region of the mRNA. And when they are transcribed, they fold in such a way that they form a terminator structure. And this terminator structure prematurely terminates transcription of the downstream gene, right? However, the characteristic of these ribose switches is that they are able to bind to particular ligands or small molecules, for example, vitamin molecules, or let's say small metal ions. And binding of these ligands to these ribose switches causes them to change their conformation. So they fold and unfold and thereby disrupt the premature terminator that is formed in these 5' untranslated regions, and then allow transcription to proceed, for example. So therefore they function at the transcriptional level. So they are transcriptional anti-terminators, let's say. Another regulatory structure, another type of regulatory RNA that is seen in bacterial cells as well as in eukaryotic cells are thermometers, RNA thermometers. So these are similar to ribose switches, except that they are also present in the 5' untranslated region. However, when they fold, they block the ribosome binding site, and therefore in this case, translation is inhibited. However, with a slight increase in temperature, for example, so these can generally be present as sensors for a bacteria to know that it's entered the human body, for example. So if you have a bacteria that's in the environment and enters the human body through food, the increase in temperature to about 37 degrees, what temperature? 98, 99 degrees Fahrenheit will cause unfolding of this ribose switch, the RNA thermometer, that then allows translation of the particular genes to occur. However, by far, the largest groups of regulatory RNAs in bacteria is this third group, which are the cis and trans encoded small RNAs. And this example that I showed you previously is an example of this category of, let's say, trans encoded small RNAs. So these third group, they are named as cis and trans depending on whether they are encoded on the strand opposite to a coding gene, for example, here. So this is a cis encoded small RNA, or whether they are located at a locus that is far away from the gene that they are targeting, therefore trans encoded small RNA. And they function by antisense pairing to their targets, and the mechanisms I have already told you. They can block ribosome function, they can cause degradation of the mRNA sRNA pair, etc. All right, so this figure will help you understand the role regulatory RNAs play and why it is important for us to understand regulatory RNAs in context of the gut microbiota. So as you see over here, this is the at least state of knowledge of roles played by various regulatory RNAs in disease-causing bacteria and different infections. And as you see, for example, in Salmonella, there's a whole host of small RNAs that are known and that play a role in its virulence and its ability to cause disease. You have small RNAs involved in meningitis, gastritis. Helicobacter pylori has a lot of small RNAs that play a role in its infectious cycle, let's say. However, in case of the gut bacteria, we do not have any information on these small RNAs, and therefore we do not know we are missing basically an entire strata of regulation. We do not know how these bacteria, how these small RNA permit the bacteria to occupy their niche, how they influence the interactions of these bacteria with other members of the microbiota, with other members in the host, with other cells within the host. And therefore, this brings me to the main topic of this talk, which is the RNA landscape of the dominant gut commensal B theta. And now, at the time of starting this work, there was just 2 papers on small RNAs in Bacteroides in general. One paper spoke about cis-encoded small RNAs, which are the small RNAs that are present opposite coding genes. And these small RNAs were found to regulate some of the polysaccharide utilization loci in Bacteroides. While another group of small RNAs was found to play a role in regulation of the conjugative transposons. However, barring these 2 papers, there was really no information available on the transcriptome of Bacteroides, B theta specifically, and its small RNAs or its coding sequences. There was really a lack of information. Therefore, to further understand or to improve our understanding of this gut model organism, our first aim was to annotate this transcriptome to as high a resolution as possible. And by annotating the transcriptome, what I mean is to know where transcripts start, where transcripts end. This is very important to know the boundaries of transcripts, because knowing this, you know where genes can be coded, where non-coding RNAs are present. You can identify promoter structures, so where promoters are present, and you can even identify novel genes that are, let's say, expressed under different conditions. So the technique that we applied, or that I applied, is known as differential RNA sequencing. And the technique involves growing B theta under 3 growth phases. So we took 3 growth phases to start with, which were early exponential phase, mid exponential phase, and stationary phase. So these are the phases of growth of bacteria. So you have the early exponential phase, which is when it just starts picking up its exponential growth in new media. So we did this growth in rich medium. And then we isolated total RNA. Now, it must be remembered that total RNA has different types of transcripts. It has what are known as primary transcripts. Primary transcripts are transcripts that are produced directly from RNA polymerase, whereas the processed transcripts are those that are further processed. And so they have different 5' ends, so a 5' phosphate and a 5' OH. So for the purpose of mapping where transcription starts, these transcripts are really of no use to us. And therefore they need to be, they can be eliminated. So what we do, once we have the total RNA library, we divide it into half. 1/2 of the transcripts are treated with an enzyme known as terminator exonuclease. What this enzyme does is it degrades all of the processed RNAs. And by doing that, all that remains in this RNA library are the primary transcripts, while another library, which is our control library, has all of the types of transcripts. So you have the primary transcripts as well as the processed transcripts. And then what we do, we generate cDNA of these libraries and sequence them. So we sequence the 3 conditions and the 2 different types of libraries. And by observing the pattern of reads that align to the genome in the treated versus the untreated libraries, we are able to see where transcription starts. For example, in the treated library, all of the primary transcripts would have a greater number of reads aligning at their start point, and therefore you would see a strong peak at the start of the transcripts as opposed to in the untreated library. And this is what I will show you in the figure on this slide. So I will explain to you what we see in this figure. So you have 3 growth phases: early log phase, so early exponential phase, mid exponential phase, and stationary phase. And you have the library that's treated with that is not treated with terminator exonuclease, and the library that's treated with terminator exonuclease. Okay, and below you have the gene that is that these reads are aligning to in each library. And as you can see, we can easily mark or determine where transcription starts by looking at the peak or the enrichment of reads in the terminator exonuclease treated to the untreated library. And so we did this for the entire genome or transcriptome of B theta under these 3 conditions in triplicate. And of course, each of these, we have a pipeline or a bioinformatic pipeline that can detect these transcription start sites, but each one needs to be manually annotated, and that takes quite a bit of time to manually annotate each one of them. So we inspect them to make sure that the computer is making a right call, or that the software algorithm is making the right call. And so once you have an idea of the transcription start site, so you know where transcription of a particular transcript starts, you can then map the transcriptome. So you can identify its promoters, because promoters are always present several nucleotides upstream of a transcription start site on the genome, right? You can also identify 5' untranslated regions, which is the distance from a transcription start site to where the coding gene AUG, so the initiation codon, lies. So within this 5' untranslated region, you have a lot of the regulatory segments that have the ribosome binding sites and various other enhancers and other the small RNA landing pads, for example. You can even identify operons, open reading frames, and what is of major interest to us, small RNAs. So to jump into a little bit of the results, we identified a total of 4,507 transcription start sites in the genome and plasmid of B theta. And these transcription start sites, we further classify them into 5 different types: primary, secondary, internal, antisense, and orphan. This categorization is based on the location of these transcription start sites, or where the reads align relative to known coding genes. So if we have a huge number, or the maximum number of reads aligning to a particular point that is within 300 nucleotides upstream of gene X, this point would be the primary transcription start site, while all other, let's say, lower aligning reads would imply secondary transcription start sites. So this is a way to classify them. For example, antisense transcription start sites would be where reads align antisense to a coding gene on the reverse strand, and thereby these reads might signify antisense transcripts that play a role in maybe silencing of these sense encoded genes, or they could also be unknown antisense genes that have not been identified as yet. Similarly, you have internal transcription start sites, which are those that align within a coding sequence. So if you have a gene that is already known, you have reads aligning within it and forming a peak, signifying that transcription is also starting from within. And these could signify the starts of sub-operons, for example, right? Another category that is of interest is the orphan transcription start sites. And these are transcription start sites that do not occur anywhere close to any known coding sequences. And why they are interesting is because these would signify, or could possibly mean, that we have transcripts of unknown origin that could signify unknown coding sequences that were not annotated as of now, so something novel, or small RNAs that further can be analyzed. So as I said, we were able to identify several features of the transcriptome. Another feature is the 5' UTR length, or the 5' untranslated region. So this is the region on the mRNA that is not translated, or that includes generally where the ribosome binds. It was found to have an average length of 52 nucleotides, which is the same, or relatively the same, as in all bacteria. So nothing super interesting about this. However, we did find a few genes that had 5' UTRs that were excessively long. And when we looked into these genes further, we found that there were possible sites where small RNAs could bind. And I will talk about a few of these a little later. The other things that we identified, or the other features that we identified, were the promoters. Now, Bacteroidetes has a known canonical promoter, which is known as the sigma ABFR. It's similar, I would say it's relatively similar to the promoters in E. coli, where you have the -10, -35 box, so the Tata box. However, in B theta, we have a -7 and a -33 box, and this is not rigid. In B theta, this -33 box can vary quite a bit. So therefore, there's this variable spacer region. And that is just a feature in B theta that is still under investigation. Yes, we also identified a new motif that was not reported in the literature previously, that also resembles possibly a promoter or a transcription factor binding site, maybe, where you have a -10 and a -30 box that are inverted repeats. And the interesting thing about this was we found a lot of these motifs present upstream of genes that were involved in entry into the stationary phase. But yes, we are still characterizing and still looking into this at the moment. To come to the focus of the work was to look at non-coding RNAs in B theta. And we identified over 200 we predicted, let's say that's a better word. We predicted over 200 non-coding or regulatory RNAs, non-coding RNAs in B theta in the genome as well as the plasmid. And for example, we found 14 riboswitches, 13 RNA thermometers, 78 cis antisense RNAs, and 124 intergenic small RNAs. Now it's good to say that we predicted them, that's done by running a analysis pipeline. However, we need to validate these. And so we validated several candidates, of which I show you a few of them. So we do the validation using a technique, using northern blots, where you have probes that are designed that are antisense to the RNA that is being transcribed. And then when you run your total RNA on a gel, you can detect the particular small RNA band. So it's like running an agarose gel with a PCR product. However, in this case, you're running total RNA on a polyacrylamide gel, and you are detecting your product using radioactive probes. Yes, so we detected a few candidates. I will highlight 2 or 3 of them that are interesting. The first is the 6S RNA. Now, this is a highly conserved RNA that has been that is known throughout bacteria, and it plays a role in regulation of the activity of RNA polymerase. In the next slide, I will show you how it does that. But this is just to show you that we validated several important or interesting candidates. We also identified another small RNA, which is a cis antisense RNA. And this belongs to one of the categories that was already published. So the ones that bind antisense to the PUL genes, or the ones that regulate PUL genes that are involved in metabolism, which was one of the only papers that was previously published. And therefore, this was a validation of our technique, or the part validation of our technique, that it actually worked to identify these groups of small RNAs. The third one that I will also talk about is GIB-S, which is an intergenic small RNA that we have characterized functionally, as well as looked into what regulation it could mediate. And this is going to form the next part of my talk, where we will focus on GIB-S. Okay, so if you remember, I told you that the secondary structures, or the way these RNAs fold, plays a role in their function. And this is evident, and the 6S RNA, let's say, is a good example of that. So the 6S RNA folds into a structure that has a huge loop within it, or a huge bulge. And this bulge, if you recall let's say your elementary studies of transcription, this bulge resembles a transcription bubble. And what this does is this mimics the structure on DNA of a melted -10 box of a promoter, or a -7 box. And therefore, you have RNA polymerase gets pulled away, let's say, from its targets on the DNA, and can then bind to this RNA, relieving the DNA and thus reducing the expression of several genes that would normally be expressed if this 6S RNA was not present. And research has shown that the 6S RNA generally increases or is highly expressed during a transition from the, let's say, late exponential phase to the stationary phase. Because in the stationary phase, you do not need a lot of gene expression to be occurring. It's a phase where stresses are starting to be imposed on the bacteria. And therefore, to limit the amount of transcription that is occurring, this 6S RNA titrates the RNA polymerase away from its targets and reduces the level of gene expression. Another small RNA that we found of interest was BTNC201. This has, I would say, one of the most canonical shapes of a small RNA, where you have a 5' loop that is generally involved in maintaining stability of these RNA molecules as they block, let's say, nucleases from coming and trying to bind. And you have a 3' row independent terminator at the end, marking the end of the transcript. So this is the length of the small RNA, which is about 99 nucleotides. This is of unknown function, and it's a candidate that we are also currently working on to see what it does, basically, in B theta. The third candidate is our one of choice, which is known as GIB-S. I will tell you why we named it in the next slides, but this is also an intergenic small RNA. It is partially conserved, meaning that it's present in a few of the Bacteroides species present in the gut. We found it to be induced by the carbohydrate NAG, which is N-acetyl glucosamine. So NAG, NAM, they're also present in the cell wall of bacteria. And we have found this small RNA to function as a metabolic switch. I will go into the details of this small RNA now, just to show you how we work with these small RNAs and our way of thinking and analysis. So GIB-S is, I said, a relatively, let's say, relatively well conserved small RNA. So it's present in all of these 1, 2, 3, 4, 5, 6, 7 species that we looked at. And it is generally located between a tRNA locus and genes that are required for metabolism. Now, carbohydrate metabolism, and this is not really surprising because B theta, as we know, is dedicated to utilizing and processing of carbohydrates in the gut. So in order to find out what its possible role could be, or what triggers this small RNA to be expressed, we decided to profile or look at its expression during growth of B theta under different carbohydrate sources. So what we did, we grew B theta in minimal media that had several different carbon sources. So we used minimal media with glucose, we used minimal media with fructose, galactose, mannose, NAG, xylose, and maltose. And what we found when we performed a northern blot was that GIB-S, or this small RNA, was highly induced when we grew it under NAG. In this, so GLC-NAC is the carbohydrate term for writing for N-acetyl glucosamine. So it's the code. And therefore, we decided to name this small RNA GLC-NAC induced Bacteroides small RNA. Therefore, you have GIB-S as what it's called. So we know, or we have an idea of what condition induces the expression of GIB-S. The next interesting question was, does it regulate any targets? And if so, what are these targets? How does the interaction occur? Is it through direct binding? Is it through some intermediate partners? So we used let's say a biochemical assay known as enzymatic structure probing, where we determined the structure of the small RNA biochemically and found it to have a relatively linear 5' region. So this is the 5' end of the small RNA. This is its 3' end. Now it has a relatively linear 40 nucleotides at the 5' end, followed by, as I mentioned, a secondary structure in the middle and a 3' terminator at the end. And this structure is quite interesting, as it probably, or we hypothesized it to have a role in binding of this small RNA to proposed targets. So what we did to identify targets was we generated a deletion mutant of the small RNA in Bacteroides, where we removed the gene, and another complemented strain where we put the gene back to show that when we remove the gene, these are the changes. When we put back the gene, the changes are brought back to the wild type. So this is done to validate the function of a particular gene. And we performed RNA sequencing of the 3 strains in stationary phase, at stationary phase in rich media. Once we do the RNA sequencing, we identify genes that are differentially regulated. So what genes are highly upregulated or downregulated in the mutant relative to the wild type strain. And this led us to identify 2, 4, 6, 7 genes that were differentially regulated. 5 of these genes were highly upregulated in the deletion strain, while 2 of them were also downregulated in the deletion strain. Now, just looking at these expression levels, if you have these 5 genes, let's say, upregulated in a deletion strain, so where the small RNA is absent, it would imply that under normal conditions, the small RNA is possibly repressing these genes, which is why when you remove the small RNA, you have increased expression. So we next went to, we looked at in silico. So we used target prediction algorithms to try and identify interaction points of where our small RNA of interest could likely bind to these targets and interact with them. And luckily, I'd say, we found that in 3 cases, this small RNA GibS binds, or in 2 cases, it binds across the AUG start codon as well as the ribosome binding site. So for gene BT0771 and BT3893, you see that there is binding of the small RNA across the ribosome, the initiation codon and ribosome binding site. Now, this is interesting because this forms or is evidence of canonical repression mediated by small RNA. So several intergenic small RNAs in the literature and previously have been shown to mediate repression of their target genes by blocking the ribosome binding site and blocking initiation of translation, and therefore, and thereby also mediating degradation of their mRNA targets. And therefore, what we wanted to next determine was the small RNA GibS directly or indirectly interacting with these RNA targets. So to do this, we used a technique known as EMSA, which is known as which is the full form is electrophoretic mobility shifting assay. Sorry, just give me a minute. Yes, so in an electrophoretic mobility shift assay, you look at direct interactions between a small RNA or between 2 RNAs of interest, whether they are directly or indirectly interacting. And to do this, you incubate one RNA, let's say the small RNA, with increasing concentrations of its target, in this case, BT3893. And then after you incubate them for a given amount of time, you run these products on a gel with the unbound RNA as a control. Now, what happens is if the 2 RNAs interact, they become larger and therefore run slower on a polyacrylamide gel. And so what you see over here is when you have the free small RNA, it runs quick. However, as you increase the concentration of its target, more and more of the small RNA starts to bind, and you have a much slower migration pattern in the gel. And therefore, this told us that, yes, this small RNA is indeed directly binding with this target, BT3893, that is in turn giving us these or resulting in these differential expression levels. Unfortunately, yes, we do not know exactly what BT3893 does. The thing is, in Bacteroides theta, it being such a, it's not very well functionally annotated. So we do not know the functions of, I'd say, more than 50% of the genes. Although we have a reason to believe that BT3893 is involved in a metabolic pathway that we are currently again looking into. So to summarize, this GibS small RNA is induced by a carbohydrate known as NAG or N-acetylglucosamine, and the increased levels of GBS repress two genes or two metabolic pathways while it stimulates another metabolic pathway. And therefore, we have reasonable evidence that suggests this small RNA might play a role in switching of metabolism in this organism. However, we are still looking into it. And I would like to say that what is currently of interest to us is if we went into the data and we looked at further conservation. So we looked at the presence of GBS in even more organisms and even more different Bacteroides species, let's say. And what we found was that while the small RNA was highly conserved in all of these genomes that we looked at, many of these targets that we identified in B theta were not present in several of these other organisms. This could mean that either GBS is highly conserved and the other and has no function in these other organisms, or we've likely missed out targets in these. And therefore, we are currently still looking into identifying even more targets for this small RNA. This is just an addition to our work. So what we have done now, we have set up a database known as ThetaBase that can be freely accessed by everyone, for the students and whoever is interested in working with Bacteroides theta, where we have all of the expression data loaded, and you can look at your genes of interest. You can look at different loci, for example, capsular genes. You can look at genes involved in metabolism. You can look at genes involved in small RNA, and it gives you all of the expression levels. You can further go into the browser and look at all of the sequencing data, where transcription starts, where small RNAs are located, where we have terminators, etc. And this, we hope, really helps build the Bacteroides community and really supports research in this field. So just to give you some general conclusions, I've told you that B theta is an emerging model organism. Knowledge of its transcriptome is essential, of course, for further study. And therefore, we present this high resolution transcriptome of B theta that has 4,500 or so transcription start sites. We've identified promoter structures, transcript features, more than 200 non-coding RNAs. GBS, we've also shown that GBS is a 145 nucleotide long small RNA. It's NAG or GalNAc induced, and it regulates several metabolic genes. The regulation in sRNAs, at least this study provides evidence that small RNA mediated regulation is also widespread in gut bacteria, which has not really been shown before. And further work is, of course, necessary to understand the diversity and extent of this regulation. And we hope that ThetaBase will really help research in the field. Now, for my second last slide, I just want to tell you about work that we are currently, or that I am currently doing in the lab. If you recall, I told you that B theta, when present in this gut environment, it encounters several different conditions. So different nutrient conditions, different conditions of stress, for example, antimicrobial peptides, bile salts, antibiotics, oxygen levels. And right now, what we are doing is we are profiling the growth of B theta under 17 to 18 different conditions, different carbon sources, different stress conditions. And we are re-annotating the transcriptome to try and see that our first annotation worked well, but did we miss out any genes? What functional pathways are involved? How they are playing a role? And all of this data will soon be, once we publish the work, it will be available to the scientific community at large. And we hope to set up ThetaBase version 2 very soon for everyone to be able to use. Another project, which is a pet project of mine and that I'm interested in looking at, are extracellular vesicles in extracellular vesicle RNA, sorry. So what are extracellular vesicles? These are membrane-bound vesicles that bud off from the membranes of all cell types, eukaryotic cells, prokaryotic cells, all. And evidence has shown of late that these vesicles contain, or have been shown to contain, small molecules like DNA, RNA, proteins, etc. However, there is very little information as to what generates these vesicles and how their contents are modulated and what's the function. So it is now a project in our lab where we are looking at the RNA content of these extracellular vesicles in context of the gut environment, and how or whether this RNA plays a role in signaling between different bacterial species, between the bacteria and their hosts, or whether they are just there and as a byproduct of a biological process that maybe, like, for example, when cells die, these vesicles spontaneously form. So these two are projects that we are currently working on in the lab. And with that, I would like to thank, of course, my boss, Professor Alexander Westermann, the entire group, and to everyone for listening in to my talk. I'm ready for questions. Thank you.
Thank you so much, sir, for such an intriguing session. I would now request Professor Dr. Mahashita Mitra Ghosh to moderate the question and answer session. Over to you, ma'am.
Before Mahashita speaks, I'm sorry I missed the beginning. Ryan, you spoke so nice. I really was listening to you and I really liked your talk.
Thank you. Thank you very much, sir.
Doing such a nice work after your PhD. I really appreciate your zeal. And I just, before Mahashita asked the formal questions, I just wanted to ask you one question.
Sure.
That is RNA going out to the biofilm for communication or like that?
There is evidence. So, for example, if in a biofilm, you would have nucleic acid components, you would have protein components, dead cells, etc. Whether these nucleic acids play a role in regulating, for example, as with small RNAs, whether they regulate target genes is not well known, but there is definitely nucleic acids present in biofilms. Yeah.
So it can be a good investigation, basically, to see their communication or communicating role.
So in context of the gut, the environment being as dense as it is, so in the gut you have roughly 10 to the power of 12, which is, I think, 10 trillion organisms per mg, or per gram, sorry, of fecal contents. And therefore, you would expect communication to be extremely important. And so I think it would be a good avenue to look at down the line. Yeah.
Thank you. Thank you very much. Over to Mahashita. Mahashita, please continue.
Welcome.
Yes. Brian, it was an excellent talk. As sir has shown, you just started from the very scratch, and you have very well understood that you are addressing students from BSc, MSc, and faculty members and PhD scholars as well. So your talk was considering the status of all the students, and it gives me pleasure that every time I am there in the end part of your journey, right? So far as I remember, in your BSc part 3 exam, I was the external of paper 8 and internal of paper 7. Right. So at that time, I also ended up your story of BSc. And even after so many years, when we are again meeting for after 13 years, it's really very nice. And although I was not involved in your teaching part, but definitely you have covered the story, and it is supposed to be the most promising topic of the era, right? So to understand the gut microbiota, and especially the role of small RNAs, right? And so beautifully you have gone through the little experiments that have definitely helped our students to learn that how to address any problem. So this is the best part of your lecture, right? So that you have very well explained the experiments, that how a problem is to be addressed, right? So there are some questions in the chat box. So one of our students is from MSc semester 3, Rushav Jawarkar is asking that if gut-brain axis is deformed, what is the outcome?
Okay, I was expecting these kind of questions. So the thing is, as of now, the gut-brain axis, we do not have direct causative evidence. So we have correlative evidence. So for example, in patients with dementia or patients with depression, for example, the levels of certain gut species have been found to be different or dysbiotic. But as to the actual cause, whether the difference in these gut bacteria is actually causing depression as a phenotype is not known. It's an extremely new avenue that people are looking at. However, what they have found, at least the little bit that I know in this field, is that with small, in case of mice, for example, they have found that in mice that have sort of a depressive phenotype, they have ways to know that mice are depressed. I did not know this till recently, but what they found was if they transfer the microbiota from a mouse that is depressed to one that is not, so if they do a fecal microbiota transfer, they find that the mice that receives this novel or this microbiota also starts showing signs of depression. So the research at the moment is very new, and it's not really we cannot really conclusively say anything about whether it is actually causing it in humans or not. But yeah, this is an avenue that really is building now for the future. Yeah.
Okay. Definitely, a large amount of work is to be done to say anything conclusive regarding the gut-brain axis. Okay. So he has again also asked you to elaborate
Ma'am,
on the.
sorry to interrupt. Ma'am, these questions we have taken from YouTube. The people who are viewing the live stream from YouTube, they have addressed these questions. Rishabh is a part of our tech team. He has forwarded the questions to our Zoom meeting.
Okay. Sorry. So Rishabh has question from the YouTube viewers is they have said that if you elaborate on the changes of microbiota in large intestine with respect to commonly used penicillin-based antibiotics on long-term use.
Okay. So it's really, let's say, the use of antibiotics in general is detrimental to populations in the gut microbiota, and penicillin, for example, affects the cell wall, right? So it's not super specific, and therefore you would have widespread, broad spectrum effects on different gut species, although the extent to which certain species are affected as opposed to others differs, because several bacteria, for example, B theta also, they have several outer layers of defense. B theta has an extensive capsule. I'm talking about B theta because I know a little bit more in this case. So B theta has an extensive capsule. It has an S layer, which is a layer of technically of polysaccharide armor that is surrounding the membrane. And therefore, for antibiotics such as penicillin, etc., to possibly damage the membrane, you would need slightly higher concentrations, I assume. But in general, not only with penicillin, but streptomycin, etc., they really deplete the microbiota. And this is actually a model that is used, for example, in salmonella research. We use what is known as a streptomycin pre-treated mouse model. Now, a streptomycin pre-treated mouse model is used so that we can study inflammation caused by salmonella. The streptomycin pre-treatment depletes the gut microbiota, killing several of them, and therefore their ability to resist invading pathogens is reduced, and therefore there is less protection in our gut, and you can study inflammation. So I would say that yes, there is an effect, but it's more broad. It's not very specific.
Okay. So another question, Daniel, is why there is no form of ribosome binding protein in B theta?
So it's not that there is no ribosome binding protein. So just 2 months back, we published a paper where we found, let's say, we can call not ribosome binding, RNA binding protein, right?
Yeah, RNA binding.
Sorry. Yeah. RNA binding. So yes. So we found an RNA binding protein, but when I was talking about there is no global RNA binding protein. Now, a global RNA binding protein is one that binds to several different RNA species around. We still have not found that in B theta, or how can I rephrase it? We have not found the well-known RNA binding proteins that are known in other bacterial species in B theta. There's probably other global RNA binders, but they are yet to be found, I would say. That would be the conservative way to say it. But they are definitely there. We have identified one of them. It's just that it does not show the extent of globalness as the other ones. Yes.
Right. And another question is why there is a peak in primary promoter for 5' UTR length?
So there is, okay, let me go back to the graph. I need to explain this. So this is, I should have explained this better. So this is the length of the 5' UTR, and this is the number of detected 5' UTRs. It has nothing to do with where the promoter is located, for example. So the peak, as you see over here, for example, is at 37 to 38, but this is just a few, let's say a few of them. You have a broad average at 52 nucleotides, which is where the majority of 5' UTRs lie. I'm not too sure about a peak in the promoter being concluded from here with that question, actually. So I really don't, I wouldn't be able to specifically answer with regards to the promoter, but all I can say is the peak that you see here is the number of the 5' UTRs that were detected at this length. Yeah.
Okay. So students, are there any other questions? I could find this many in the chat box. Am I missing anything? Please add to it.
No, ma'am. These are it.
Hey, Daniel, it was an
Yes.
excellent presentation.
Thank you. Thank you very much,
And
ma'am.
now the name is clear to me.
Thank you, ma'am. So should I stop sharing my screen?
Yes, sir, you can.
Yeah.
Thank you, sir, for enlightening us.
Thank you. Thank you very much.
Thank you, sir, for enlightening us with such an intriguing lecture. And it's really a great honor for us to have you today among us. I thank also Dr. Mahashweta Mitra Ghosh for graciously moderating this session. And I also thank the audience for being so interactive during this session. However, we will not be taking any further questions from here on. I now request Dr. Mahashweta Mitra Ghosh to deliver the vote of thanks, followed by presenting the e-bouquet and plaque to Dr. Daniel Ryan. Over to you, ma'am.
As I have stated earlier as well, that it was really very much interesting to hear from you because semester 3 BSc students and semester 3 MSc students do learn these techniques a little bit, and you have elaborated it so nicely, and definitely your way of delivering it has given the students the opportunity, or they have developed so much of interest. So after your deliberations, they will definitely be more aligned towards research of this kind. So that is the best part. That is the best part of your lecture, what I can find. Yes. That this kind of research, they will be definitely attracted towards it, the way you have presented it. What else, as Arup sir has said and Kasturi madam has also said, they have taught you. I was not there in the journey, but I still am enjoying that how nice you have acquired the skills to be you are a wonderful researcher. You are an excellent teacher. Okay, So
Thank you so much.
My thanks to all the organizers of the MTIM team. So our students have taken enormous efforts, BSc and MSc, say Medha and Anonna and the BSc students, Ohona and uh, Jessica, and all of them, so who are working behind the screen to execute this event with immense perfection, right? And I'm really thankful to Daniel that in spite of your busy schedule, you have given your consent to teach your juniors, and we also have learned a lot from you. And I really hope to meet you someday in person in St. Xavier's College. Okay, so
Sure. Thank you, ma'am. Thank you. And thanks to Arup sir and all of the organizers, Dr. Chowdhury, everyone. I mean, it's really great to be able to talk to you all and the students. And just as a small closing note, at our institute here at the Helmholtz, we have 4 Zavarians. So yes, it's hopefully going to inspire a lot more students. And if you all are looking to work in
Harshita is there. Harshita is
sorry?
there. Harshita.
Arshita, no, I'm sorry, I don't know her name, but we have Chandadish Ghosh from chemistry. He's my batch. He is here, as well as another student from computer science who's working as a bioinformatician. And I forget the other student. She's very new, but yes, she is also here. So we have 4 students at the Hiri from Xavier's. Yes.
Harshita is in MPI.
A girl has gone to your institute very recently.
Yes.
Yeah, I know.
Yes. Yes, but it was great. Thank you very much.
It was mesmerizing. Your talk was just simply mesmerizing.
Thank you, sir. Thank you.
Long way to go, Daniel.
Long way to go. Yes, sir.
Thank you so much, sir, for such a wonderful session. This brings us to the end of our first technical session of the day. We will now move on to the second technical session of the day, which will be taken over by Srinjika and Borsha.
A very good evening.
Just a moment. Before the second technical session starts, I would request all our students to join this session. This is for their benefit only. These sessions are being arranged. Modern Trends in Microbiology, we are just the organizer, but the benefit is for yours. So if you don't join, you will be missing. So kindly, please spread across this message that students should join this particular seminar.
So, should I continue?
Yes, please.
A very good evening to one and all present. I, Srinjika Veronica Pandey, along with my mate, Borsha Bokshi, would like to welcome the respected judges, the participants, and the audience to Technical Session 2 of Modern Trends in Microbiology, Chapter 18, Day 2, Presentation by Undergraduates. The judges for this technical session are Dr. Joydeep Ghosh and Dr. Kasturi Sarkar. All the participating teams are requested to keep the following instructions in mind. Each team would be given 10 plus 2 minutes to perform, 10 minutes to present, and 2 minutes to answer questions. The warning bell will be rung at 8 minutes. Final bell will be rung at 10 minutes, after which the floor will be open for questions. Participants are to turn their camera on during their presentation. We shall have the first presentation of Technical Session 2 by Borsha Bokshi and Deshna Baid.
1 minute. Have the judges joined? JG sir, Case ma'am.
Yeah, I'm there.
Yes, yes.
Thank you, teacher.
Is my screen visible?
Yes.
A very good evening to one and all present. Myself, Varsha Bukshi, and my friend Vishnagar will be now presenting our topic. COVID-19 pandemic is a defining global health crisis of our time. Every day, people are losing jobs with no way of knowing when normality will return. But the most tragic loss is the death of someone here. One such cause of death is our today's topic of presentation, cytokine storm of COVID-19 and its cure.
The term cytokine is derived from a combination of two Greek words, cyto meaning cell and kinos meaning movement. Cytokines are cell signaling molecules that aid in cell-to-cell communication during immune responses and stimulate the movement of cells towards the site of inflammation, infection, or trauma. Cytokines are small proteins that are crucial in controlling the growth and activity of immune system. When a harmful pathogen intrudes, the first immune cell detected releases messages in the form of cytokines. Receptor engagement triggers intracellular signaling cascades leading to an altered gene expression in the target cell, which lead to the biological effect. Cytokines isn't only restricted to any foreign pathogens entering the body, but also to the brain cells that create a sickening feeling for the person to rest. Their mechanism of action are autocrine, paracrine, or endocrine depending upon the nature. There are various types of cytokines in the body. Interleukins that are produced by the leukocytes such as lymphocyte and monocyte that are involved in the activation and differentiation of immune cells. Chemokines that play an integral role in chemotaxis. The other type of cytokines being interferons that interfere with the replication of bacteria and viruses entering the host body. Or the tumor necrosis factor, the TNF is a cytokine type that causes the death of a cell by interacting with the tumor cells. The most striking type of cytokine is the colony stimulating factor that activate intracellular signaling pathways which cause the cells to proliferate or differentiate into specific kind of blood cells. The COVID-19 disease is still that name that haunts us even in our deepest of sleeps. COVID is the reason for students like me and Varsha and others of my batch to not give an MTIM presentation on the famous SXC podium, but in front of our laptop screens. COVID-19 virus began in December 2019 near the Wuhan city, Hubei province, China, and has reached to more than 150 nations, including China itself, that has resulted in the World Health Organization to declare this disease as a pandemic. With the total worldwide cases confirmed to be 228.5 million and total deaths as 4.6 million by Johns Hopkins University. Now, what is the most strange about this virus is its structure. Even though it is a medium-sized virus, its RNA genome consists of 29,903 nucleotides, with the largest genome being concentrated for its structural genes. It is enveloped positive stranded RNA, and the mRNA is encased in a nucleocapsid. The lipid layer is its minus point, that is, and of course a plus point to us individuals, because that's where we attack the virus by detergents and soaps to kill it. The name corona was given to it because of the crowns for spikes that are made of glycoprotein spike peptometer. They allow it to attach itself to the human cell receptors in the upper or the lower airway. The virus spreads from an infected person to a healthy person by the respiratory droplets spread to the healthy person and enters its nasal passage into the lungs via the bronchi, bronchioles, and finally to the alveoli. What happens inside the alveoli is what my friend Varsha will take you through.
Alveoli are the tiny balloon-like structures, as you can see in the slide. On the surface of the type 2 alveolar cells, the angiotensin-converting enzyme 2 receptors are present. Now, upon the entry of the viral particle inside the alveoli,
The ACE2 receptor is present on this surface. The S-glycoprotein of the virus binds to this ACE2 receptors. Now, following this binding, the viral particles enters into the cell cytoplasm, releases its RNA, and starts replicating. With the help of the host ribosome, it synthesizes all other membrane proteins and envelope components. Upon assembling all the components together, multiple copies of the viruses are produced. These viruses are released into the alveoli mainly by bursting the cells, resulting in irritation and production of symptoms like breathing difficulty, dry cough, etc. Now, in response to this viral infection, our body produces IgM and IgG antibodies, which stimulates the humoral and cellular immunity. They, in turn, mediate the production of pro-inflammatory cytokines to fight the viral infection. But in some COVID-19 patients, a sudden acute increase in the circulating levels of different pro-inflammatory cytokines, mainly being interleukin-6, interleukin-1, and tumor necrosis factor alpha, results in a deadly local and systemic response known as a cytokine storm. Now, the sudden increase in the cytokine levels causes inflammation and edema that results in severe lung damage due to pneumonia associated with acute respiratory distress syndrome. Damage to the renal tubules may also lead to the development of cardiorenal syndrome type 1. Patients with underlying cardiovascular conditions can also develop cardiac injury, irregular heart rhythm, myocarditis, or pericarditis. Skeletal muscle damage is also observed in some patients. The cytokine storm can lead to multi-organ failure and had already resulted in the death of many COVID-19 affected patients. Now, in case of young adults, there is generally a balance between the pro-inflammatory and anti-inflammatory cytokines. But what happens in elderly adults is that the level of pro-inflammatory cytokines is generally increased, and the level of anti-inflammatory cytokines is generally decreased due to many age-related processes like alternation of ACE2 receptors, excess production of ROS, vitamin E deficiency, etc. This phenomenon is known as inflammation, and thus the elderly adults are more prone to develop cytokine storm.
The diagnosis of cytokine is a very difficult task because of the symptoms it shows, and it is only traceable when a greater damage has been done. The following on your screen are a few diagnostic processes which can be used, with them, the most accurate of them being one, that is analysis of the C-reactive protein in the body, the presence of higher ferritin levels, and third is the presence of interleukin-6 in the bloods at a higher level. Now, what has been added to this is the medical imaging. Medical imaging of chest is also a vital tool for the diagnosis of COVID-19. That's because it majorly marks the cytokine inflammation. Second, the treatment of cytokine is one of the most challenging affairs because of the lack of conventional diagnostic method and also because of its various types and each type having individual drug sensitivity. It is difficult to maintain the required level in the body after the cytokine storm is detected. The only possible treatment line in this disease is the control of the ongoing cytokine production and to resume the host homeostasis. However, appropriate terms of treatment depends upon age, immune status, and the comorbidities of the individual person. To curb cytokine storm, the major principles used is inhibition of pro-inflammatory cytokine production, like the IL-6, which is done by tocilizumab, chloroquine, and hydroxychloroquine, along with a few types of corticosteroids. IL-1 can also be suppressed by colchicine and the TNF-alpha inhibitor. Along with that, blood purification methods and intravenous immunoglobulins are promising as well.
Now, the QTY code is a modified protein concept which has a potential for the treatment of cytokines. In this, the hydrophobic amino acids missing isoleucine, valine, and phenylalanine are replaced by hydrophilic glutamine, threonine, and tyrosine to make insoluble protein soluble, and thus the detergent-free equivalents of the membrane proteins can be designed. These variant receptors almost have the same physiological characteristics to those of the innate receptors. Mainly 6 variants of interleukin receptors, chemokine, and interferon receptors are produced by this method. These receptors are mainly fused to the Fc region of IgG protein to form antibody-like structures. These antibody decoy act as a decoy to dampen the excessive cytokine levels.
APA club back filtration system uses these variants for rapid removal of cytokines. They mainly act as molecular sponges that soak up excess level of cytokines, but it had only been tested in mice. And human clinical trials have just recently been started. Thus, we come to a conclusion that cytokine storm is a leading side effect of cellular immunity, which is a potential threat for the COVID-19 affected patients. To alleviate the symptom and treat the disease, it is very much essential to remove excessive cytokines efficiently and rapidly. The irony is, when our body needs our immune system the most, we're trying to find ways to reduce its effect to prevent any further casualties. These are the best references from where we took our help. And thank you all for your patience here.
Thank you so much.
Very nice presentation. Very good.
Thank you, ma'am.
Okay, now I have a question. So you mentioned about IL-6, right?
Yes, ma'am.
Now, what kind of treatment or treatment was applied against IL-6? Can you name IL-6 blocker?
Yes, ma'am. Tocilizumab. It's a monoclonal antibody that's used to prevent.
What is it? Antibody against IL-6?
IL-6 receptor. It is an agonist.
Okay. And IL-6 is a pro-inflammatory cytokine. Can you name an anti-inflammatory cytokine?
IL-10, interleukin 10.
Okay. Okay, thank you.
Thank you so much.
Jeji sir.
I have no question. It was a nice presentation.
Thank you, sir.
Thank you, professors. Thank you, Borsha and Deshna. Up next, we have the presentation by Anvesha Datta Chaudhary and Kinjal Chandra.
I hope my slide is visible.
Yes, Kinjal, you are both visible and audible.
Thank you so much. Today I, Kinjal Chandra, along with my fellow classmate Anvesha Datta Chaudhary, have prepared a presentation on the topic Microbes, the Future of Sustainable Fashion. Since the title of our presentation itself makes mention of the word sustainability, let us touch upon its meaning. The Brundtland Commission of the United Nations in 1987 defined sustainability as meeting the needs of the present without compromising the ability of future generations to meet their own needs. Next, let us see why there is a pressing need for the fashion industry to embrace sustainability. According to statistical data, the fashion industry is the second most polluting industry in the world after oil and gas. The accelerated polluting habits of the clothing industry is attributed to fast fashion, a phenomenon that involves exploitation of cheap labor and utilization of massive amounts of energy and water to introduce new trends in the global market every season. In 2015, the global textile consumption was estimated at a shocking 95.6 million tons. With the advent of fast fashion, clothing has become disposable, the quality of clothing has regressed, and we have lost the skill and time to mend damaged clothes, leading to mass production of textile waste. In the following slide, we are going to discuss two plausible methods using which sustainable fashion can be attained, which are to resell, recycle, and refurbish old garments alongside proper management of textile waste, and to develop eco-friendly and biodegradable alternatives to clothing and textiles. Interestingly, microbes can help in achieving both of the aforementioned goals. A bacterium, Ideonella sakaiensis, is capable of digesting plastic PET fibers, that is polyethylene terephthalate, which is ubiquitous in synthetic textiles. Synthetic materials may otherwise require a minimum of 200 to 300 years to fully decompose in the soil. Now, over to my friend Ornisha.
Thank you, Kinjal. Now, microbes are being experimented with to revolutionize fashion and tackle the pollution conundrum. Many dyes used in the treatment of cotton, for example, azo dyes, are highly carcinogenic and toxic. Microbes can be used to develop a range of fabrics and dyes as started by Algae Life, which is a German-Israeli firm used algae for this purpose. Similarly, Faber Futures, a firm under Natsai Odrich Chiesa, also harnessed the phenomenon of fermentation using the bacteria Streptomyces silicolor, which is capable of yielding navy blue and bright pink or red pigments under controlled growth conditions. This method requires minimal water and is completely eco-friendly. The picture alongside shows Chiesa holding up a fabric that has been dyed in patterns using the same phenomenon. With augmented technological advancement, mankind has developed different kinds of fibers by manipulating microbes. C-Cell, a firm retails apparel, which is made from a mixture of brown algae, Oscophyllum nodosum, also known as the knotted rat, and lyocell, which is a form of soft wood pulp. The fabric is comfortable, biodegradable, and eco-friendly in nature. Spider silk, a material known for its strength, lightweight, and flexibility. It is not possible to farm spiders at an industrial level. A method involving recombinant DNA technology uses Rhodovulum sulfidophyllum, a marine photosynthetic purple bacterium, to yield artificial spider silk. A brand, AN Silk, recently collaborated with Adidas, the multinational sportswear company, to manufacture bioengineered sneakers using spider silk, the picture of which can be seen alongside. Using fungal mycelia as alternatives, with great innovation, Anila Huitink of NEFA also developed a mycelial fabric which leaves behind zero waste and is completely biodegradable. A prototype of mycelial jacket prepared by NEFA can be seen alongside. Besides, scientists at Bolt Threads also developed a fabric named microsilk, which is made from lab-grown spider webs. The material is the fruit of genetically modified yeast spliced with spider genes. The same fabric was also used by Adidas in collaboration with renowned fashion designer Stella McCartney for a range of bioengineered sportswear. Now, over to Kinjal.
Thank you, Anvesha. Probably the most exciting and remarkable development in the field of bioengineered sustainable fashion is that of vegetable leather developed by Suzanne Lee of BioCouture. Her initial interest focused around kombucha, a symbiotic culture of bacteria and yeast, also known as SCOBY, which is used to brew tea. Kombucha is also consumed widely as a detoxifying drink in many Asian countries. The yeast component of kombucha is identified to be Zygosaccharomyces bailey, and the bacterial counterpart is known as Combogati-Bacter xylinus, which was formerly known as Gluconacetobacter xylinus. This bacterium is popular for its remarkable property of forming cellulose. The bacteria forms a dense mat of nanocellulosic fibers on the surface of the SCOBY. This layer, once removed and dried, resembles animal leather in look and texture, hence gaining the name vegetable leather. Now, let us look at the pros and cons of BioCouture's microbial leather. This fabric can be grown to shape in completely natural media, as we can see with the 3 jackets. It is flexible and can be stitched to form self-adhesive seams and can be composted at the end of its useful life. Besides, the process uses very little resources and almost no harmful chemical dyes. Also, waste materials are used as raw materials for this procedure. However, the material is highly hygroscopic in nature and undergoes unsolicited decay over time. Now, I'll hand over the final part of our presentation to Annisha.
In this slide, I'll be discussing how nanocellulosic fibers are generated from simple microorganisms spanning over about 5 steps. In the beginning, the microorganisms required to produce the fabric are sorted and stored in a freezer at optimum temperature and pressure. To activate and increase the level of multiplication among the bacterial colony used and the metabolism of the bacterial colony, they are transferred to flasks and later placed on an agitator where they are pre-cultured. Later, the material is transferred to trays containing nutrient-rich solution. A cellulose membrane is observed on the surface of the static cultivation, measuring about 30 by 50 centimeters. Moving on, the film is then removed from the solution. After about 15 to 30 days, sterilized and dried in a process which takes about 3 to 7 days. The biofabric now produced is then stabilized in order to prevent it from absorbing much water, and is further washed and treated with different substances to increase its strength, malleability of the fabric being produced, and to eliminate odor from the material. Now, as the final step, the fabric is then dyed and is now ready to be in use. A pictorial representation of the entire process can be seen on the right side of the slide. The future of microbial products is promising, with consistent efforts to bring about a revolution in the industry and make it more cognizant and compassionate towards Mother Nature. Researchers at Biologic USA are engineering intelligent fabrics that can effectively respond to environmental conditions surrounding an individual. They analyzed a bacteria widely used in Japanese cuisine known as Bacillus subtilis natto. These bacteria respond to the environmental humidity, expanding or reducing their size. A biofilm was created from bacteria, and layers of it were also printed, known as the second skin. The technology and machinery to produce such fabrics is very exclusive as of now, and in order to hit the commercial scale, it will require great capital investment and human employment. Hence, these fabrics will first be available only to the luxury market. However, researchers are hopeful that in future, not only bioengineered fabrics, but also other biofabricated products like perfume, tissue jewelry, and fungal furniture will also be available in regular stores. With this, we finally come to the end of our presentation. These are some of the references we used to prepare this presentation. Thank you all for giving us a patient hearing.
Good presentation, Kinjal and Amnisha.
Thank you, ma'am.
Thank you, ma'am.
I have a question that biofabrics, they are easily digestible, or they are digested by microorganisms mainly, right? So what about the longevity of these fabrics? How long can we use them?
Yes, ma'am, that is a concern because all of these are, like you said, these are biofabrics which will undergo unsolicited decay over time. But this is just this is just a step towards the direction, like a small step towards the direction. Like we mentioned, C-Cell retails apparel made from algae as well as softwood pulp, which is lyocell. So that is also contributing towards increasing the longevity of the material. So these are just tiny steps towards a better future of fashion, which is more sustainable.
Ma'am, can I include one more thing?
Yes, sure.
Yes, as I mentioned in my slide for nanocellulosic fiber formation, after the biofabric is produced, we are using various organic substances in order to increase the flexibility and malleability of the fibers. So along with producing the biofabric, we are also using different substances which will maintain the malleability and the longevity of the substance. Now, most of the ideas and all are not present on the web because of international intellectual property rights. So because the theory might get stolen from the web, because of IP rights, they are not yet available on the web. But as much was available, ma'am, we used it and we made this presentation.
What happens if we wash them with detergents and then iron them? Will they remain the same?
Ma'am, apparently, according to the results on the web, they will remain the same. It won't harm much. Okay, but as I already mentioned, not much information was provided on the websites because of the fear of their idea and technology or machinery getting stolen because of the IP rights. So that is exactly why we did not gather much information on this topic as such. But as far as it was written on the websites and wherever we gained our knowledge from, it was written that nothing of that sort will happen. It will be longeable. Sorry.
Also the fabric that we mentioned, especially the sea cell fabric and the algal fabric, they are completely resistant to moisture and ironing and heat because they are in combination. However, the materials that are completely produced from the biological sources are a bit difficult to handle. So they are still being experimented with. This is at a very nascent stage now. Hopefully, we will have more biofabricated products besides clothing in the future.
Yes. Okay, good.
Thank you, ma'am.
Yeah, it was a very interesting topic, and I have no questions.
Thank you, sir.
Thank you, professors. Thank you, Kinjal and Anvesha. Now we shall have the presentation by Falguni Orao and Indrakshi Majumdar. Over to you, Falguni and Indrakshi.
Just wait a second. Is my slide visible?
Indrakshi, you are visible.
Is my slide visible?
Not
okay,
yet.
slide is not visible. Okay. Wait for 1 minute. Can I just reach once again?
Yes, please. Yes, it is visible now, Indrakshi. Please proceed.
Okay. Okay. I'm gone. Good evening, professors and one and all present here. Today, I, Indrakshi Majumdar, and along with me, Falguni Orao, we are presenting on the topic cystic fibrosis. First, let us talk about what is cystic fibrosis. Cystic fibrosis, that is CF, is a progressive serious genetic condition which leads to severe damage to the respiratory and digestive system. This damage results due to the buildup of thick, sticky mucus in organs like lungs, liver, pancreas, and small intestines. And due to persistent lung infection, it limits the ability to breathe over time. Most children are screened for CF at birth by screening method and maybe by the age of 2. Next, we'll move on to the symptoms of CF. The thick, sticky mucus associated with CF often ends up blocking the passage that brings air in and out of the lungs. Some of the major respiratory symptoms are wheezing with a persistent cough that produces thick mucus or phlegm, shortness of breath while exercising, recurrent lung infections. This abnormal thick mucus also ends up blocking the channels that brings enzymes from the pancreas towards the intestines. Some of the major digestive symptoms are greasy, foul-smelling stools and constipation, nausea and loss of a swollen abdomen. Next, we move to the fibrosis occurs due to the defect, that is mutation in CF gene, which is the cystic fibrosis transmembrane conductance regulator gene. And this ends up changing the protein which is responsible for bringing the chloride ion out of the cell. So the chloride ion unable to the cell surface and attract water towards it. This results in the thick, sticky mucus in various organs. And also the solid concentration in the sweat. These lead to a lot of infections, inflammation, respiratory failure, and many such complications. More than 90% of CF patients have mutation in F508. It is a deletion of 3 nucleotides, which results in the loss of amino acid phenylalanine at the 508 protein. Next, move on to optimum respiratory sample for CF airway infection. Bronchoalveolar lavage, CF, is often associated with neutrophil-dominated airway inflammation. The sample suggests that the sequential analysis of the bronchoalveolar lavage fluid may have higher sensitivity for detecting the changes in inflammation changes in the CF patients. Expectorated or hypertonic saline-induced sputum is used for those patients who have trouble producing the sputum spontaneously. So this helps in breaking the ionic bonds in the mucus and hence helps reducing the viscosity of mucus when hypertonic saline is added to it. Curved swabs help in providing additional information for direct antibiotic therapy in young non-expectorating CF patients. Oropharyngeal swabs is used for the culture of Pseudomonas aeruginosa only. Next, we'll move on to the role of ground stain and bronchotest methods for the evaluation of CF respiratory samples. The specific use of gram stain is used for evaluating both adequacy and morphology of pathogens, and it has excellent sensitivity, specificity, and predictive values of bacteria as compared with microbiological culture techniques for the prediction of specific organisms. One of the other techniques that has been suggested for the evaluation of sputum quality is the use of colorimetric card. For example, the broncho test by Williams Medical, which is a visual method to assess the sputum color. Thank you. I will be handing it over to Falguni for the rest of the part.
Thank you, Indrakshi. Now I, Falguni Orao, is going to describe the other detection and treatment methods of CF. Use of selective media and dilution methods for bacterial cultures of CF samples. CF airway infections are polymicrobial and contains both relatively rapidly growing organisms and those that are auxotrophic, making it possible for some organisms such as mucoid Pseudomonas aeruginosa to overgrow slower growing organisms. Selective media inhibits the overgrowth of non-target organisms and can be helpful in recovery of fastidious organisms. Current recommendations for the use of selective media in CF clinical microbiology are given as follows. For Pseudomonas aeruginosa, there are no such recommended media. For Burkholderia cepacia complex, BCSA and mass selective agar are recommended. For Staphylococcus aureus, chrome agar, staph aureus selective and differential agar and also mannitol salt agar are recommended. For Haemophilus influenzae, blood agar supplemented with hemin plus besit and chocolate agar plus cefsulodin are recommended. Organism identification. Once organisms are isolated in pure culture, some may readily be identified by appearances like colonial morphology, pigment production, growth, and temperature, and also combined with a few simple assays. However, because of many phenotypic changes that may be induced in the CF airway related to selective pressure and hypermutability, gram-negative CF isolates often require additional testing like PCR, Malditoff, MS, detection and identification of small colony variants Staphylococcus aureus. Organism identification by polymerase chain reaction and next generation sequencing methods. The clinical importance and infection control implication of specific bacterial organism such as Pseudomonas aeruginosa, Burkholderia cepacia complex, and Mycobacterium abscessus complex in CF make rapid and accurate identification imperative. The use of 16S rRNA PCR amplification and sequencing and real-time PCR amplification of multiple gene targets can significantly improve the accuracy of identification. Next generation sequencing is the term applied to sequencing strategies such as pyrosequencing, dye-termination, and ligation methods that enable sequencing of long stretches of DNA and analysis of the whole genome. The method have revolutionized sequencing by increasing throughput, affordability, and simplicity. Organism identification by Malditoff MS. Malditoff MS is a more recent and rapid method for bacterial identification and is being used routinely in many clinical microbiology labs. During Malditoff analysis based on POF information, a spectrum called peptide mass fingerprint is generated and the identification of microbes is done by comparing the PMF of unknown organism with those in the database. The only member of the Burkholderia cepacia complex that can reliably be identified by Malditoff MS is the Burkholderia multivarians. Detection and identification of small colony variants of Staphylococcus aureus. Genetic variants of Staphylococcus aureus called small colony variants emerge during chronic CF infection and are associated with poor clinical outcomes in a pediatric population. SCV grows slowly on other media due to metabolic defects. Staphylococcal selective agar is necessary to avoid overgrowth of other organisms, and prolonged incubation is critical to detect growth and hence used. To detect Staphylococcus aureus SCVs, it is useful to subculture Staphylococcus morphotype medium onto both a supplemented medium that will readily support normal colony growth and a medium on which small colony growth is demonstrated. Antimicrobial therapy in treatment of cystic fibrosis. Much of the improvement in survival of individuals with cystic fibrosis is due to advancement in antimicrobial treatments. New aerosolized antibiotic formulations have recently been introduced, such as inhaled aztreonam, and others are in development like inhaled levofloxacin and liposomal amikacin. Licensed dry powder formulations such as tobramycin, inhalation powder, and cholestyramine are also used. Antimicrobial resistance can still develop, which can be mitigated by using non-antibiotic treatments, antibiotic adjuvants such as gallium, antimicrobial peptides, and antibiofilm compounds such as alginate oligosaccharides and GALI. Vaccination strategies and antibody therapy using IgY against Pseudomonas aeruginosa have also been attempted to prevent initial infection. Conclusions. The conventional therapies for CF involves monitoring pulmonary function, airway clearance, and infection control. For correcting of CFTR defect, a two-step therapy including lumacaftor and ivacaftor can be used. These act as chaperones during protein folding that increases CFTR protein towards the cell surface. Additional molecular correcting agents are under development. Research on nanoparticles as nanocarriers for drug delivery to the lungs by inhalation has opportunity to strongly modify symptomatic treatments for CF patients.
These are things that have been used for the present. You all gently listen to us.
Thank you, Indrakshi and Falguni.
Nice presentation. Why did you choose this topic? Because cystic fibrosis is known for many years, right?
Yes, actually, the thing is that cystic fibrosis is true. It is known for many years, but actually, it is a very rare disease. So there are different sorts of people actually don't even know about this, and not only that, because it was not actually curable before, right? And now we are eventually moving towards its cure. Like we are trying to look towards the transmembrane sort of defects and trying to improve the transmembrane defects so that there is even more effective way we can look at it. And not only that, we are also looking at the different sorts of organisms that affect the secondary sort of infection that affects us, that can also be decreased because of these new techniques that is upcoming in these years, in the upcoming years too.
Yes. Identification of the microorganisms is more accurate nowadays. That's true, with the help of the new techniques. Right? Okay. Nice presentation.
Yes, ma'am.
Yeah,
Thank you, ma'am.
Yes, it was nice. I just have one very basic question. What is the frequency of cystic fibrosis in human population?
So frequency, maybe in our country, if I just say, since it's very rare, so maybe it is found in like 1 person in every 10 million people,
1%?
or maybe even more. One in 10 million people, or even more.
And it is more popular in European and South African countries.
Yeah, exactly. It's not much in India, but American, European countries, it is more prevalent.
Okay, I'm done.
Okay.
Google is saying 1 in 2,500 to 3,500 white newborns, and 1 in 17,000 African Americans, and 31,000 Asian Americans.
Yeah.
Not in million.
130. No, I was only specifically talking about India, not in general, not in Asia as such, but.
Okay, thank you.
Thank you.
Thank you, ma'am.
Thank you, ma'am and sir.
Thank you, professors. Thank you, Indrakshi and Falguni. Up next, we have the presentation by Ushmita Chakraborty and Snata Pandey.
I'll be sharing my screen now. Hope it's visible.
Yes, it is.
Yes, you are both visible and audible, and so
Thank you.
Good evening and welcome to the presentation on fungi in the human gut microbiota. It is well known that the mammalian intestinal microbiome constitutes a complex community of prokaryotic and eukaryotic microorganisms, including the prokaryotic bacteria, cellular viruses, eukaryotic protists, rarely some members of the domain RT, and a topic of today, the eukaryotic fungi. Structural and metabolic signals derived from this microbial ecosystem engage in crosstalk that helps define the trajectory towards host health or disease. The gut microbiome is involved in proper sleep, mental health, efficient immunity, and have been implied in obesity, autoimmune diseases, and so on. Early stages of gut microbiome establishment are more dynamic when compared with other microbiomes, and environmental factors can influence the composition and diversity of this community as well as the trajectory of its assembly. Although it is extensively known to us how bacteria living in our guts aid our normal functioning, studies on gut microbiome, that is, fungi in the gut microbiota, have escalated only very recently. It is increasingly recognized as a critical player in the maintenance of health and homeostasis of the host. Accumulated evidence also suggests the significant involvement of gut fungi in maintaining host dynamics, in maintaining a vast ecosystem with several interactions with the gut bacteria, and the potential of utilizing fungal metabolites in clinical applications. And hence today we will give a summarized view of our gut microbiome. But why is fungal microbiota not as thoroughly discussed? This is because of two basic reasons. They are much fewer in number. In fact, only about 0.01% to 0.1% of our gut microbiome is constituted of fungi. Secondly, fungi have been studied using culture on selective media under a microscope. It is only over the last few decades that culture-independent methods of molecular biology like PCR and next-generation sequencing, that DNA-based detection of fungi have been developed, which are able to shed some light on this less studied diversity of fungal communities of the human gut. Now let us look through the composition of fungi in the gut discovered so far. As shown in the slide, fungi makes up the rare biosphere in our gut. As our guest lecturer explained to us, gut bacterial and fungal communities inhabit similar intestinal habitats, and fungal bacterial correlations have been identified in human cohort studies. Fungi are a ubiquitous component of the mammalian microbiome. By far, the 0.1% of microbiome in our gut predominantly have the members of phylum Ascomycota and few members from phylum Zygomycota and Basidiomycota. The most predominant genera in the intestinal niche are Malassezia, Candida, and Saccharomyces, although there also occurs Cryptococcus, Aspergillus, and Bladosodium. The composition varies from human to human, and the exact reason behind this is not clear. It is hypothesized that distinct colonization early in life from mother during birth and different environmental exposures such as antibiotic use and diet bring about such diversity among individuals. Infants have most dynamic communities. All are seen to carry Candida albicans. Although the pure number of fungi in infants is attributed to the young age when they have not been fully colonized by all the fungi in adults. It has also been seen that fungal richness is higher in healthy females as compared to healthy males. Now, I would like my friend Snata to continue.
Thank you, Ashmita. Now let me walk you through some of the reasons as to why we need to study the gut microbiome in the first place. First and foremost, nobody is fungus free. Fungi occur as common cells in our guts as a part of our holobiomes, as Ashmita mentioned, and they are actually very important in the maintenance of host homeostasis as well as of a healthy gut ecology. They do not remain isolated, but they extensively interact with other gut communities, and in doing so, they benefit us, provided, of course, they occur in low amounts. For instance, Candida albicans, which is a well-known human pathogen, is actually a permanent resident in the guts of the healthiest individuals, and provided it occurs at its colonization level, which is 1 to 2% of the total fungal count, Candida is actually performing functions for us, including, of course, training the host immune system, as well as fermenting carbohydrates in our diet while interacting with the Archaea methanobrevibacter. It does so while interacting with the Archaea methanobrevibacter, and the carbohydrate fermented products are actually eaten up by our gut bacteria as food, and these bacteria in turn help to keep the Candida levels under check. So these are examples of fungal bacterial and fungal archaeal interactions. Besides this, we also find fungal interactions wherein Saccharomyces and Pichia are independently helping to keep Candida levels under control. Mixed species biofilms are formed in a number of diseases, including colorectal cancer. For instance, in Crohn's disease, which is one of the inflammatory bowel diseases, it's been seen that Serratia marcescens, Candida tropicalis, and E. coli are sticking together to form these interesting digestive plaques or mixed species biofilms. Besides this, gut fungi are a very crucial constituent of the gut-brain axis besides the gut bacteria. This is because they obviously trigger the gut immune cells to produce a number of neurotransmitters and cytokines, which are affecting the emotional, cognitive, and limbic functions of the brain. Moving on to the second reason why we need to study gut fungi is because changes in the gut fungal composition have been implicated in a number of diseases. For instance, returning to Crohn's disease, we have seen that at the phylum level, Basidiomycota to Ascomycota ratio gets increased. At the genus level, Candida levels increase while Saccharomyces levels fall, and at the species level, Malassezia globosa numbers increase while Malassezia sympodialis numbers fall. In ulcerative colitis, which is another form of IBD, again, Malassezia are seen to have a negative correlation with many gut bacteria. Irritable bowel syndrome has shown, in the case of irritable bowel syndrome, it's been seen that Kazakhstania turicensis is one of the very important markers. Kazakhstania is, of course, one of the coffee risks. Besides IBD and IBS, in anxiety depressive behaviors, in obesity, or any sort of an increase or decrease in body weight, which is not related to lifestyle or diet changes, in the case of atherosclerosis, alcoholic liver disease, and so on, it's been seen that gut fungal dysbiosis has been implicated. Moving on to the neurological diseases, in the case of multiple sclerosis, it was hypothesized, though not yet proven in 2010, that Aspergillus and Candida housed in the GI tracts of MS patients are actually producing some mycotoxins which are affecting the astrocytes and oligodendrocytes, the blood-brain barrier, and degrading the myelin sheath. Gut fungal dysbiosis, the dysbiosis of Saccharomyces and Aspergillus has been seen in autism spectrum disorder patients, and such dysbiosis is also seen in Rett syndrome patients. The link between gut fungal alteration, composition and its alteration, and Alzheimer's disease was confirmed in 2015, wherein it was seen in murine models that administration of antibiotics over a period of 5 to 6 months resulted in obvious alterations in the gut communities. This changed the chemokine and cytokine levels in the blood of these models, which resulted in changes in the solubility of the amyloid beta plaques in the brains, which resulted in changes in the morphology and reactivity of the microglial cells, and thus altered the progression of Alzheimer's disease. Pancreatic tumors have been seen to be infiltrated with Malassezia, which are producing some glycoproteins on their surface, which are being identified by mannose binding lectin, which is triggering the C3 protein to initiate a complement cascade and trigger further tumor growth. Moving on to the third reason why we to study gut fungi is because a healthy gut fungal composition is very important in prevention of systemic infections by limiting pathogen growth, maintaining barrier function, as well as by training the host immune system. It's been seen that due to antibiotic consumption, if there is bacterial eradication taking place in our guts, then we become susceptible to a number of diseases. However, fungi in our guts can effectively replace these bacteria and render us less susceptible to these diseases. For instance, monocolonization by Saccharomyces and Candida has been seen to be protective to colitis and influenza A virus because of the presence of mannans in the fungal cell wall. Interestingly, individuals who are suffering from any sort of individuals who are completely healthy and who any history of a fungal infection, and individuals who do not have any mutation in their card9 gene are seen to carry some antifungal antibodies in their bloodstream, while individuals who are not having any mutation in their card9 gene and individuals who are immunocompromised are not seen to carry such antifungal antibodies in their bloodstream. This is indicative of the fact that intestinal fungi are acting as intestinal fungal vaccines. Finally, The gut fungal microbiome and its study can be extensively used in therapeutics. Already, antifungal compounds like miconazole and fluconazole are showing therapeutic benefits in MS and autism spectrum disorder. Interestingly, in individuals suffering from ulcerative colitis and those who are being treated with fecal microbiota transplant, among these, those who are having high candida levels in their guts prior to the treatment are showing better response to the treatment as compared to those with lower candida levels in their guts prior to the treatment. And the treatment in turn is helping to lower the candida levels in the guts of these individuals. Finally, fungal vaccines are a topic of heavy research today, although no fungal vaccine has yet been licensed. Now, I would like Ashmita to conclude our presentation.
Thank you, Snata. To conclude from the accumulated evidence, gut microbiome has an indisputable role in homeostasis and disease development, despite considering only a small proportion in the gut. Nevertheless, the journey of uncovering the mystery of gut microbiome must be continued. Eating the right amount of food items and lifestyles with less stress and more sleep help to keep the number in control. Meditation, yoga, and relaxation are very crucial in preventing this biases, thus maintaining a proper physical and mental health. Thank you for your patient hearing. Here are some references for further reading.
Thank you.
If I am under antifungal therapy for a long time, can it actually change the bacterial community of my gut? Got my question?
And yes, sir.
Yeah.
Well, the reverse is true because antibiotic can change the bacterial composition, and that can obviously have overgrowth of candida and so on. But well, antifungals.
Think in this term, caspofungin or the recent antifungal therapy, actinomycin, if these drugs are given, actinomycin D.
So I'm not aware of this, sorry.
But actually, long-term change can actually will reduce the signaling molecule. So if there is a positive impulse towards bacterial growth, that will stop. So naturally, this will reduce the some of the bacterial population which are dependent on the fungal signaling. That might be a reason.
Definitely.
Yeah.
Because as I said, these are extensively interacting with each other. So if there is a change in one, there
So
is
you
definitely supposed to be some
apply your
alteration in
mind,
another.
you apply your brain and answer this question.
Yes, sir.
Every
Yes.
question do not have a direct answer. Yeah.
Thank you, sir.
Okay, good presentation, but I think the content was too much, right?
Yes, ma'am.
Yes. Okay, no question. Thank you.
Thank you, ma'am.
Yeah, no questions from me as well. It was nice.
Thank you, sir.
Thank you, professors. Thank you, Snata and Ashmita. Now I would like to hand over to my dear friend, Borsha. Borsha, please take over.
Next group of presenters are Bharati Barnwal and Rishabh Shah.
Am I visible?
Yes, you are.
Okay. Good evening, one and all. I am Paridhi Barnwal, and together with Rishabh Shah, would be discussing the roles of extremophile in the industrial sector. Extremophiles are organisms that thrive under extreme conditions, conditions that are difficult for other organisms to inhabit. The word extremophile is derived from the Greek extremus, meaning in extreme conditions, especially at the point of death, and philia is one of the 4 ancient Greek words for love. Most members of extremophiles are from the domain Archaea. A few are from Eubacteria and Eukarya. Now, Rishabh would explain the types of extremophiles.
The types of extremophiles present on Earth are Acidophiles, which are organisms that thrive in very acidic conditions, usually pH 2.0 and below. Alkaliphiles, which are organisms that survive in an alkaline environment at a pH of around 8.5 to 11 and optimally at 10. Halophiles thrive in high salt concentrations ranging from 0.3 to 5.1 molar. Thermophiles thrive in very high temperatures ranging from 41 degrees centigrade to 122 degrees centigrade. Hyperthermophiles are thermophiles that thrive at temperatures at or above 80 degrees centigrade. Psychrophiles, also called thiophiles, thrive in very low temperatures ranging from minus 20 degrees centigrade to plus 10 degrees centigrade. Piezo have optimal growth under high hydrostatic pressure. More operationally, an organism that has its maximum rate of growth at a hydrostatic pressure equal to or above 10 megapascal, which is equal to 99 atmospheres or 1450 psi when tested over all permissible temperatures. Oligotrophs can grow in an environment with very low nutrient concentrations, while xerophiles can thrive in areas with very low water availability like deserts. My partner Paridhi will speak about the industrial uses of extremophiles.
Lignocellulosic raw materials come from a wide range of natural sources and are an abundant renewable resource for the production of biofuels and value-added chemicals. They are considered to be better substitutes for lignocellulose pre-treatment in terms of pollution. Biological pre-treatment methods involving lacase have been reported to significantly improve the delignification of lignocellulose. Biological pre-treatment of biomass with extremophiles and lignin decomposition enzymes reduces the amount of degradation byproducts and the time required. The alkali and acid tolerant nature of extremozymes used is helpful for the process as well as complete hydrolysis of cellulose and hemicellulose at high temperatures. Bioethanol is often regarded as the most promising alternative and additive for gasoline among all biofuels. Treatment of biomass using thermostable cellulase produced by thermophilic zeobilis species have been shown to yield a hydrosilate that is readily fermented by Saccharomyces cerevisiae to produce 0.45 to 0.50 gram ethanol per gram of glucose with a 99% utilization of glucose. Thermophiles Caldicellulose rupta besky and Clostridium thermocellum have been reported for their potential to use cellulosic hemp as well as pre-treated lignocellulosic biomass as a substrate to yield bioethanol. The graph below shows the increase of use of biofuel over the years in different parts of the world.
Lignin is a complex aromatic heteropolymer and is closely associated with cellulose and hemicellulose, which are two major components of plant cell walls. Recent studies have found that some microorganisms have evolved metabolic pathways, which can provide a biological solution for high-value utilization of lignin to overcome the heterogeneity problem in the lignin appreciation of modern biorefineries. Several extremophiles have shown their potential for lignin valorization, such as the halotolerant and alkalophilic bacterium Bacillus ligninophilus L1, which can significantly degrade lignin at the optimum pH of 9 and produce aromatic compounds, including vanillic acid, vanillin, etc. Biomining, also called bioleaching, is a process in mining and biohydrometallurgy that extracts valuable metals from a low-grade ore with the help of bacteria or archaea. Extraction rates are around 90% from biomining, unlike the 60% from traditional heap leaching. Metals like silver, gold, nickel, iron, and most commonly copper are extracted using biomining. The two main types of bioleaching are direct leach bioleaching and indirect bioleaching.
A novel application area in the medical field for extremophiles is the use of extremolites. The illustrations given below show how ectoin, an extremolite, is applied in the medical field. Another example of use of acidophilic extremolites are probiotics. Extremophiles are also known to generate antimicrobial peptides and diketopiperazines. Halocyanes peptides from halophiles found in Halobacteriaceae and Sulfolobus species, which are effective at killing alkyl cells, assist canines in recovering from surgery. Diketopiperazines found in halophiles like Naloterigena hispanica affect blood clotting functions. They also affect quorum sensing pathways, which are important in pathogens like Pseudomonas aeruginosa, which is a causative agent of pneumonia. This could be an alternative method for the countless drug-resistant Pseudomonas aeruginosa infections that occur each year.
An enzyme called Taq polymerase, which is extracted from the thermophilic bacteria Thermus aquaticus found in hot springs, is used during PCR and does not get denatured during the high temperature denaturation phase. This is because of its thermostability. This makes the entire process very efficient and cost effective, as there is no need to refill the polymerase enzyme after every cycle, and thus no need for constant hands-on operation.
The table given shows us the uses and potential uses of extremozymes and their source. Now, we shall study the industrial uses of 4 extremophiles from the table, namely Anoxibacillus thermarum, Alteromonas, Micrococcus antarcticus, and Bacillus techilensis.
Anoxibacillus thermarum is a thermophile found in hot springs. The enzyme alpha amylase is extracted from it. In biorefineries, it is used for the hydrolysis of starch into simpler soluble products. The thermostable alpha amylase is preferred over that extracted from other non-thermophilic bacteria in large-scale biotechnological processes and biorefineries due to their high temperature tolerance. In the baking industry, it is used for improving the quality of bread. It is also used for clarification of beer or fruit juices and the pre-treatment of animal feed for the improvement in digestibility of fiber. Alpha amylase is present in 90% of liquid detergent formulations. This enzyme has stability at alkaline pH and high oxidative stability, thus maintaining the necessary stability required under detergent conditions. Beta-galactosidase, after immobilization, is used for the hydrolysis of lactose in milk. The lactose hydrolyzed milk is then used in preparation of flavored milk, cheese, and yogurt. This prevents lactose crystallization in frozen and condensed milk products and accelerates the acidification process of lactose hydrolysis, which otherwise is the rate-limiting step. A study that was conducted in 2019, where this enzyme was extracted from Alteromonas species ML117, is mentioned in the references. Micrococcus antarcticus is a psychrophilic bacterium found in Antarctica, useful for the psychrophilic beta-glucosidase enzyme extracted from it. In the flavor industry, beta-glucosidase is used to enhance the quality of plant-based food and beverages and to reduce the bitterness in fruit juices. It is also used in the tea beverage industry to increase the essential oil content, which further enhances the flavor. Additional beta-glucosidase enzyme is used to hydrolyze the glucosidic isoflavones present in soybean, which are present mainly in the inactive form of glycosides into aglycones. Aglycones exhibit higher biological activity and get absorbed faster and in larger quantities during digestion. Bacillus techilensis, strain armati, found in fecal soil, is used for the production of thermoalkali stable xylanase. It is used in the paper and pulp industry, where treatment with xylanase is done by substituting chlorine as a bleaching chemical with hydrogen peroxide and ozone. Thermoalkali stable xylanase can treat brown stock without acidification. This has resulted in reduced costs by 89%, better quality products with increased brightness and lesser pollution. Xylanase treated dough used in bakeries is more flexible, has improved gluten strength, increased moisture retention, and better shelf life. In fruit juice processing, xylanase is used as a part of the maceration process. Now, my partner Paridhi will elaborate on the challenges that are faced during the use of such extremophiles and their extremozymes.
The main challenge is to grow these extremophiles in the laboratory and develop bioreactors for the commercial use without knowing their optimal growth media and conditions. Advancements made in molecular biology of extremophiles are too limited to investigate the routes they adopt for themselves at molecular level under harsh environmental conditions. However, modern biology has made it easier for researchers to sequence the entire genome of bacteria and explore the system's biology approaches that enable extremophiles to cope with the surroundings. Reference libraries for the chemical properties of extremolytes would make it easier to screen a variety of them against specific diseases. These are the references we used to prepare this presentation. Thank you for your patience.
Thank you, Paridhi and Risha. I will request the judges to give their remarks.
Paridhi, it is very important to maintain eye contact. You should not have this habit of reading out. This exercise means that you speak maintaining eye contact.
Sorry, sir. I'll pay attention to this the next time.
So it was a very comprehensive overview of this topic. Thank you. No questions.
Thank you, sir.
Thank you, sir.
Okay, very nice presentation.
Thank you, ma'am.
Thank you, ma'am.
We were talking about biomining.
Yes,
Do the microbes have any role in arsenic toxicity?
Not that I'm aware of. No. Actually, the main thing that the microbes do is secrete the enzymes that make the chemical reactions undergo, which actually extract the metals far more efficiently than normal heap leaching. Yeah, that's.
Microbes can reduce arsenate to arsenite and can convert it into methyl arsenate. So it can even convert it into gaseous form. That is a unique role they play. Even tocopherol is including in some of the microbes.
Okay, sir.
Okay.
Thank you, sir.
Thank
you, judges. I will now call upon our next presenters, Goindri Ghosh and Rajeshwari Chakraborty.
Is my screen visible?
Yes, it is.
A very good evening to one and all present here. This is Rajeshwari Chakraborty.
And this is Oindri Ghosh. And today we are going to present on the topic, the role of microbes in human population dynamics.
So, if we talk about human population, there are currently a total of 7.8 billion people on this planet. And if the population keeps growing at this constant rate, then by 2050, the world will be inhabited by more than 10 billion people. But this ever-increasing population has a lot of ill effects on nature and mankind. Some of them are depicted in the adjoining figure, which shows that overpopulation leads to food and water shortage, degradation of the environment, faster climate change, poverty, and a lot more issues like rise in unemployment, high cost of living, outbreak of pandemics and epidemics, and the people become more likely to be exposed to infectious diseases and malnutrition. Over to you, Oindri.
Now, a very interesting fact about human population is that we humans have an exceptional age structure when compared to other animals. Our children remain dependent on their parents for an unusually long period of time, and our elderly live an extremely long time even after they have stopped procreating. Now, we all know that nature has its own way to stabilize its population. In most species, individuals die shortly after the reproductive phase, but humans are weird. We have an extra long senescence phase. But how? Many scientists at present speculate that microbes play a major role in shaping the human population. Yes, you heard it right. Our body has 10 times as many microbial cells compared to our own body cells. So isn't it obvious that the influence of these microbes on our body extends well beyond just aiding digestion and producing bodily odors? The human microbiota also aids brain development, reproduction, and defense against infections. A healthy and diverse microbiome is essential for natural, cognitive, and emotional processing as well. The human microbiota consists of the 10 to 100 trillion symbiotic microbial cells harbored by each person, which primarily includes bacteria in the gut. The gut microbiome includes bacteria, bacteriophages, viruses, fungi, protozoa, and archaea, and controls most of the physiological functions of our body. Over to you, Rajeshwari.
Now, coming back to how these microorganisms shape the human population. So basically, apart from all the functions mentioned previously, the microbes also control the fertility and mortality rates in human population. To test this idea, a group of scientists from New York and Vanderbilt universities conducted certain experiments under the leadership of microbiologist Martin Blazer and mathematician Glenn Webb. They presented their argument in a paper titled Host Demise as a Beneficial Function of Indigenous Microbiota in Human Hosts. In the experiments, the researchers created a mathematical model of early hunter-gatherer societies which represented an ancient population having isolated communities. And this population was divided into three categories: pre-reproductive or juvenile, reproductive, and post-reproductive or senescent. After this, they added mortality risks based on some particular microbial profiles. They monitored on how the population would respond to different combinations of fertility and mortality rates, and also developed a baseline case using the best estimates of these rates that they could find. The experiment was divided into two parts. Firstly, they added a risk factor based on Shigella. Shigella includes a family of bacteria which causes intestinal infections or shigellosis, which is very contagious. Shigella is also one of the leading bacterial causes of diarrhea worldwide. In another version of the experiment, Helicobacter pylori type mortality factor was added. Helicobacter pylori is a bacteria that coexists peacefully in people's stomach for most of their lives. It even has some beneficial effects, like it helps in regulating the levels of acid produced in stomach. But H. pylori is also a major cause of peptic ulcers and stomach cancer in elderly persons. Over to you, Indri.
Now, these experiments were based on some assumptions. Since the experiment was based on ancient population, it was assumed that people haven't changed much in the last generations. In particular, they had the capability to live up to 120 years, which seems to be the current maximum lifespan of humans. Of course, they had shorter average lifespan because of the larger number of sources of mortality that have been largely eliminated in modern society, with fewer outbreaks of infectious diseases due to improved sanitation and increased availability of food and modern medicines like antibiotics. Another assumption made was that microbes are passed between generations. Although mother-to-infant microbial transmission is quite evident, but how long these microbes subsist within a maturing human is still unclear. It was observed that on adding risk factor based on Shigella, the population crashed because Shigella increased the mortality rates in children, whereas addition of Helicobacter pylori type mortality factor stabilized the population. As we can see in the adjoining figure, that in older adults, there seems to be a decline in microbiota diversity compared to the younger members, which is also referred to as dysbiosis. The elderly individuals generally have lower numbers of firmicutes and bifidobacteria. These groups of bacteria reside in the human gut and help in digestion. And also there is an increase in bacteroidetes and certain proteobacteria, which are thought to play a role in the bowel disease. Over to you, Rajeshwari.
From this research, Blazer and Webb inferred that a true symbiont will be beneficial for us when we are young, but they prefer to kill us after we have stopped procreating. This might sound very strange because in symbiosis, both organisms involved are benefited. But when it comes to the benefit of the population, the symbiotic microbes want to keep the entire community of its hosts healthy, even if it comes with a cost to an individual host's health, because an unstable population will lead to the loss of their hosts, which will further lead to the destruction of their own population. The stabilization of human population on adding Helicobacter pylori type mortality factor, which increased death rates in the elderly, proves that evolution has favored microbes that target aging, so that more natural resources remain available to the procreating members. And this is how nature removes its elderly population with the help of the microbes, so that life continues to go on. Another fact regarding human population revealed through this research was that our fertility and mortality rates perfectly supports the unique age structure of the human population. Over to you, Indri.
Blazer and Webb study not only found a new path in the field of microbiology, but also its knowledge can be applied to design better preventives and treatments for many age-related disorders. Because we now know that the microbes of our own body, which had once been beneficial, can be the agents of mortality in later life. One such application of the knowledge might be seen in the making of probiotics with Bifidobacterium and Lactobacillus genera for elderly populations, because the number of these bacteria are decreased in them, but they needed in their body for proper digestion. We would like to conclude by stating that the symbionts we harbor in ourselves control the biological clock of aging in humans. Thus, this huge microbial world not only contributes a lot to our living, but also plays a major role in human population dynamics, thereby maintaining the growth of our species, which in turn is beneficial for the continuation of life on Earth.
These are some of the references which have helped us in making our presentation. And finally, we would like to thank everyone for lending a patient ear.
But Rajeshwari, I lent a patient ear to you, but you didn't maintain eye contact. That is a crime.
Sir, actually, I'm doing it on my phone, and it's like the angle is.
That's very important when you present. You see, you need to look at the screen. Now, if the microbiome is removed from a particular part of the body to another part, how will it adapt?
If the microbiome is removed from one part of the body
to other, suppose in a theoretical condition, the microbiome is removed to a new place. Suppose, say, for example, from the gut to the buccal cavity. What will be the initial ways by which it will adapt? Or can it adapt at all?
Suppose if the gut microbiota, which is well adapted in its own, if in the gut it plays roles like digestion and like production of some vitamins, but if it is taken to somewhere else, I don't think it will able to adapt in that way, because if it is I don't think so it will be able to adapt, because it is already adapted to its own environment. And like if archaebacteria is there, then it might adapt to various extreme conditions as well. But other bacteria, it might find, like in neutral environment, there are neutrophiles which are more more adapted to that environment. If we change the environment to acidophilic, then they will not be able to survive, I guess.
But they can always adapt through the formation of biofilm.
Sir, even if it does adapt to the other part of the body, but it won't be able to function that efficiently, which it was functioning in the other part.
That should be done.
Because the environment is changed, even if it adapts to it.
Precisely. Thank you.
Thank you.
Okay, nice presentation. What is the difference between microbiota and microbiome? Is there any difference between these two words?
We often refer to as gut microbiota.
I'm not sure if it is totally different, but microbiota refers to the microbes collectively, and microbiome
at a particular region, I guess. It's microbes present in the particular part, or like that, I think.
Okay, so go and check. Okay, nice presentation.
Yeah, it was a very interesting topic, and the presentation was also very good. And I have no question.
Thank you, sir.
Thank you, Rajeshwari and Oindri. Our next group of presenters are Orkuto Hore and Shubhodip Poddar.
Is my screen visible?
Yes.
Okay. Good evening, respected judges, professor, and all my dear viewers. The topic for today's evening from us is the obstacle and opportunities in a forward vision for cancer nanomedicine. So what is nanomedicine? Nanomedicine is an emerging key technology in the 21st century. Renowned physicist Richard Feynman said that the nanoparticle and other other nanoscale tools will improve the diagnosis and the pharmacological treatment of several human diseases, including cancer. So, in the tumor tissues, the fenestrated endothelial wall is giving access to particles from which the concept of enhanced permeability and retention effect has been introduced, which led to a new journey to the researchers. Now, moving on to the some of the distinguishing features by help of which the malignant tumors can be differentiated from the benign tumors. The malignant tumors shows invasiveness. That is, the malignant tumors have pore boundaries and thus they can penetrate into nearby structures. The next point is the malignant tumors can metastasize. That means they can easily get detached from the tumor, and they can move distantly via blood or to the lymphatic system. The next point is that the malignant tumors occur or recur more often, and they may recur locally, regionally, as well as distantly. The next point is that for the malignant tumor cells, the programmed cell death or apoptosis is checked, or it not occurs. And in case of malignant cells, they require more nutrition and oxygen supply, and which is termed as angiogenesis, and it is controlled by various activator as well as inhibitor molecules. Now, moving to the nanoparticles, which will be explained by my friend Orko Prabhu Roy. Orko, over to you.
Thank you, Shubhadeep. The first type is protein drug conjugated nanoparticle. The main problem with this is that biodegradable linker between protein and drug is destroyed by proteases. Nowadays, the linker maintains itself until reaching a target site, and this problem is almost solved. The next one is lipid nanoparticles. These nanoparticles are created by adding amphiphilic liquid to water or other hydrophilic liquids. These are spherical shaped with lipid bilayer. Common lab methods of creating liposomal nanoparticles are sonication, extrusion, reverse phase evaporation, and solvent injection. Now I am going to discuss about polymeric nanoparticles. They promote customization of many properties like molecular weight, biodegradability, and hydrophobicity. Generally, dense matrices with well-known degradation curves are seen. Some other types of nanoparticles are carbon nanoparticles, gold nanoparticles, silver nanoparticles, quantum dots, lanthanide nanoparticles, dendrimeric nanoparticles, and hydrogels. Now coming to the next topic, nanocarrier properties. At first, physicochemical properties. Nowadays, mainly spherical and neutral nanoparticles are used because neutral charge after extravasation helps in quicker diffusion of nanoparticle to the tumor tissue. Solubility, degradation, and clearance. Hydrophobic nanoparticles, except PEG-violated ones, are randomly cleared by reticuloendothelial system from bloodstream. Targeting. There are two types of targeting, passive and active. Passive targeting is mainly based on enhanced permeability and retention effect, that is EPR effect, that is passive accumulation of nanoparticles in leaky blood vasculature of tumors. In active targeting, ligands attach to the surface of nanoparticles, and these ligands are highly specific for receptors and other cancer-specific overexpressed targets on the surface of tumor cells. Now coming to stimuli, some external stimuli are temperature, light, ultrasound, magnetic field, electric field, and some internal stimuli are changes in pH, redox, ionic strength, and stress in target. Now coming to the next one, combination therapy and theranostics. Nanomedicines carrying multiple therapeutic agents may promote a good treatment process. Mobility and surface area, they are highly mobile in the free state and have large surface area. Now my friend Shubhadeep will discuss about biological barriers.
Nanomedicines are basically designed to prevent the tumor cells, and it is designed for the purpose that it can directly attack to the tumor cells, or not the fresh or the cells which are behaving normally. But during the application of the nanoparticles, some of the barriers are to be visible. The first one is the nano biointeractions in the blood. the blood proteins or the proteins present in the blood binds with the nanoparticle, which changes its physicochemical characteristics and its hydrodynamic charges and its chemistry, and which may lead to the malfunction of the nanoparticle. The next point is the sequestration by the means of a mononuclear phagocytic system. Sequestration is generally controlled by the resident macrophages of the liver and spleen. By the presence of nanoparticles, the process of sequestration is accelerated, and which results the accumulation of non-biodegradable nanoparticles in liver and spleen. The next point is the tumor extravasation. On reaching to the tumor microenvironment, this problem is to be faced. Initially, it was thought that it will be favored by the EPR effect, but practically it went wrong. Firstly, the vascular hyperpermeability restricts the nanoparticle convection, as the vasculature solely depends on the cancer type and also the cancer patient. So the theory of leak vasculature was incorrect. The next one is the intratumoral distribution. During deep and uniform nanoparticle distribution, high interstitial pressure acts as a barrier and will be especially hindered for having denser stroma. High collagen content and other factors in this case acts as a barrier. Now, moving to the last point, that is the intracellular uptake, trafficking, and cargo release. The action of nanoparticles takes via endocytic pathway and also via lipid fusion and translocation. As the targeting ligands were buried under protein corona, and thus the hydrodynamic size and charges of the nanoparticle is changed, which may lead to the different mechanism, such as which it can refuse to the cellular entry. Now, here are all the points which I have said earlier. Now, moving on to the opportunities, which will be explained by my friend Orko Prabhu. Orko, over to you.
Thank you, Shubhadeep. Turning barrier into allies. The first point is harnessing the protein corona. Protein corona can be used for identification of disease and may be used as templates to form patient-specific nanoparticle. For example, a specific protein biomarker in the corona of fullerene derivatives has been detected in lung squamous cell carcinoma patients. Hijacking circulating blood cells. Single-wall carbon nanotubes hijack circulating monocytes and get accumulated at cancer sites. The next one is exploiting off-target biodistribution to immune-rich organs. Nanoparticles are able to polarize tumor-associated macrophages and thus alter the immune response, which may result in prevention of metastasis. Study of nanobio interaction. To produce more efficient nanomedicines, the study of biological barriers as well as nanobio interaction must be improved. Changing administration process. In spite of intravenous administration, non-invasive intranasal administration may increase the drug at target site. Now, my friend Shubhadeep will talk about imaging, clinical trials of nanomedicine, and then he will conclude our presentation. Shubhadeep, over to you.
Thank you, Orko. And now I am moving to the imaging. The imaging mainly allows the non-invasive measurement and the biodistribution and pharmacokinetics of this drug delivery system in the models of diseases. So by the help of imaging, we can detect some of the facts, which includes that EPR is the most common uptake mechanism for the nanoparticles, and also EPR effect strictly varies between diseases, patients, and also it is heterogeneous. Nowadays, for imaging, PET or positron emission tomography or MRI or ultrasound is being used. Now, moving to the clinical trials. So in this graph, we can see that the clinical trials of different drugs as per approval of FDA, except the green ones, the trial information are not publicly available. The white dashed line seen in the graph shows the average elapsed time from phase 0 to 1 clinical trial of different type of nanomedicines. Now, moving to the conclusion of our session. As we can see that the definition of cancer nanomedicine is expanding beyond that of a nanosize delivery. But in preclinical and clinical researches has shown that the cancer nanomedicine may show earlier diagnosis, alert the immune system against tumor. So as we know, there are different type of problems, but we hope that in in earlier future or in the coming future, all of our problems will be overcrossed, and soon the nanomedicine will be available for all of us. Now here some of the references from which I have taken or we have taken help, and this is our team, Orkoprabhu Roy, Ahona Nandi, and this is me, Shubhodip Poddar. Thank you so much for hearing.
Thank you, Shubhodip and Orkoprabhu. I would like to request the judges to give their remarks.
Okay, very nice presentation.
Thank you, ma'am.
Thank you, ma'am.
Can you comment on the toxicity of the nanoparticles?
Yes, ma'am. Yes, ma'am. The toxicity of nanoparticle is a very controversial thing, because it is said that the nanoparticle which was invented in US, that is which name is doxorubicin, it causes cardiotoxicity. And it is said that the cardiotoxicity is caused by anthracyclines. So it is also a type of barrier that some of the nanomedicines are not good for our health. That is, they are stopping the tumor cells, but on the other hand, they are creating a new problem for our body.
True. Okay, good.
And ma'am, sometimes they are not very specific. They also affect some normal cells, except cancerous or malignant cells, actually. That's a major problem with nanomedicines nowadays. In some cases, not for all nanomedicines, in some cases.
If it is not targeted delivery, then it will surely cause problems.
Yes, ma'am.
Yes.
Nice presentation. Good.
Thank you, ma'am.
Thank you, ma'am.
Yeah, it was nice. I just want to ask you one small question.
Yes.
In your presentation, you have mentioned one particular nanomolecule, which is very unique in the sense that it has a perfectly symmetrical structure. Which one is that? One of the rare nanomolecules which have perfect symmetry in structure. You have shown that in your slide.
So do you have to mention the type or name?
Name.
Oh, name. So sir, I don't know, at least.
Sorry, sir.
Yeah. It's okay, no problem. That will be all.
Thank you, sir. Thank you, sir.
Thank you, teachers. Thank you so much.
Thank you, judges. Our next set of presenters are Shreya Ghosh and Srija Mukherjee.
Am I audible?
Yes, you are.
Okay. Good evening, everyone. Myself, Shreya Ghosh, and my partner, Srija Mukherjee, will be presenting on the topic probiotics and immune system. There are several microorganisms in our body to keep us healthy, and probiotics are a part of the larger microorganism in the body and microbiome. They are good bacteria which exert prophylactic and therapeutic effect by improving internal microbial balance and maintains balance between necessary and excessive defense mechanism, including innate and adaptive immune response. The main benefit is that they help to restore the balance in the intestinal microbiota. Sources of probiotics. Lactobacillus and Bifidobacterium commonly have probiotic properties. Probiotics can be introduced into our body through fermented foods, nutritional supplements, drugs, and so on. At right levels, probiotics aid digestion and improve nutrient absorption. Probiotic foods like yogurt contains lactic acid bacteria, which neutralizes deleterious effects of gut pathogens, enhancing the gastrointestinal system. Let us see how the gastrointestinal system influences our immunity. The gut accounts for 25% of the immune cells in the body, and the gastrointestinal tract being the largest interface between us and the external environment, most of the immune cells reside in the walls of the large intestine. The most common illness associated with alterations of gut microbiota include IBD, GI infections, colorectal cancer, liver disorders. Microflora of the gastrointestinal tract stimulates the immune system to respond rapidly to infections with pathogens, and through bacterial antagonism, it inhibits the colonization of gut by harmful pathogenic bacteria. The gut microflora comprises of Enterococcus faecalis, Clostridium difficile, Campylobacter, Escherichia coli, Lactobacilli bifidobacteria. Probiotic microorganism lactic acid bacteria positively influence the composition of gut microflora. Now we'll see the evidences of probiotics as a savior. Researchers have shown the effectiveness of probiotics in the treatment of the following: inflammatory bowel disease, allergies, urinogenital infections, cystic fibrosis, gastrointestinal infections. Let's see how probiotics influence our body. Dietary supply of probiotic bacteria stimulates IgA immune response and transport of antigens through Peyer's patches. Peyer's patches are primary sites in the gut mucous membrane where specific immune responses are performed. Probiotic bacteria influence the processing of mucus layer antigens according to the type of dietary content. If the amount of dietary antigen is lower, then the effect of probiotic bacteria in mucus antigen is decreased. Now, I will give the second half of the presentation to Srija. Over to you, Srija.
So now we will see how probiotics exhibit the property of immunomodulation through two different pathways. First, through the direct pathway, probiotics increase the activity of the macrophages and the natural killer cells and modulate the secretion of immunoglobulins and cytokines. While through the indirect pathways, probiotics enhance the gut epithelial barrier, alter the mucus secretion, and successfully compete with and exclude the harmful pathogenic bacteria. Now, the picture in the given slide explains the mechanism of immunomodulation by the probiotics. Specific probiotic bacterial strains regulate the mucin expression and therefore indirectly regulate the immune system by supporting a healthy mucus layer. Now, we will see in details about the property of immunomodulation by probiotics. The immunomodulatory effect of probiotics is attributed to the release of cytokines, including the interleukins, the tumor necrosis factor, the interferons, the transforming growth factor, and chemokines from the immune cells. Probiotics have reported to influence the cytokine production by the APCs, which initiates the adaptive responses. In a nutshell, probiotics is a promising research arena in the field of medical sciences, and further studies can be suggested to determine the precise action of probiotics. New strains of probiotics can be established by understanding the cytokine secretion by the Th2 cells, the GCs, monocytes, B cells, and the Treg cells. Probiotics stimulate the B cells and influence the cytokine production, and as a result, it increases the gut barrier, which initiates the adaptive responses in the host body. The degenerative and the other diseases caused by the pathogenic microorganisms can be cured by probiotics based on their immunomodulatory properties. Now, probiotics can at times also cause adverse effects, which include amines like histamine, phenylalanine, etc., which are present in the probiotic food, can at times excite the central nervous system and thus cause headache. Certain bacterial strains found in probiotics can also increase the histamine levels. They can cause unpleasant digestive system, such as temporary increase in gas, bloating, constipation, and thirst. Now, we will look at the factors which we need to keep in mind before we choose the probiotics for us. Because these probiotics can at times trigger allergic reactions and as a result can cause skin rashes. Thus, a person who is having allergy and lactose intolerance should refrain from using probiotics too much. Probiotics can also cause adverse effects in case of a person suffering from compromised immune system and a person who is consuming antifungal infection drugs. Now we will see about the future prospects of probiotics. Studies suggest that using probiotics may be a benefit at inducing and maintaining a remission of ulcerative colitis and pouchitis. However, Crohn's disease has limited studies and its prevention and remission has not been associated with taking probiotics. Thus, the engineering of specific protective molecules like interleukin 10, ganglioside, and trefoil factors into probiotics for in situ release will give promising results. Thus, a complete understanding of interaction between the genetic, the environmental, and the microbial influences within an individual is required for more effective probiotic interventions for microbiota associated conditions. And further studies on probiotic properties will enable us to understand in a better way the application of probiotics in nutrition and health factors. The following are the reference sources that we have used for our presentation. So now we have come to the end of our presentation. Let's be motivated to extract the optimum out of probiotics. Thank you, everyone.
Thank you, Shriya and Srija. I would request the judges to give their remarks.
Okay, nice presentation.
Thank you, ma'am.
You presented the topic from a new aspect. Okay, that means that is all probiotics are not for all of us, right? Now, is there any screening method, or in future can we expect a screening method for choosing the right probiotic?
Yes, ma'am, these trials are going on because we know that as I said, that the probiotics may at times they contain, all the probiotics are not for everyone. Like it depends upon the various strains that we use. Like some may, in case of person who have allergies, now when they take a particular strain of probiotic, it can cause excite their nervous system. As a result, the blood flow increases or decreases. So for that case, that probiotic, that individual should not take that probiotic. But similarly, that probiotic strain may not cause some adverse effect in case of other persons. So the trials are going on to check whether which individual should use depending upon their body, on their microbiome composition and all. And it's definite that all probiotics are not for everyone, and trials are also going on.
Okay, thank you.
Okay. Thank you, ma'am.
Yeah, it was nice. I have no questions.
Thank you, sir.
Thank you both the judges and all the groups that have presented. This brings us to the end of the second technical session of the day. Now, we will move on to our third technical session. It will be taken over by Natasha Mary Joseph and Jodhpur Shorter.
Thank you, Barsha. So we shall now proceed to the third technical presentation of today, which is the presentation by the MSc second year students of our microbiology department. The judges for this session are Dr. Mahashweta Mitra Ghosh and Professor Debjani Datta. Before we begin with the presentations, I would like to tell all the participants the rules. The time limit for each presentation is 10 minutes, followed by 2 minutes Q&A session by the judges. The first warning bell will ring at 8 minutes sharp, and the final bell will ring at 10 minutes, which will indicate the end of the time allotted for your presentation, beyond which you would be interrupted. In case you face any difficulties while presenting, you would be requested to mail your presentations to the presentation committee. Jyoti?
Thank you, Natasha Di. I would like to again reiterate that the presentation time is 10 minutes and there will be 2 minutes for the question and answer session. The first warning bell will be rung at 8 minutes and the final bell will be rung at 10 minutes, after which the participants are requested to wrap up the presentation. Now I will call upon our first group of presenters for the third technical session, Pratusha Khan and Shreyoshi Paul. Over to you.
Is the slide visible?
Yes, it's visible. Both
Yes,
video
it's
and slide.
previsible.
Okay, thank you.
Will you start?
Yes, Shreyoshi.
No, the ma'am. I'm asking to the ma'am. Ma'am?
Yes, I'm there. I'm there.
Yes, okay, ma'am.
Yes,
Yes,
go on.
please. Okay, ma'am. Thank you. Thank you.
Yeah.
So if you're, yes, ma'am. So if you're asked where the human body's nervous system is located, what would be your answer? Of course, you will answer brain and the spinal cord. But is that only so? Or if you're having some stomach related problems, will it always cause your physical discomfort only? Could microbes have the ability to send you in depression? So many questions. A very good evening, everybody. My name is Shreyoshi Paul. So today, me along with Pratyusha Khan are going to present the topic Depression, a conspiracy of microbes. But before jumping into the depression part, let's first understand how microbes are controlling our brain. Yes, you heard it absolutely right. Microbes, specifically gut microbes, are controlling our brain. So today lots of gut related talks, but don't be bored. This is going to be really interesting. So Pratyusha, next slide. Okay, so science says gut feeling is not a metaphor anymore. Our brain and gut actually communicate with each other. Now, how? So there is a bidirectional link between the central nervous system and enteric nervous system. And yes, here another nervous system comes into the picture, that is enteric nervous system. Of course, like our brain, it cannot check or balance your checkbook, or it cannot compose a fascinating article, but of course it can trigger big emotional shifts. And interestingly, our gut contains 500 millions of neurons which can send signals to the brain. Vagus nerve is one of the biggest nerve present there, which can send signals to the both the directions. That means from gut to brain and from brain to gut. Now, this enteric nervous system is so powerful that they can work without any direct input from the brain. In essence, gut can tell your brain the information before brain perceives it. So after listening this, what you are thinking about? Our brain's power is little overrated? Okay, that's up to you. Now, Pratyusha, go to the next slide. Okay, so to maintain this gut-brain communication, what is needed? So the presence of several gut microbiota, rather to say healthy gut microbiota, are very much essential. Now, what defines healthy gut microbiota? So healthy gut microbiota should provide some essential benefits. Benefits like they should develop immune system, they should uh, help in the nutrition acquisition part, or also resistant to certain infections, and etc, etc. There are so many gut microbes in gut microbiota, and of course this one slide is not sufficient to accommodate all of them. And they can secrete several neurotransmitters like gamma aminobutyric acid or several short chain fatty acids, which are very much essential to maintain this gut brain talk in a proper way. But what do you think? Any kind of alteration in this gut microbiota will cause always some stomach related problems? No, actually not always stomach related problems. This has the potential to send you in depression. And yes, this can be another major reason behind one's depressive like behavior. Now let's see in the next slide how normally this gut microbiota talk to our brain. That means the signal sends signals to our brain, and whenever they get altered, what the different things happened. So as you can see in the diagram, that this gut microbiota interact with the immune cells, prompting them to make cytokines which can send signal to the brain via the vagus nerve. And they also can interact with the gut cells like enteroendocrine cells and obviously make different neurotransmitters and metabolites, which I have told you earlier, and send signals to the brain. And whenever they get altered, these alterations can trigger the production of microbial lipopolysaccharide. And this can induce gastrointestinal inflammation. And not only that, this gastrointestinal inflammation can also cause neuroinflammation. Now, what's happened there? So, normally tryptophan is present in the gut, it reaches to the brain and produces happy hormone serotonin. But whenever this gut microbiota gets altered, tryptophan cannot anymore reach to the brain. So, no tryptophan in the brain, no serotonin, no happy hormone. Instead, tryptophan is broken down by the enzyme which is secreted by those altered gut microbiota and produced kynurenine. And this kynurenine can induce depression. Let's move into the next slide. So not only in depression, this altered gut microbiota also has the ability to be the reason behind anxiety, fear, and many other behavioral defects. They also influences amygdala neuronal morphology and function. Now, what is amygdala? Amygdala is basically a key brain area which is present in the base of the brain, and they can define different expression or behavior. Now, these microorganisms have conspired against us and send us in depression. Can we do something to reverse this conspiracy? So let's hear it from Pratyusha.
Thank you, Shreyushi. We are what we eat, and I kid you not when I say this. As Shreyoshi has mentioned before, lactobacillus and bifidobacterium has been shown to be most helpful for mental health. Pathogenic bacteria induces the production of inflammatory immune compounds and metabolites which signal the brain, altering its function, activating HPA axis and leading to depression, anxiety, and altered behavior. While the beneficial bacteria produces anti-inflammatory compounds and metabolites such as fatty acid, GABA, serotonin, which benefit the central nervous system by improving brain function, memory, and behavior. Hippocrates rightly said, all diseases begin in the gut. To vividly discuss how gut health is important for mental health, let's look into this diagram. Healthy food habits and proper sleep and exercise helps in maintaining the diverse intestinal microbiota, and symbiosis is maintained via keeping in check the number of pathobionts in the gut. While junk food, the use of drugs, lack of exercise result in gut microbiota dysbiosis. By dysbiosis, I mean the gut microbial species leading to decrease in healthy gut microbiota and increase in pathogenic species. This directly influences the HPA axis of CNS and increased intestinal membrane permeability. Altered microbiota leads to enteritis, colitis, irritable bowel syndrome, which leads to malfunctioning of the CNS. All these comorbidities either directly or indirectly cause irritative behavior and depression. Moving on to the next slide. Hippocrates, the father of medicine, rightly said, "Let food be thy medicine and medicine be thy food." Psychotherapy and antipsychotics have long been used to cope up with depression. Psychotropic medication influence the diversity and composition of microbiota and also have gastrointestinal side effects. Probiotics seem to be promising in restoring the healthy gut microbiota. Probiotic reduces production of inflammatory chemicals like cytokines or may directly act on tryptophan, which is thought to be very important in gut microbiota interaxis. Adding probiotic foods like yogurt, tofu, sauerkraut to our diet would help cope up. Let's reverse the conspiracy of the microbes. Probiotics provide the resistance to infection, reduces the effects of inflammation, and also modulates the immune system and reduces the effect of oxidative stress. Now, this schematic diagram of maladaptive microbiota gut-brain axis and the pathophysiology of the depression. Chronic exposure to stressors like poor nutrition, psychological stress triggers release norepinephrine, which alters gut microbiota composition by shifting to one which is enriched with pathogenic bacteria. Acetylcholine and glucocorticoids increase intestinal barrier permeability, which is very importantly linked to the HPA axis hyperactivity. Altered gut microbiota also results in inflammation of the gut and also altered neurotransmitter system, which leads to depression, anxiety, and altered behavior, giving rise to negative emotions. A lot more is yet to learn about the gut-brain link in depression. The area of research of gut microbiota on depression is still limited. Increased bacterial translocation and intestinal permeability's role in pathophysiology of depression shows a vast area of future research. These are some of the references that we have used. We are ending the presentation here. Thank you.
Thank you.
Thank you, Pratisha. Ma'am, we would like you to judge.
Yeah.
Yes.
Yeah, Devjani, you start, please.
Can I ask one question?
Yes, sir.
Yes, Aurukda, please go ahead. তারপরে আমরা
আচ্ছা দেখো অ্যামিগডালাকে ইনভেট করছে মাইক্রোবায়োম যদি অ্যামিগডালাকে ইনভেট করে can we say that microbiota actually can control precision, human precision? Because what is amygdala? It is a part of limbic system. So in that case, if the limbic system is modified, can our precision be harmed? Of course, depression you are concerned, but precision is also a very important thing, you know. So অর্জুনকে জিজ্ঞেস করেছিল তুমি কি দেখতে পাচ্ছ, বলেছিল আমি শুধু পাখির চোখ দেখতে পাচ্ছি। So that is precision. তো সেটাও কি তাহলে মাইক্রোবায়োম কন্ট্রোল করে?
Sir, actually it might control. We are not so sure about it, as because microbiota has a huge role in controlling different parts of our brain, so it might control, but actually we are not
Because
aware
you
about
only said amygdaloid body. Amygdala is a part of the limbic area, and that in one way is controlling our precision in the central part of the forebrain. Anyway, you try to find it out.
Actually, sir, as microbiota can alter the amygdala, so it might can do that actually.
Can you know if the amygdala is damaged, আমরা fearless হয়ে যাই, চিত্তজেতা ভয় শূন্য হয়ে যায়, কোনো ভয় থাকবে না। মানে তোমাকে যদি amygdala ড্যামেজ করে দিলে, তোমাকে যদি বলা হয় চারতলা থেকে ঝাঁপ দাও, তুমি ঝাঁপ দিয়ে দেবে।
Yes, sir, because there will be no emotions actually, because amygdala control all the emotions and define the
থিওরি আছে যে টেররিস্টদের যে হিউম্যান বোম যাদের তাদের অ্যামিগডালাকে জাস্ট ম্যাসিরেট করে দেবে, ব্যাস তারপর তারা যা খুশি করবে। দে বিকাম রোবট বেসিক্যালি। এনিওয়ে, থ্যাংক ইউ।
Thank you, sir.
Okay, I have one question. Am I audible?
Yes, ma'am.
Okay, so say I am suffering from depression. Okay, from your discussion, I got the idea that I might not have a proper gut flora. Okay, but what should be my approach? What should I do to find out if my gut microflora is the reason for my depression?
Actually, ma'am, you will feel certain kind of symptoms, like there will be some stomach related problems. It's whenever the gut microbiota is involved in altering several behavior, or whenever it's the reason behind somebody's depression, then it's not like that you won't feel anything or you won't get any symptoms. You will feel some stomach related problems as well. What happens whenever we feel some stomach related problems, we tend to believe that it's like we don't go to doctor or
it's only
somewhere.
associated to digestive system, but actually not so. And we already know how our diet is, so we know our routine, how irregular sleeps we go through. So that would lead us to know that our routine, our diet is responsible, our gut microbiota is responsible for causing our depression. The person who goes to a good diet and good exercising, proper metabolism and proper sleep, they are less prone to depression.
No, I basically wanted to ask you what kind of experimental analysis or what should be done so that I actually find out, because depression can have a lot of other issues as well, isn't it? It may not be only due to the gut flora. There can be other reasons. So how do we link that? Is there any protocol? Is there any scheme or strategy which will, I mean, ultimately evaluate and tell me that this is the reason?
Ma'am, basically the studies are going on mice recently. Now,
Okay.
no human-based study has not yet been started. On living organisms, we haven't seen any paper regarding that.
Okay. Okay, thank you.
But ma'am, I want to add one thing. If you
Yes.
are having like depression, but at the same time you will also have some gut related problems or stomach related problems. It's not like that ki you won't have any gut related problems, then also your like depression is linked to the gut microbiota. It won't happen like that. If your depression is linked to your gut microbiota, you will definitely develop some gut related or stomach related problems.
Okay, so a very confident Dr. Shreyoshi Paul. Very good. Thank you.
Thank you, ma'am. Thank you so much.
Okay, simple question. You said now that the conversion of tryptophan to kynurenine is done by the pathogenic ones. So, have they taken this kind of approach to estimate the level of money in the mice models? So, how they estimate that are they working on this amino acid levels to check it out whether there is a chance of depression or something like that? Because you said that the transmission of tryptophan will be affected, right? So
Yes,
are they
ma'am.
using this as one of the parameters? Because I'm trying to
Yes,
give
ma'am.
you a clue onto the answer that Devjani madam might be asking from you. So is that one of the parameters to check it out?
Right, Mahashatadi.
Okay, I guess she was asking
Yes.
for this answer from you.
Yes.
Okay, yeah. So
Yes, ma'am.
instead of just looking into the observations, right? These observations are the part of doctors. That's why she said. But being a microbiologist, what you should look for, right? You should look for into the biochemical pathways or the other any other microbial assays. Are they going to help you out? So you have to check it out in the gut microbiota. What is there? So you can very fast go for a metagenome study, right, of the gut, and check it out the level of pathogenic ones, right, and the level of good ones. So if you get a comparative metagenomic study of a healthy gut and a diseased gut, then you can conclusively say that yes, sir or ma'am, you are having depressions as because your metagenome is loaded with pathogenic microbes. Okay, so I guess this can be one kind of approach. Okay, but above all, you both of you spoke extremely well. I was mesmerized to listen to all of you. Thank you.
Thank you, ma'am. Thank you.
Yes, it was a good presentation.
Yeah.
As far as the slides and your way of presenting is concerned, it was good. Yeah, the topic has been a repetition because we have been hearing throughout the evening about gut microbiota and all this. But anyway, the presentation way, it was very good.
That is not their fault, you see. This is the trend of research in the recent days.
Yes.
The trend
Yes,
of
that's
research
true.
is gut-brain contact,
Yes.
basically.
Yes.
Thank you, teachers.
Thank you, participants, for this very interesting presentation. Next, we call upon Rishabh Bharadwaj and Falguni Chaudhary to present their topic.
Good evening, everyone. Our topic of presentation is the contribution of microbial biotechnology to mitigating the coral reef degradation. Next slide, please. Now, first we will briefly describe about what are coral reefs, their habitat complexity and microbial diversity, their environmental influences on coral microbiome, and then we will look forward towards the microbiome manipulation and the studies involving them. Next slide, please. Now, coral reefs are one of the most diverse and economically important marine ecosystems on the planet. So what are corals? Corals are meta-organisms which can have a multipartite relationship between the cnidarian host. Basically, they form a symbiotic relationship between dinoflagellates, archaea, bacteria, and they form a complex system which we call as coral holoband. Next slide, please. Now coming to the different types of corals, as we can see on the slide, branching corals, massive corals, stable corals, and plating corals. Branching corals is basically where they branch their tips towards the sunlight and for their high water flow. Then the massive corals are very the slow growing corals, and plating corals, they attach themselves to the rock surface, and when they reach for sunlight, they grow upwards and outwards. Next slide, please. Now, environmental influence on coral microbiome. So coral bleaching is one of the most important factor for the coral bleaching is one of the most important factor for the coral reef degradation. Now, what is coral bleaching? When coral gets stressed by heat and temperature, they react by expelling the algae, marine algae, which is known as the zooxanthellae, and leaving a ghost transparent skeleton behind. So, unusual high water temperature can be the major cause of this coral bleaching. Beside this, light and salinity can also affect on corals. Next slide, please. Now coming to the microbiome manipulation. So what's the need of the microbiome manipulation? So to prevent this mass bleaching effects of corals, scientists have manipulated the microbes, because as the coral reefs are declining, harvesting and fishing can also be the major factor why for the coral reef biodegradation. So the new approaches, which includes the isolation of natural microbiomes, can improve the host performance and fitness in the face of the climate change. Now, what led scientists towards this microbiome manipulation? What are the key experiments by which they know that we can manipulate the microbes to prevent these mass bleaching effects will be further discussed by my partner, Rishabh. Over to you, Rishabh.
Thank you, Falguni. So some of the experiments that were conducted, and from that we come to know that maintaining a healthy microbiome is thought to be essential for the well-being of corals, as destabilization in the composition and function of their associated microbial communities have been shown to take place in disease state and under stressful environmental conditions. So elevated seawater temperatures and coral bleaching are typically associated with a shift towards opportunistic and or pathogenic bacteria, with concomitant decline in coral health. While an optimal microbiome may help protect the host from environmental pressure or compromised health by preserving beneficial functions, its dynamic nature may also confer an adaptive potential. For instance, the experiment that was conducted, reciprocal transplantation of Acropora hyacinthus fragments between thermally distinct environment on the same reef resulted in an adjustment of the microbial communities to the new conditions. While the microbiome of the cross-transplanted corals was indistinguishable from the microbiome of the native coral in the same pool, it changed compared to the microbiome of the back-transplanted counterparts. Now, if we look at the figure, moreover, when subjected to short-term heat stress, of the preceding 17 months in the more variable and warmer thermal region were found to bleach less and retained a more stable microbiome. These results suggest that microbial community composition influences the response to heat stress, although host genetic adaptation and acclimatization are known to play additional and very important role. These results suggest that microbial community composition influences the response to heat stress, although preliminary studies indicate that coral-associated prokaryotes can be manipulated through inoculations with specific taxa. Like in this experiment, bacteria collected from the coral Misummelia harti were cultured on a selective medium to isolate strains that were capable of degrading water-soluble oil fractions. So let me tell you, water-soluble oil fractions are the harmful hydrocarbons that have been released during oil spills, and these compounds are highly polar in nature. So when subjected to conditions stimulating accumulating an oil spill, polyps of Musamelia harti inoculated with a water-soluble oil fraction degrading bacterial consortia were less negative affected compared to the non-inoculated polyps as assessed by higher photosynthetic experiments. So what all these experiments tell us? These experiments tell us that exposure to a specific microbial mixture therefore conferred health benefits to corals under stressful environmental conditions. So many of the human activities that degrade coral reefs are extremely woven into social, culture, and economic fabric to regional coastal communities. Now, coral reefs face many threats from local sources, including physical damage or destruction from coastal development, pollutions that originate on land but find its way into coastal waters, like sedimentation from coastal development, urban runoff, nutrients from agricultural and residential fertilizer use, pathogens from inadequately treated sewage, stormwater or runoff from the livestock pens, toxic chemicals including metals, organic chemicals, and pesticides found in industrial discharge, trash and microplastics from improper disposal and stormwater runoff. Overfishing is also a major cause for the decline of corals. That is, overfishing can alter the food web structure and cause cascading effect. Now, in the diagram, we can see, in the picture, we can see blast fishing is a technique that is used. So explosives are used here to stun or kill fishes in a group, and this also harms the corals. And coral harvesting is done for the aquarium trade, jewelry, and cuirass lead to harvesting of specific species and further lead to the destruction and reduced biodiversity. Now, at last, we put forward some of the conclusion. Despite the recognition that coral reefs are threatened by human activities, measures to reduce negative impacts and sufficient steps that have not been taken yet. The United States have put forward targets that ought to be reached in coming decades to protect the marine ecosystem and avoid further damage to coral reefs. These goals include sustainable management of marine zones by reducing pollution and destructive fishing, as well as the transfer of scientific knowledge to improve ocean health and support developing countries that rely on coral reefs. Intervention methods such as the manipulation of the microbiome and genetic engineering have been successfully applied to terrestrial organisms to increase their tolerance to stress. Numerous lines of evidence suggest that a transplantation of these technologies to corals and their symbionts might effectively enhance coral resilience and contribute to success of coral reef restoration efforts. Now, we also put some future prospects. Studies using metagenomics methods have also been studied to unveil the function role of unique microbial communities significantly that contribute to coral holobiont protein nitrogen cycling, and the existence of conserved and intimate relationship between corals and microbes could open new avenues of research.
Rishabh, I would really like you to wrap up with your presentation.
Okay.
This is the second
So,
bell.
We also put some questions. Why is the incidence of bleaching increasing, and will the frequency of intensity of coral bleaching continue to increase? These are some of references that we have used. Thank you for listening us patiently.
Thank you for listening to us.
Thank you, participants, for this very nice presentation. Now I would request the professors to ask their questions.
Falguni and Rishabh, you have chosen a very interesting and very important topic that is a really matter of concern nowadays. And definitely the approaches that you have discussed, that microbial population is to be used or to be studied well, and the over excessive fishing is to be ruled out. Okay, so from the viewpoint of a microbiologist, what kind of microbes or you would prefer to be used to minimize the coral reef depletion? I mean, what kind of microbes, or specifically just name few of them, right? And can you have some kind of planning that microbe versus interaction with the corals, right? So there can be some kind of crosstalk being going
Ma'am,
on
the
between the
microbes which we can use, maybe the archibacteria, because they have the tolerance towards the high temperature, and high temperature can be the major cause of this coral bleaching effects. So if somehow we manipulate this because they have an association with archibacteria corals. So if we somehow manipulate this their interaction with the corals, archibacterial interaction with the corals, definitely, ma'am, I think so we can get an idea about what what kind of microbes we are using as archibacteria interaction. So if we change the temperature, or if we reduce something their interaction, if we know the pathway by which they are interacting with the corals, if we try to suppress them, then we can definitely decrease this mass bleaching effects of corals, according to me.
Okay,
Yes,
and what
this is
else?
a very this is a very vast topic. Actually, most people neglect these corals because not much studies that take place nowadays, and the seawater condition continuously keeps on changing. So we need to search for the microbiome that are very stable in those conditions, like the temperature continuously changes, and there is also change in pressure, etc. So archibacteria might suit if we continue the research in this field.
Don't you feel that a comparative study has to be done on a seasonal basis?
Yeah, ma'am. But
the study should be made, right? And
yes,
apart from this, as you said, that there are oil spillage, so oil eating or oil degrading bacteria should also be included in
yes,
in the bag, or the goodie bag, comprising
yes,
of good microbes. Okay, so it was a nice presentation, both
Thank
of you. Congratulations, Falguni and Rishabh. Over to Devjani.
No, I have no questions. I would just like to say that I enjoyed the presentation.
Thank you, ma'am. Thank you.
Thank you, ma'am.
Okay, so next up we have Harishu Kumar and Shaumita Shah for their presentation. Over to you.
Okay, good evening, everyone. I'm Harishu Kumar, and I, along with Samayata, are going to talk on the topic studies on immunogenicity against lipopolysaccharide and outer membrane protein of enteropathogenic Escherichia coli. Next slide. Here are the topics which we are going to cover in our presentation, starting with the introduction. Next slide. Enteropathogenic Escherichia coli, or in short, EPIC, is the most significant diarrheal bacterium. It is the major cause of childhood death in the developing countries, ranking as the second cause of death in the children under 5 years. Enteropathogenic Escherichia coli causes pediatric diarrhea in infants and occasionally causes sporadic diarrhea in adults. Although there are a wide range of antibiotics available to treat infection associated with enteropathogenic Escherichia coli, improper or overuse of antibiotic worsens the situation of patient. Multi-drug resistant in bacteria has also created a problem to the use of antibiotics globally. So it's better to prevent the disease than to cure. Now, how can we do that? Well, here comes the role of vaccination. The solution is to develop a heat-killed whole cell immunogen that can act as a low-cost vaccine against enteropathogenic Escherichia coli to offer wide-spectrum protection. But before going into the development of the vaccine, let's discuss a bit about the morphology of enteropathogenic Escherichia coli. Next slide. To begin with its morphology, all the diarrhea-causing Escherichia coli pathotypes, which is enteropathogenic Escherichia coli, are classified on the presence of specific genes such as EA and BFP genes, which are also used for identification and characterization of enteropathogenic Escherichia coli. EPIC or enteropathogenic Escherichia coli involve a separate histopathology called attaching effacing lesions, which enables the intimate attachment of bacterial cell wall to the epithelial cell surface and effacement of the host enterocyte microvilli. Next slide. Now let's talk about lipopolysaccharide, which plays a key role in pathogenesis. LPS found in the outer membrane of EPIC are large molecules consisting of a lipid and a polysaccharide composed of O antigen, outer core and inner core joined by a covalent bond. EPIC possesses a 54 megadalton plasmid, which is required for the expression of specific polysaccharide chain of the lipopolysaccharide, which play a major role in characteristic virulence activity of the organism. LPS is a pathogen-associated molecular pattern, so it also stimulates the innate immune system and mediates a local or systemic inflammatory response. LPS can also stimulate non-immune cell and initiate the inflammatory process. Next slide. Now, before going to pathogenesis, let's discuss the mode of transmission. Like other diarrheogenic Escherichia coli pathotype, EPIC transmission occur via fecal oral route with contaminated weaning food, contaminated hand, or contaminated fomites serving as vehicle. Now, coming back to pathogenesis, here is a diagram, and in the first column of the diagram, the first stage of EPIC infection is given, which involves initial attachment of the bacterium to the host intestinal epithelial cell. During this stage, enteropathogenic Escherichia coli produce dense microcolonies on surface of tissue in localized adherence pattern. The bacterium is attached to the host cell via plasmid-encoded bundle-forming pilus. In the second column, the next stage of pathogenesis includes production of effector proteins such as ESP-A, ESP-B, and ESP-D. The type 3 secretion system in the bacterium is to generate and create a pore, which permits the translocation of those effector molecules. ESP-A is a filamentous appendages which surround the whole bacterium. This appendages interacts with the host cell membrane and form a translocation tube. Through that translocation tube, the ESP-B effector protein is translocated into the host cell and then into the cytoplasm, while the ESP-D is transferred into the host cell membrane only. The last stage of EPIC pathogenesis is the attachment of bacterial membrane protein intimin, which is an outer membrane protein encoded by EAE gene. Here in the last column, you can clearly see a pedestal formation. Now, what's a pedestal formation? Well, AE pathogen inducing attachment effacing lesions, which causes the destruction of the surface microvilli and form an actin-rich structure, which is called pedestals upon which the bacterium sits. EPIC infections leads to alteration in physiologic functions such as increase in epithelial permeability, alteration in iron secretion, loss of absorptive surface, and inflammation-induced tissue damage, eventually leading to diarrhea. Next slide. For treatment, since EPIC strains have appeared resistance against many antibiotics, while initially the antibiotic are given, but suppose in case the condition worsens and the patient become resistant to antibiotics, because EPIC strains have appeared resistance against many antibiotics, including penicillin, cephalosporin, trimethoprim, sulfamethoxazole, fluoroquinolone, and aminoglycosides. So it's better to prevent the disease than to cure, like I said earlier. And for that, the solution is vaccination. Now over to you, Samayata. She will be discussing about the development of vaccine. Thank you.
Thank you, Adishu. So now we'll talk about the development of vaccine, as multi-drug resistance has been a problem, as Adishu has already mentioned. So vaccination is a non-natural procedure to induce an effective immune response. Vaccines are basically modified infectious agents that get injection into susceptible host cell and induce specific protection but not infection. So these agents are prepared as either heat killed, inactivated, or activated entire pathogen, maybe as toxin or as entire molecules involved in pathogen survival and multiplication in infected hosts. Therefore, as diarrhea has been the important secondary cause for the 70,000 deaths over worldwide, so the causative agents being the enteropathogenic Escherichia coli, and several trials of vaccine has been already going on, so that we aim to develop a low-cost vaccine candidate combining the 5 main EPEC pathotypes that are basically enteroaggregative, enterohemorrhagenic, enteroinvasive, enterotoxigenic, and enteropathogenic Escherichia coli. So to overall provide a wide spectrum of protection against the 5 main pathotypes of E. coli. Next slide, Vijeta. So now we'll basically see the immunogenicity determination. For basically going on determination of immunogenicity, first you have to basically select the bacterial strains. There are 5 different bacterial strains that were that can be collected on the basis of the genotypic characterization. But before that, we have to also go for the isolation of the whole cell lysate. And for that, we have to basically grow the bacterial culture overnight at a room temperature and then centrifuge and sonicate it. So the membrane lysis happen, and then we get the proteins that we require in the super 10 that we collect later on, and then again centrifuge it. So after that, we can basically go for the PCR-based method to basically characterize the strain for the standardization to amplify a single copy or more copies of a nucleotide segment, generating thousands and millions of copies of a particular nucleotide sequence. Following that, we have to basically see the protein estimation that is basically gone through and done by the Bradford assay. Bradford assay is basically a technique that is used for quantification of the total protein content in a sample by the help of a protein dye complex mixture, where the binding of the happens and the color changes from brown to blue, and the chromatin blue dye has been used. Next slide. So now for the measurement of the immunogenicity efficacy, we basically go after the protein estimation. We have to see the number of what are the type of protein that are basically content. For that, we basically go to the Western blot technique, which is the important technique to identify the specific proteins. And for that, we basically transfer the proteins that are electrophorized into a nylon membrane, and then the protein bands are shown. The thickness of the bands tells us about the amount of protein that are present, and from there we basically go through the standard curve of the immunogens IgG titer value, which we have already shown in the picture, to basically tell us the standard curve or the amount of IgG immunoglobulin present. But we measure that from the sample of the sera, which we take from the non-immunized and immunized animal models that we basically present during and after immunization. Next slide. So concluding this slide, in this way, we can basically develop a vaccine, which is a whole cell heat-killed vaccine from the LPS and OMP for the EPEC pathotypes, which has been interesting to see, and the clinical studies in humans have be valuable if they are going on, and this future it might be a great weapon to fight against the enteropathogenic Escherichia coli infection. So making it a booster dose, it would be effective for the gut microbiota prior to any clinical trials. Next slide. So talking about some future prospect about this development of vaccine, heat kill vaccine has been the most important is that it's very easy process and very cheap and safe for the possibility to return to a more heinous form. Heat kill antigens offer protective immunity after use of a booster dose, and it provides multi-drug resistant bacteria and also. So in this way, we can develop a vaccine and provide significant protection. Thank you everyone for patiently listening. Have a great day. And these are the references we have taken from.
Thank you participants for this very interesting presentation. Now I would ask the professors to ask their questions.
Parishu and Shamaita, you have again discussed about enterohemorrhagic E. coli. Definitely, it is an enteropathogenic E. coli that is a real threat. We can understand. So you have talked about the development of vaccines. So can you exactly tell me at what stage the vaccines are now existing, or can apart from vaccine, what else can you think of from your concept from the topics that you have learned so far in your BSc course and MSc course? Apart from vaccine, what else? You have spoken very well that heat kill vaccines are the best because means all the epitopes will be there, and we can immunization is always better. But definitely this how long this will be effective and what will be the frequency of that vaccine. So there are many things and how effective the vaccine is. These all things are to be taken care of, right? So, so at what stage vaccine development is right now? Harishu and Samaita, both of you.
Stage exactly, but what I have read and I'm aware of is that the retail vaccine is basically the epitopes are present and we can easily, and it's cheap also, because the antigen and the anti-sera have been immunized and the non-immunized, and we can easily get the antibodies and the IgG immunoglobulin titer that is present. So I don't know, like as far as I know, the vaccine development is what I am aware of.
That's great. That's great. But have they done the studies on animal models, or now have they done the study on animal human system or like that? Yeah, your voice is cracking. Yeah.
Sorry. Yes, ma'am. Nice models it has been performed so far. What I have read to paper.
So apart from vaccine, what else can be done to combat a pathogen which is drug resistant?
I'm not really aware of it. Like, can I look into it? Like, I'm not aware of.
Okay. No problem. No issues. Nice presentation. You all of you have prepared well. Very good. Thank you, Harishu and Samaita. Devjani.
Yes. Yeah. Harishu and Samaita, very well explained. Okay. And patiently, slowly, you have gone into the topic, explained very well. I just have a very simple question, and that is, we know that there are so many microorganisms, bacteria also. There are various types which causes diarrhea. What is the significant symptom for EPEC? What is the distinguishing symptom for diarrhea caused by EPEC? I hope you got the question.
Yes, ma'am.
Yes.
Improper bowel movement is my one symptom.
Okay.
Yes, but watery diarrhea, the low breath and
That
vomit.
That is very similar to that happens for almost diarrhea, right? Is there anything very significant about it? Just look out and find out. Okay.
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 Thank you.
Thank you, ma'am.
Thank you.
Thank you so much, Harishu and Samantha. The next presentation will be from Shinjini Chaudhary and Camelia Roy.
Ma'am, can you let me know if you can follow the slideshow?
Yes, we can see it, Shinjini.
No, like the slideshow, the changing of the slides.
Yes, I can see the slides changing.
Okay, thank you. A very good evening to one and all present here. I am Shinjini, and today my classmate Camelia Roy and I feel honored to have the opportunity to address such a distinguished audience. The topic of our presentation is generation of neurospheres from mixed primary hippocampal and cortical neuron isolated from E14, E16 Sprague-Dawley rat embryo. The authors of this article are Gaurav Das, Varsha Gupta, Juhi Khan, Deepshika Mukherjee, and Surajit Ghosh. This article was published in Journal of Visualized Experiments, often known as JOVIES, on 6th May 2019. We have divided the presentation into 4 major segments: introduction, assay protocol, results, and conclusion. Now, I would like to begin my presentation by giving some background information. The brain is a complex network of neurons interspersed with non-neuronal cells. In order to learn about the mechanisms that govern the regulatory parameters of this machinery, this system needs to be reconstituted in vitro. Cell culture, predominantly using clonal cell lines, is a significant method and provides a snapshot of the underlying mechanistic processes that are relatively difficult to carry out in vivo. However, the contamination and the significant genotypic and phenotypic departure from their tissues of origin have reignited the interest towards the primary culture.
Primary culture is a quintessential method for in vitro assay given their normal cell morphology and their closer resemble to the in vivo environment. However, the yield of neurons are very low. This is due to the lack of the paracrine tropic support which they are supposed to get from the surrounding cells. Moreover, the primary culture is a 2D culture and does not represent the heterogeneity of the brain. This has given rise to the need for a more believable 3D culture system that has emerged as new tools in neuroscience research. Now, these tools are known as the neurospheres. Now, neurospheres are non-adherent 3D clusters of cells that are rich in neural stem cells as well as the neural progenitor cells. The paramount importance of the neurosphere lies in the fact that they are rich in the neural progenitor cells, as a result of which they can differentiate into neurons as well as the non-neuronal cells. Assay protocol. The model organism for this assay is an E14-E16 time pregnant Sprague dolly rat, where the day with the vaginal plug detection is marked as E0. The rat was sacrificed, and after sterilizing the abdominal area, a V-shaped cut was made. Then the embryonic sacs were taken in a Petri plate and the head was decapitated from the embryo. Then under the stereo microscope, the brain was carefully removed. The meninges were also removed and the hemispheres containing the hippocampus and the cortex, they were collected. Then dissociation media along with the plating media was added and aspirated at intervals. The density of the cells were determined and then the cells were counted. Finally, the cells were diluted to obtain two separate tubes for high density plating and low density plating respectively. Cells were examined 4 hours after plating. The neurons and the neurospheres further formed, they were cultured, and then the cells were characterized by immunostaining with the cytological markers, and this imaging of the cells under the microscope was also performed. Over to Camelia.
Thank you, Shinjini. Now I'll be discussing about the results of this experiment. This diagram illustrates the adherence of the cell after 4 hours of plating. The left one is the shows the cell adherence in high density plated neurons, and the right one shows cell adherence in low density plated neurons. Here we can clearly see more adherence was found in high density plated neurons. The DIC images obtained after 24 hours of plating show extensive synaptic connections. This figure shows the phase contrast images of low density plated neurons after 7 days of culture, where we can see extensive sprouting of neurons. The immunocytochemical staining was performed by using neuronal marker TUJ1, which is red in color, and axonal marker tau, which is green in color, which reveals the neuronal nature of the culture. Now, non-neuronal markers such as GFAP and O4 were used for astrocytes and oligodendrocytes respectively. The absence of staining of this marker represents the purity of the neuronal culture. In all the characterization studies, the nuclei shown in blue were stained with HUX 33258. The low density seeded cells were observed under 4 different channels continuously for up to 7 days, where we can clearly see that this protocol supports the growth of neurons over astrocytes. The neuronal and astrocytic population observed through microscopy were then estimated for quantitative analysis. Initially, about 2 to 3% of the astrocytic population was found to survive, but later it was found that all the astrocytic population slowly perished over time, and the growth of neurons took over the entire culture. This bar graph represents the percentage population of this bar graph represents the percentage ratio of population of TUJ1 expressing cells to that of GFAP expressing cells. Then the longevity of the neurons cultured for about 30 days were assessed, and cell viability was measured, where we see that neurons show more than 90% of the cell viability even after 30 days of culture. From this figure, we can clearly see the spontaneous formation of neurospheres in high density plated neurons after 7 days of culture. After 8 to 10 days, we can clearly observe the distinct formation of bridges consisting of radial glide-like extensions in between neurospheres. Now, the neurospheres rich in NPCs were found to co-express both nestin and TUJ1 respectively, and both show positive staining. The presence of GFAP in the neurospheres forming high density plated culture were marked by the green color of the GFAP staining, though significantly less was observed in comparison to TUJ1 expressing neuronal population. This was also reaffirmed by the quantitative analysis expressed in the form of bar graph, in which we can see the 17% of GFAP expressing non-neural culture in comparison to 83% of the TUJ1 expressing cells. Now, the health of the cells were determined by performing live and dead cell assay, which was performed by using calcine AM, that is green in color, and propidium iodine, that is red in color. We can observe the neurospheres give enormous amount of green fluorescence and no death staining was observed, which indicates that no death has occurred in between at least up to 15 days of culture. This line graph represents the voluminous expansion of neurospheres with the passage of days. Thank you. Over to Shinji.
So in this protocol, a simple recipe has been formulated to culture the primary neurons from the rat and then plate it into two variable densities, high density as well as the low density. In conclusion, a single strategy has been used to generate both the two-dimensional as well as the three-dimensional platform, which can be used for screening of the neurotherapeutic candidates. Now, this assay is very simple and cost-effective as the number of reagents and steps are very less. Now, after the high-density plating, the neurospheres, which are very rich in the neural progenitor cells, they can be used to study the neural differentiation into neurons as well as the radial glial cells or the non-neuronal cells, which in turn reflect the early stage of the cortical development in vivo. Thank you for listening. Have a good day.
What is the nature of this neurosphere?
These neurospheres are actually floating cultures, which are they're actually clusters which are very, very rich in the neural stem cells as well as the neural progenitor cells. As a result of which they can differentiate.
Which particular age of the embryo they are produced?
So during the E14 and the E16, like when the Sprague-Dawley rat, like the day of the vaginal plug detection is marked as E0. So if we count like that, the day E14 and the 16th day, during that time, if the experiment is conducted, then we can find the neurospheres.
What can be their fate? Why they are produced at all?
So neurospheres are produced because the embryos are very rich in the neural progenitor cells, and when we use high density plating, like when we obtain the neurospheres, and then the neurospheres can differentiate into now if we check the viability, we can find like the cells were viable up to about 15 days.
No, basically why I am asking this, because the neuroglial cells or the progenitors, they are not specifically producing neuronal cell. They can produce other tissues also. Record says that. So that's why I am asking that if they are displaced from that point, will they have any other fate? Because that was studied in mice. So what about, human embryonic stem cell? Has there been any report on that? Displacement of this neuroglial cells or neuroglial progenitors producing any other organ?
So
you got my question. You got my point.
I got it.
It's very interesting because we have specifically very few types of cells as the stem cells, and these are producing the different hosts of tissue. And ultimately, that we have the ectoderm, endoderm, and mesoderm. Ultimately, these are our fate. So the stem cells tend to be categorized into these three types. So naturally, multiple organs are produced. So who is the key player here? Naturally, the growth factors, what we supply in their culture. So you will not be astonished when the neuronal cell can actually be producing cardiomyocytes. It can be.
So in the maintenance medium, like we have plated it in low densities as well as in high densities. In case of the low density,
how can you differentiate between low density and high density?
So cell count was measured, and then.
No, but how it is controlled? Cell density, how is it controlled in vitro? By adjusting what? Because a cell density will be depending on what? The nutrient. So by adjusting the nutrient supply, you can actually control the low density and high density. And thereby in vitro condition, they can have different fate. Anyway, interesting. Thank you.
Thank you.
I basically have no question. It was an interesting talk. And as Sir was trying to ask you, so we got into some more insights. Thank you.
Thank you, ma'am.
Okay, so now we were moving on to our next group of presenters, Anvit Shadhukan and Pritam Haldar.
Is my slide visible?
Yes, your slide is visible.
Mm. Good evening, everyone. Me, Pritam Haldar, and Anvit Shadhukan will be presenting on the topic Typha angusta, the phytomedicine. Typha angusta is a scientific name of the plant, which is commonly known as Lesser Indian reed mace, which is distributed in the tropical regions of India, China, and Japan. Some of the medicinal properties of the plant are: it can be used as an anti-inflammatory agent. Naringenin present in the plant can be used as a therapeutic agent in controlling the vascular problems, treatment of any disease causing blood loss, like nosebleed, etc. Phytomedicines or herbal medicine are used over drug or modern medicine because it is cost effective and it is easily available, and it has a lower probability of causing infection and has lesser side effects. Come to the next slide, which is collection and extraction of plant inflorescence from Typha angusta. Plant inflorescence was taken and dried for a week at a temperature of 25 to 30 degrees Celsius. It was then placed in a desiccator containing silica gel and cobalt chloride. Then the drying process stopped when 70% of the silica gel changed color from blue to pink. 100 gram of dried plant inflorescence was extracted and 200 milliliter of distilled water was added to it. The aqueous extract was then filtered using N1 Wattman filter paper with a temperature of which was concentrated at a temperature of 40 to 60 degrees Celsius. It was then stored at 4 degrees Celsius. Come to the next slide. The base of the ointment was prepared using 35 gram of cetosterol alcohol, 17.5 gram of PEC 600, 270 gram of petroleum jelly, 270 gram of liquid paraffin, and 400 gram of methyl paraben. And then the herbal extract was added to it. So the control drug used for the experiment are povidone iodine and Bernal. Povidone iodine is the reference drug for excision and incision wound. Bernal is the reference drug for the burn wound. Next slide shows the differences between excision and burn. Excision is a removal of tissue using a sharp knife or any other cutting instrument. Incision is a surgical cut through a skin, and burn is a damage or injury to the skin using heat or fire. Over to Anvit.
Thank you, Pritam. So when we conduct the macroscopic study for excision on rats, can you please move to the next slide? It was observed that on 20th day, for the TIO treated wounds, there was 100% wound contraction, both for the reference drug as well as the TIO treated ointment drug. Now, TIO stands for Typha angusta inflorescence extract ointment, which comprises of the base of the ointment along with the herbal extract. Group 2 refers to the reference drug, which is povidone iodine, and Group 1 represents the control, which is only the base of the ointment without the herbal extract. This percentage wound contraction rate was being measured using the formula wound area on day 0 minus wound area on day n by x, where n stands for the observation days on the multiple of 4, that is 4th, 8th, 12th up to 40 days, and x stands for the wound area on 0th day multiplied by 100. Next slide. So when we are moving to the observation table that indicates the percentage wound contraction rate for excision, we observe that there is 100% contraction on 24th day for the TIO treated wounds in excision as well as the reference drug, whereas for the control it went up to 40 days for 100% contraction. This is also represented in the graph where group 2 and group 3 is being shown in the range of 16 to 20 days, or rather 20 to 24 days for the percentage 100% wound contraction, whereas for group 1, that is the control, it goes up to 40 days. Now, when we move to the incision, once again we observe that at 20th day, there is 100% wound contraction for, can you move to the next slide, 100% wound contraction on 20th day. And this indicates that group 4 represents once again the control, which is only the base. Group 5 represents the reference drug, which is povidone iodine once again, and group 6 represents the TIO ointment. So the percentage wound contraction rate is been measured using the same formula as said earlier. Next slide. So once again, when you observe the percentage contraction rate for incision on 20th day, there is 100% contraction for the TIO treated wound, whereas for group 4, that control, it goes up to 40 days. This has been shown in the graph once again that group 4, it goes for 40 days, that is the control. Group 5 and 6 ranges in between 16 to 20 days. Next slide, please. Now, when we see about the burn, for group 7, we observe the control. Group 8 refers to the reference drug, which is Bernol, and group 9 once again represents the TIO treated drug for the burn wound, and there is 100% wound contraction on 20th day. So the percentage wound contraction rate was being measured using the same formula as mentioned earlier. Next slide, please. So now over here, we once again observe that there is 100% contraction on 24th day, as well as for the TIO treated, and it goes for the control, it goes up to 40 days. Next slide, please. So when we move to the histopathological studies, after 14 days of the entire wound healing process, the cells were isolated from the wounds, and it was observed that the TIO treated wounds for the 3 different types of wounds, that is excision, incision, and burn, had a greater amount of fibroblasts and spindle cells and collagen, giving it a more organized structure. Obviously, it has a greater granulation tissue with greater epithelialization and angiogenesis, that is the blood vessel formation, which indicates a better process of wound healing. Such studies were understood using hematoxylin and eosin staining by taking a median section of the tissue sample of the dermal wounds, and the intact dermis was stained using this stain. Next slide. So obviously, when we have 9 different results where the excision, incision, and wound model is being taken, the control, as mentioned earlier, is simply the base of the ointment without the herbal extract. Reference drug is povidone iodine and Bernol, where Bernol is for burn and excision, incision, it is povidone iodine. TIO is the Typha angusta inflorescence extract ointment treated wound. So obviously over here, the red arrow indicates the spindle cells, the black arrow indicates epithelialization, the yellow arrow indicates RBC, which is obviously understood from angiogenesis, that is indicated from orange arrow, that is the blood vessels are indicated using orange arrow. Now, when we understand or move to the biochemical markers that are responsible for excision, incision, and burn, we can move to the next slide. Obviously, hydroxyproline estimation is used to represent the collagen content, and enhanced wound healing is being understood using fibroblast migration and collagen formation. Now, the spectrophotometric measurement of the hydroxyproline content was done on the 14th and 20th day tissue that is extracted out from the 3 different kinds of wound models, and it was observed for the TIO treated wounds in the 3 different cases. On the 20th day tissue that is being extracted in these cases, it had a higher amount of hydroxyproline, which was understood using spectrophotometric analysis at a wavelength of 540 nanometer, implying that it has a greater amount of collagen content. Now, when we move to the proteins, the other proteins that are generated due to cell proliferation, and please go to the previous slide. And obviously, once again, the proteins that were found were measured using biuret assay, and that was also observed that for 20th day, it had a greater content of protein for the TIO treated wounds, the 3 different wound models. Next slide, please. Now, when we come to catalyst, now reactive oxygen species like superoxide anion is responsible to break down collagen, which slows down the wound healing process. Now, antioxidants, or rather cellular antioxidants like catalyst, forms complexes with superoxide anions and thereby enhance the wound healing process. And so this catalyst content was measured once again spectrophotometrically at 240 nanometer wavelength on the 20th day of the wound healing process, and it was observed that 100 milligram of the tissue had a greater amount, or rather tentatively an almost similar amount as compared to the reference drug used in either cases for the three different wound models. Over to Preetam for concluding.
Thank you.
Preetam?
Coming to conclusion, the main objective of the experiment was to heal the injury with the shortest possible time with minimal pain and discomfort. So TIO showed multifaceted properties in healing, excision, incision, and burn wound types. The future prospects, further research is being done to show to see the underlying molecular mechanism or signal processes associated with the TIO, and efforts is being taken to mass produce and distribute the ointment at a reasonable rate. These are the references. Thank you.
Thank you, participants, for this very nice presentation. Now I would ask the professors to question.
Achha, what is the nature of the active principle in the Typha extract?
You mean, sir, the action of the
active principles, compounds.
Okay, so Typha angusta is just a plant, and when we are
I know that. I know what is
base of-
Typha. I am asking what is the nature of the extract? What are the compounds that actually cause collagen repair?
Oh, collagen repair has been brought about by using hydroxyproline, and that is bringing
where they have done the assay of the extract. Did you show?
Yes, sir. So the assay of the extract is being done, but it is a little bit complex process which uses a bit of buffers and other process which we could not personally understand. However, we could decipher from the paper that the spectrophotometric analysis was done using centrifugation and extraction of the tissue.
This type of paper, you must actually explain the active principles at least. That is important. Number one. Number two, why it is present in the inflorescence and not in the shoot.
Okay, so sir, we with the Typha angusta, this inflorescence, the effect of this wound healing process comes from the pollen grains only, and the pollen grains contain flavonoids and long chain hydrocarbons and other
I got my
components
answer.
which
Thank
are
you.
primarily responsible. So
Thank you.
we have taken the inflorescence.
Onit, I will just request you to go to the first slide. I somehow missed the topic. The first slide. What is the heading? I just want to see that. And I had also the same question as sir was asking. What is the active principle? How in this plant? How actually? What is it that is helping in these wound repairs and others? So
the pollens, ma'am, specifically the pollens, and actually, ma'am, the pollens could not be directly taken in any way. So we prefer to take out the flowers of the inflorescence, and accordingly we carried out or made an extract. And the extract has a particular weight by weight. That is, we take only 10% weight by weight of it, and we mixed it with the base of the ointment. The base contains different components like PEG 600, methylparaben, and other things just for the to provide as the base as well as for the lubrication and moisture absorption property, and accordingly it is being applied. So and obviously when we use the control,
that's fine, but I think Arup sir will also agree that I don't know if sir it's taken from the pollens, I understand, but what is the active principle is still not clear. Maybe
yes, ma'am. The active principle may be the cellular mechanisms and signal processes. That is exactly
find out by
how
HPLC or LCMS. Otherwise, you cannot really comment on the nature of the phytomedicine. When you present this type of paper, make it a point that you actually state the nature, chemical nature of the extract. That is important. What are the peaks?
Yes.
Because otherwise it just becomes a hypothetical drug.
Okay, sir.
Okay, thank you.
Yeah, it was a very nice presentation. Thank you. Yeah.
I'm extremely sorry. Please go on. Ma'am, please go on. I'm extremely sorry.
It's okay, Jyotipriyo, it's fine. So they have, sir and madam has already asked the questions and they have answered it well. So it's fine. No more questions. Okay. Thank you, Jyotipriyo. Please proceed.
Thank you, ma'am.
Thank you, professors, for your valuable comments. Now, I will be moving on to our next group of presenters, Swarnaprabha Bishwas and Ushmita Dev. Please go on.
Is it visible?
Yes, Ashmita, it's visible.
Okay.
One second. Can you present it once again? I think there's a glitch.
Now is it visible?
Yes, now we can see your screen. Yes.
Yes, am I audible?
Absolutely.
Good evening, everyone. My name is Swarnaprabha and I have Ushmita along with me. Today, we are going to present on the topic allogenic umbilical cord-derived mesenchymal stem cell transplantation for treating chronic obstructive pulmonary disease by Phuong Lethi Bech et al. From VNU HCM University of Science, Vietnam, published in the year 2020. Chronic obstructive pulmonary disease was the third leading cause of death in 2005. According to the World Health Organization, 65 million people worldwide have moderate to severe COPD. In 2005, more than 3 million people died of COPD, accounting for 5% of all deaths that year. Numbers of COPD patients are expected to increase by more than 30% in the next 10 years, and COPD is expected to be the third leading cause of death worldwide by 2030. COPD is treated with medications including bronchodilators, inhaled steroids, oral steroids, and antibiotics, lung therapies such as oxygen therapy, and pulmonary rehabilitation programs and surgeries, including lung volume reduction surgery, lung transplantation, and bullectomy. However, these therapies have limited efficacy and severe adverse effects. Stem cell therapy, especially with mesenchymal stem cells, is a promising therapy for treating various diseases, including inflammation and autoimmune diseases. Mesenchymal stem cells are adult stem cells isolated from different sources that can differentiate into other types of cells, such as bone cells, cartilage, muscle cells, neural cells, skin cells. MSCs are adult stem cells often used to treat diseases such as graft versus host disease, osteoarthritis, autoimmune diseases, and liver cirrhosis. MSCs can be derived from different sources such as bone marrow adipose tissue, peripheral blood, umbilical cord blood, and umbilical cord tissue. MSCs have different beneficial therapeutic mechanisms. First, MSCs can modulate the host immune system by inhibiting some immune cells and stimulating others, thus participating in regulating the immune system. This is the main mechanism of the MSCs used to treat graft versus host disease, autoimmune diseases, and inflammatory diseases. Recent studies have shown that MSCs can directly interact with immune cells and secrete cytokines or interleukins to regulate host immune cells. The second mechanism relates to the MSC secretome. MSCs can produce a wide variety of cell signaling cytokines and growth factors targeting endogenous stem cell self-renewal and migration, and can trigger host stem cells to self-renew and differentiate to heal an injury. Finally, MSCs can home and differentiate after transplantation.
In some cases, particularly in autologous transplantation, MSCs can home and re-establish stem cell niches in the host. These MSCs can differentiate into functional cells that participate in tissue regeneration. This was a clinical trial without a control group, and all participants provided written informed consent. Eligible patients were aged 40 to 80 years with moderate to severe COPD at stage C or D, but the Global Initiative for Chronic Lung Disease, and had a smoking history of more than 10 peg per year. Umbilical cord samples were collected from donors during childbirth as approved by the local ethics committee. The criteria for umbilical cord tissues were that the tissues were from a full-term birth and negative for HIV, HPV, HVC, and syphilis. All samples used to isolate the UCMSCs met these criteria. MSCs were observed under an inverted microscope to evaluate their shape. Their phenotypes were evaluated for CD14, CD34, CD44, CD45, CD73, CD90, CD105. The purity was calculated based on the percentage of UCMSCs that were positive and negative for different CDs. UCMSCs were evaluated for sterility, mycoplasma, endotoxins, and viral infections using commercial kits. These cells were also evaluated for in vivo tumorigenicity. After the potency check, enrolled patients were intravenously infused with fresh allogenic MSCs. Participants were subsequently evaluated for safety and efficacy at 1, 3, and 6 months. A record of COPD exacerbations was maintained for each patient. Efficacy measures included improvement from baseline in pulmonary functions, total lung capacity, single breath carbon monoxide diffusing capacity, exercise performance, that is 6-minute walk test, and quality of life. After that, the results were observed. Now over to Ashmita.
Prabha, now I will be discussing about the result of umbilical cord derived mesenchymal stem cell transplantation for treating COPD. In this slide, this diagram depicts that after doing all the subcultures, UCMSCs were used to perform quality control for the master bank with identity, purity, safety, and potency assays. In this diagram, it is also clear that UCMSCs exhibited a fibroblast-like structure. From this graph, we can understand that the cells showed expression of common MSC markers, highest for CD44, CD90, followed by CD73 and CD105. However, the UCMSCs were negative or low for the hematopoietic markers like CD14, CD34, CD45, and HLA-DR. In this slide, we can see that UCMSCs were successfully differentiated into osteoblast, adipocytes, and chondroblast. In the induced medium geared toward osteoblast differentiation, UCMSCs gradually changed their morphology and gradually produced more matrix. Differentiation of UCMSCs into chondroblast was also recorded in vitro after inducing MSCs for 21 days in the induced medium. This diagram shows UCMSC potency, indicating that the UCMSCs efficiently inhibited T cell proliferation. This result showed that UCMSCs could inhibit T cell proliferation in both PHA and without PHA treatment. Here, all kinds of effects are discussed of this transplantation. Due to their capacity to suppress detrimental immune response, UCMSCs are considered as promising tools for the therapy of COPD. Intravenous administration of UCMSCs was a safe therapeutic approach that showed beneficial effects in the COPD patient. UCMSCs successfully engrafted into the COPD injured lungs of the patients within 24 hours after administration. MSC transplantation showed significantly reduced alveolar damage and alveoli number loss, a statistically improved pulmonary function, forced expiratory volume at 100 milliseconds, and mean forced expiratory flow was noticed in UCMSC-treated COPD patients. Improvement in histological outcomes and pulmonary functions were accompanied by reduced presence of inflammatory cells in the alveolar septa. Among lung-infiltrated and infiltrate immune cells, the main cellular targets of transplanted UCMSCs in COPD patients were macrophages. Furthermore, transplantation of MSCs significantly improved lung architecture of the COPD patients and caused tissue remodeling. The beneficial effect of MSCs in COPD has been also attributed to the inhibition of alveolar cell apoptosis. Beneficial effect of MSCs in COPD are mainly due to paracrine effects involved in the suppression of inflammation and apoptosis in the lung tissue, but also may be a consequence of their differentiation into the structural cells of the alveolar unit. Now, the future perspective. Results obtained in large number of preclinical studies showed that UCMSs may suppress detrimental immune response in the lungs and are able to cure COPD. However, transplanted UCMSs may differentiate in undesired cell types and may promote tumor growth and defined manner. Future studies must be focused on resolving these safety issues before MSc could be offered as a new human remedy for the treatment of inflammatory lung disease. Now, I would like to conclude my PPT. In summary, systematic administration of UCMSs appears safe. The initial results showed that UCMS transplantation improved the number of exacerbations and other different parameters in an older comorbid population of moderate to severe COPD patients with compromised lung function. The stage D COPD patient exhibited a stronger medical response after UCMS transplantation than did stage C COPD patients. Accordingly, it can be concluded that due to their potent immunomodulatory and regenerative properties, MSCs represent new therapeutic agents in the cell-based therapy of chronic inflammatory lung disease. Although this is a pilot study, this primary result provide an important and significant basis for further clinical investigation of MSCs in patients with COPD. Here is all references for our PPT from which we have gathered our information. Thanks to everyone for your attention.
What is the age in which this mesenchyme cells can be transplanted or the recipient?
Sir, it's 40 to 60 years of patients.
But is there any side effect when the cells are transplanted?
Sir, UCMS transplantation did not show any adverse effects as contrast to bone marrow mesenchymal stem cells that showed some adverse effects. So we can conclude that UCMSs were a better option for transplantation and treating chronic.
So human trial was carried out?
Yes, sir.
Human trial হয়েছে?
Yes, ma'am. Yes, ma'am. This was a pilot clinical trial on humans.
Simple. I don't know. That's why I'm asking. So UCMS cells means self or from any UCMS cell will do. মানে আমি যেটা বলতে চাইছি যে সেলফ হতে হবে কি নন সেলফ হলেও চলবে মানে umbilical cord mesenchymal cells any cells will do.
Yes, ma'am. Any cells will do from
any
subject.
Some parameters that need to be checked before transplant. Yes, means just like bone marrow matching is done or the factors that are being checked on. So what are the factors that are to be checked before transplant? Means what are to be matched?
Ma'am, the blood types could be matched. Like
what else? Many other important parameters that might elicit rejection kind of thing. So is this possible there?
Like ma'am, some kind of allergic responses or something like that.
Maybe ma'am is asking for compatibility issue.
I'm not sure.
Yes. Okay. So this approach is very good. If it works, then it will be really nice. So it will be a big boom for medical.
Yeah,
yes, ma'am. It was a recent study.
Right. Very good. You both of you spoke very well, and the best thing is your slides were also flowing very smoothly, right? So you got into the topic very nicely. So
thank you, ma'am. Thank you.
Over to David. Okay.
No, it was a good talk. I am happy with the presentation. Good one.
Thank you, ma'am.
So this brings us to the last presentation of the day, judges. It will be by Kuhu Ponchadai and Shubham Jyoti Biswas. Over to you guys.
Am I audible?
Yes, Kuhu, you're audible and we can see your screen. Go ahead.
Good evening to all my respected professors classmates, juniors, and to all other viewers. Today, my partner Shubham Jyoti and I will presenting on something which is slowly curving its way into our daily life of human being. Artificial intelligence, or AI as it is so known, is indeed proving to be our last weapon in the ongoing war against antimicrobial resistance. Antimicrobial resistance, or AMR, is a natural process whereby microbes evolve in such way so as to withstand the action of the antimicrobial agents and the threats of effective prevention and treatment of ever-increasing range of infection caused by microbes. We all know AI is a quite new thing in our ecosystem, so let us now address the elephant in the room. It is globally accepted that while the uses of antimicrobial has directly led to the control and even eradication of certain infection disease, their use, overuse, misuse, and abuse has led to the development of resistant strain of microorganisms. This phenomena has now grown to what is now an increasingly serious issue, which threatens global health and imposes both social and economical burden on humankind. In addition, previous mechanisms have somehow failed to curb the ever-increasing rampant spread of AMR, and the number of approved antibiotics are decreasing with every passing day ever since as early as 80s. But in recent years, AI has exhibited significant promise in AMR control. Now, I will briefly summarize that what is artificial intelligence, or AI. The term is used to describe the use of computers and technology to stimulate intelligent behavior and critical thinking comparable to a human being. It is ability of a computer algorithm to approximate conclusion based solely on input data. The remaining part, that is why AI is important to us, prediction of AMR and AI-based antibiotic combination AI algorithms, will be discussed by Shubham Jyoti.
Thank you, Guhu. Now, let's look at the various aspects where artificial intelligence is either playing a major part or has already shown ample promise to do so in near future. Currently, two typical methods are widely used to diagnose AMR. Antimicrobial susceptibility testing is the classic method to quantify antimicrobial resistance levels, but it is neither efficient nor does it explain the mechanism of AMR. Whole genome sequencing provides rapid, consistent, and accurate diagnosis for AMR, but it requires large and high-dimensional datasets to extract information effectively. To improve AST methods, a combined method of flow cytometry and supervised machine learning was used. This method generates a reliable result in less than 3 hours, and moreover, the amount of time also gets substantially reduced by developing a method which combines IR spectroscopy with artificial neural network algorithms. For whole genome sequencing, current studies are mainly based on KMR, which is derived from the whole genome of samples. However, KMR datasets are too large and redundant metrics, thus allowing to determine whether a particular KMR is present in the genome. We can also
You got muted, Shubham. You got muted.
Shubham, I think there's a glitch. Can you correct it?
No, it's muted. By mistake, he got muted.
Models that can predict AMR as, sorry, excuse me?
Yes, there's some kind of a network issue, Shubham. You could go to the last slide and then, because we couldn't hear you.
Okay.
Yes, from this slide.
Because am I audible now clearly?
Yes, you're audible right now.
Okay, thank you. So currently, as I was saying, there are two typical methods which are widely used to diagnose AMR. One is antimicrobial susceptibility testing.
Shubham, I think you're facing the same issue again.
I think this group might have to restart. A lot of it was interrupted.
Okay, so let me leave and rejoin. Okay, please excuse me. May I leave, judges, if you permit me to leave I shall leave and rejoin.
Please.
Yes,
please.
yes.
Whatever is good for your presentation, do it.
Thank you, ma'am. Thank you. Am I audible now?
Yes, you are audible. Yeah. Go ahead.
Okay, so should I restart or should I continue from this slide?
I think you can continue from this slide.
Okay, thank you, ma'am. So, as I was saying, ma'am, may I please keep my video off? That way, I might save some bandwidth for the voice.
Ma'am?
I don't know if it is allowed.
Yes, he can switch off his video if he is having band issues. Devjani, we can.
Yeah,
we can,
okay then.
Yeah.
Okay.
Thank you, ma'am. So as I was saying, there are currently two typical methods which are widely used to diagnose AMR. One is antimicrobial susceptibility testing, which is the classic method to quantify antimicrobial resistance levels, but it is neither efficient nor does it explain the mechanism of AMR. The another is whole genome sequencing, which provides rapid, consistent, and accurate diagnosis for AMR, but it requires large and high-dimensional data sets to extract information effectively. To improve the AST methods, a combined method of flow cytometry and supervised machine learning was used. This method generates a reliable result in less than 3 hours, and moreover, the amount of time also gets substantially reduced by developing a method which combines IR spectroscopy with artificial neural network algorithms. For the whole genome sequencing, current studies are mainly based on KMR, which is derived from the whole genome of samples. However, KMR datasets are too large and redundant to be directly used for AI applications. So they are converted to a binary matrix, thus allowing to determine whether a particular KMR is present in the genome. They can also be used to build classifier models that can predict AMR as well as investigate the relationships between genotypes and phenotypes. Puhu, next slide, please. Thank you. Since appropriate antimicrobial therapy is crucial for reducing the mortality of antibiotic-resistant infections, it is an absolute urgency to develop fast and efficient methods to guide the rational use of antibiotics. And this is where clinical decision support systems play a major part. Rule-based or traditional CDSS involves a large number of patients and clinical data to extract the rules. However, there is not enough manpower, time, or resources to devote to these tasks. Recently, the development has led to automatic extraction of these rules from a large amount of clinical data. They can not only infer the current state of the patients, predict the evolution, and obtain the optimal antibiotic use strategy, but also demonstrate that cases with prognosis rates of the AI-based CDSS group are significantly greater than with that of the control group. In this regard, quantitative structure-activity relationship or QSAR descriptors were employed to identify 50 novel AMPs from 100,000 random peptides. The bacteriostasis in vitro showed that this model has an accuracy of as much as 94% in identifying active peptides. Moreover, several physical and chemical parameters like charge, hydrophobicity, isoelectric point, aggregation tendency, and peptide sequences can also be used as features to train such models for usage in this purpose. For antibiotic combination studies, AI-assisted methods can speed up the process of exploring optimal combinations by reducing the number of experiments required. Yet, they successfully maintain almost the same high accuracy as that of the traditional methods. Here in this table, the commonly used AI-based predictive applications have been listed. Now, let us take a look at some of the most commonly used algorithms which help us in studying AMR. Naive Bayes is a classification method based on Bayes' algorithm with independent assumptions of each feature. Recent studies have typically employed this model to monitor AMR. For example, Naive Bayes was used to determine the key factor of resistance, to extract resistant patterns, and also to estimate the probabilities of ineffective treatments due to AMR. Decision tree is usually used for classification. Here is a simple decision tree model that divides a dataset into 3 classes based on 2 features and considering simple numerical values. This model is often used to allocate medical resources appropriately by estimating the burden of AMR. Multiple decision trees make up a random forest. This is an ensemble algorithm that employs multiple decision trees to improve accuracy, and RF models have been commonly used to predict antibiotic combinations. Support vector machine, or SVM, is a binary classification model. Support vector machine models have been employed to predict AMR phenotypes, that is, resistant or susceptible. One such model predicted whether E. coli are resistant to antibiotics, and the results of which achieved average accurate predictions up to as high as 95%. Another model was employed to examine the resistance of these 5 drugs. The results showed that even this model achieves accuracy of around 90%. Therefore, SVM models can definitely be considered as potential tools for AMR surveillance and clinical diagnosis. Now we come to the last algorithm in the purview of this presentation, which is artificial neural networks. These are abstract mathematical models that are based on neurons in human beings to transmit information through weighted, interconnected computer units or neural layers. Recently, new antibiotics were identified without any prior assumptions using a deep learning approach, which is one of the artificial neural networks. In mouse models, these molecules have antibacterial activities against a variety of pathogens. This discovery opens up a new possibility of using this deep learning approach to design new antibiotics and optimize existing molecules, which would spark a paradigm shift for future antibiotic discovery. Finally, here is a comparison of the 5 commonly used algorithms for AMR. It may be concluded that among these, decision tree is the most balanced algorithm with a good speed of learning and great interpretability in its results. Once again, I hand this presentation over to Kuhu to discuss the limitations, the ethical dilemmas involved, and also to highlight upon the future scope and prospects of this field. Thank you.
Kuhu, Shubham, actually the time has been exceeded. Can you wrap up the presentation?
Yeah, so these are the limitations of this of using artificial intelligence in the field of antimicrobial resistance, like very few hospitals willing to share their clinical data, and also the hospital staff and the healthcare workers are very not willing to take up AI. And okay, so the slide is not changing. I don't know why. Yeah, so here are the future prospects, like we can build AI-based continuous models to directly predict the activity of AMPs against specific microorganisms, and also develop algorithms that can use smaller sample size to train the clinical decision support system models, such as the few shot learning algorithm, which is quite a big thing to discuss right now. And in conclusion, we can say that this field is quite nascent right now, but it is showing great promise in the antimicrobial resistant field, and we can only hope that this will come to our benefit and will help us in our constant war against the antimicrobial resistance. Thank you, and I apologize for all the inconvenience that was caused during this presentation.
The biggest problem is this is based on certain algorithm.
Yes, sir.
Which may not be true for bacteria always, because they don't have brain. artificial intelligence usually tend to be applied in those cases where there are some behavioral pattern quite distinct. So based on statistical analysis, I have my doubts whether this will be a success in future. Anyway, it's a good field, but you need to see how it comes up.
Yes, sir. It can only be said that only time will tell, because right now it is showing promise, but in the future, who knows? It might go on to give good results or might like just hit a wall.
Because you see, biological science, they do not follow any principle. That's why it is biology. It is not physics, not chemistry. Taina bolo? They do not follow any principle.
There are a lot of unpredictability how each and everything will behave. There is not a set pattern in most things.
And that also with microbes.
Yes, sir. Yes, sir.
Actually, it becomes a joint venture at times in case of microbes, right? So they jointly can perform events. So that becomes very difficult to combat.
Yes.
Right. But definitely this approach is very novel, and this is good. In this respect, this is good. That thinking in this angle will definitely help the researchers to work in a better way. Okay.
Yes, artificial intelligence will definitely come up. May not be, as sir is trying to tell us, may not be with bacteria, but definitely it is the newer future approach for many other things. So if we could associate that with microbes, well and good. If not, it's okay.
Again, I think the future can be like this. If the gut-brain microbiome is there and our behavior is controlled by that, our behavioral pattern can be controlled by artificial intelligence.
Right.
Immediately it can be connected.
Indirectly, yes, it can be
indirectly.
So this can lead to another theory, in fact. Right now, I was just thinking and thinking. So it can be a good theory. Maybe future students will consider this topic.
Definitely.
Thank you, judges. So this brings us to the end of the third technical session of day 2 of MTIM. I would like to thank our judges, Dr. Mahashweta Mitra Ghosh and Professor Devjani Datta for moderating this session. I would also like to thank Dr. Arup Kumar Mitra sir for giving us his valuable inputs. Thank you, participants, for your wonderful presentations. We shall shortly begin with the cultural session. Over to you, Ananya.
Yes, thank you, Natasha. So with this, we would like to end the technical sessions for day 2. And I would like to thank all the judges, MMG ma'am, KS ma'am, DG sir, our HOD, Dr. Shudeshna Shyam Chaudhary, and of course, AKM sir, our convener of Modern Trends in Microbiology 2021, the judges who were present, our speaker, Dr. Daniel Ryan, who gave such an amazing lecture, and of course, the whole MTM organizing team, especially the MCs, without whom the program would not have been successful. And with this, we would like to move on to the cultural program also. And one more thing that we will be starting tomorrow is the day 3, the final day of this international webinar, Modern Trends in Microbiology 2021. And we will have some very exciting external poster presentations coming up tomorrow. So please stay tuned from 11 a.m. tomorrow. We will be starting. Stay tuned with us. Please like, share, and subscribe to our YouTube channel. And don't forget to press the bell icon so that you get notified when the program starts. And please also take note that we will be providing the feedback form tomorrow, for which will have questions from the lectures of all 3 days of all the guest speakers.
and following which, if you fill up that correctly, you will be receiving your certificates. And over to you, Natasha. So please announce the theme of the day 3 of the program. And let's begin with the cultural program. Thank you.
Okay. Thank you, Nanya. So the day 2's cultural program.
Yes, today the theme is Ritu Boichitro, or it also means that the theme is based on the 4 seasons which are experienced. So yes, over to the cultural committee and presentation and streaming committee, who will be streaming the cultural program live on the YouTube channel as well.
বাংলায় ষড়ঋতুর বৈচিত্র্যে পুষ্পে পল্লবে চারিদিকে সেজে ওঠে প্রকৃতি। গ্রীষ্ম ঋতু দিয়ে বাংলার ঋতু বৈচিত্র্যের শুরু। গ্রীষ্মের প্রখর সূর্যকিরণে চারিদিক জ্বলতে থাকে। এই রুদ্র ভৈরব মূর্তি নিয়ে তাপদগ্ধ বাংলা তখন হয়ে ওঠে তৃষ্ণার্ত। তার সে তৃষ্ণা দূর করে তৃষভরা বর্ষা, সঞ্চার করে নবজীবনে। সারা আকাশে তখন জল ভরা কালো মেঘ ছোটাছুটি করে বেড়ায়। যখন কেটে যায় এই কালো মেঘের জাল, দেখা দেয় শরতের সোনালি রূপ। নীল আকাশে পেঁজা তুলোর মতো সাদা মেঘ ভেসে বেড়ায়। মাঠে মাঠে দেখা যায় কাশফুল। শরতের এই উজ্জ্বলতা ধীরে ধীরে বিলীন হয়ে যায় হেমন্তের ধূসর আস্তরণে। পাকা ধানের সোনালি আভায় হেমন্ত লক্ষ্মীর মুখখানি যেন উজ্জ্বল হয়ে ওঠে। হেমন্তর হাত ধরে আসে শীত। গাছের পাতা ঝরে পড়ে চারিদিকে, দেখা দেয় শ্রীহীন এক রুক্ষ ভাব। শীতের এই কুয়াশা এবং অন্ধকারকে ভেদ করে আসে ঋতুরাজ বসন্ত। প্রকৃতিতে তখন কেবল চারিদিকে রং আর রং। ফুলের বনে মৌমাছির গুঞ্জন, আম গাছের ডালে ডালে কোকিলের কুহুতান। বছরের পর বছর ধরে ঋতুতে ঋতুতে প্রকৃতি নিজেকে এইভাবে সাজিয়ে তুলেছে। Rain is grace, rain is the sky descending to the earth. We all love music of rain. Rain soothes both soul and soil.
ঝোরা পাতা উড়ে তাকে ছুঁয়ে বলে যা জাগে এখানে বড় ফিকে সব তুই যা যা যা। তাই সে যায় ছুটে বেড়ায় ধূসর প্রান্তরে, মেঘের গায়ে হাত বোলায় রামধনু হাত বাড়ায়, মেঘের আড়ালে ভেসে থাকা সেই রামধনুকে চায়।
Gilly 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 ha, ha 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 ha, ha 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 ha. Gilly gilly mati ke jagharonde banayenge re. Hari hari ambe ki dali milke jhule jhulayenge re. हाँ धन बैजो गजनी हर जोते सबने बैल की घंटी बजी और ताले लगे भरने तैर के चली मैं तो पार चली पार वाले पाले किनार चली रे मेरा नन्ना रे 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 नन्ना रे। যাও পাখি বলো হাওয়া চলো চলো আবছায়া জানালার কাছ আমি কি আমাকে হারিয়েছি বাকি রূপকথা নাচ কানাচ আঙুলের কোলে চলে জোনাকি জলে হারিয়েছি কান শোনাকি জানালাই গল্পেরা কথা মেঘ যাও মেয়ে চোখে রেখো এই আবেগ যাও পাখি বলো হাওয়া চলো চলো আবছায়া জানালার কাছ আমি কি আমাকে হারিয়েছি বাকি রূপকথা নাচ কানাচ।
শরৎকাল, নীল আকাশে সাদা মেঘেদের আনাগোনা, বাতাসে শিউলি ও পদ্মে মধুর সৌরভ, মাঠে মাঠে কাশ ফুলের মেলা, চারিদিকে বেজে উঠেছে আগমনী সুর। কি অপরূপ এই সৌন্দর্য!
নমস্কার। কবিগুরু রবীন্দ্রনাথ ঠাকুরের কবিতা শরৎ। এসেছে শরৎ, হিমের পরশ লেগেছে হাওয়ার পরে, সকাল বেলায় ঘাসের আগায় শিশিরের রেখা ধরে, আমলকি বন কাঁপে যেন তার বুক করে দুরু দুরু, পেয়েছে খবর পাতা খসানোর সময় হয়েছে শুরু। শিউলির ডালে কুড়ি ভরে এলো, টগর ফুটিল মেলা, মালতী লতায় খোঁজ নিয়ে যায় মৌমাছি দুই বেলা। গগনে গগনে বর্ষানো শেষে মেঘেরা পেয়েছে ছাড়া, বাতাসে বাতাসে ফেরে ভেসে ভেসে নাই কোনো কাজে তারা। দিঘি ভরা জল করে ঢল ঢল নানা ফুল ধারে ধারে, কচি ধান গাছ খেতে ভরে আছে হাওয়া দোলা দেয় তারে। যেদিকে তাকাই সোনার আলোয় দেখি সে ছুটির ছবি, পূজার ফুলের বনে ওঠে ওই পূজার দিনের রবি। ধন্যবাদ।
Spring is a lovely reminder of how beautiful a change can truly be. Lush green environment with bright leaves, beautiful flowers, buzzing bees and colorful butterflies. Spring is a healthy season with fresh air and sunshine. A perfect summer day is when the sun is shining, the breeze is blowing, and the birds are singing. Monsoon gives us the essence of life, calmness, growth, rebirth, and fertility. Autumn represents the preservation of life and its basic necessities. Winter is a long sleep with a warmth of its own. Spring is a spectrum of colors filled with the fresh smell of nature. Every season is beautiful in its own way.
দু চোখে হঠাৎ করে কাল বৈশাখী,
চৈত্রের শেষ বেলা পাতা ওড়ে নাকি,
গত বছরের মায়া ভেঙে যাবে বলে
রাজপথ ভেসে গেছে অচেনা কাজলে।
দু চোখে হঠাৎ করে কাল বৈশাখী, চৈত্রের শেষ বেলা পাতা ওড়ে নাকি, গত বছরের মায়া ভেঙে যাবে বলে রাজপথ ভেসে গেছে অচেনা কাজলে। তুমিও অঝরে তাকে শুধু ভালোবেসো, এসো হে বৈশাখ এসো এসো, এসো হে বৈশাখ এসো এসো। বলে যে যোগে যোগে তোমার লাগে আছি জেগে, মরণ বলে আমি তোমার জীবন করি পাই। মেঘ বলেছে মেঘ বলেছে যাব যাব, রাত বলেছে যাই, সাগর বলে কুল মেলেছে আমি তোমার নাম। মেঘ বলেছে
শূন্য এখন ফুলের বাগান, দোয়েল কোকিল গাহে না গান, কাশ ঝরে যায় ঝরে যায় ঝরে যায় নদীর তীরে, কাশ ঝরে যায় ঝরে যায় ঝরে যায় নদীর তীরে। যা কব সাদ বিষাদ কালো দীপালিকায় জ্বালাও আলো, জ্বালাও আলো আপন আলো, শোনাও আলোর জয় বাণী রে। মেরু রাতে ওই গগনের দ্বীপ গুলি রে। এক বছরের শান্তি পথের কালের আয়ু হয়, কাকা ধানের বিদায় ঋতু নূতন আশার ভয়। পৌষ এলো গো, পৌষ এলো, শুকনো নিঃশ্বাস কাঁদন ভরা দুর, বিদায় ক্ষণের ভাঙা গলার সুর। উঠো পথিক যাবে অনেক দূর, উঠো পথিক যাবে অনেক দূর, কালো চোখের করুণ চাওয়া ছাড়ায়। পৌষ এলো অশ্রু পাথরহীন পারাবার পারারে, পৌষ এলো গো, ওই যে গেল গো উচ্ছটিকার ঘুমটা পড়া দিগন্তরে দাঁড়ায়। পৌষ এলো গো, পৌষ এলো অশ্রু পাথরহীন পারাবার পারারে, পৌষ এলো গো। কিছু স্বপ্ন কিছু মেঘলা, কিছু বই টই ধুলো লাগা, কিছু ইচ্ছে সারা দিচ্ছে এই বসন্ত রাত জাগা। যে পলাশ ফুলের কাব্য, নীতি নৃত্যে ফুল ছিঁড়তে শুধু তোমার কথাই ভাববো। আজ হাওয়া বেপরোয়া দিল সন্ধে পাখির ঝাঁক, এই বিকেল আর বিলকুল হৃদ মাঝারে থাক। নীল দিগন্তে ওই ফুলের আগুন লাগলো লাগলো, নীল দিগন্তে বসন্তে সৌরভের শিখা জাগলো, বসন্তে নীল দিগন্তে। আজো সখী মোহু মোহু কাহে পিক কুহু কুহু, কুঞ্জবনে দুহু দুহু দোহার মানে যায়।
आज मधु चांदनी प्राण उन्मादिनी, आज मधु चांदनी प्राण उन्मादिनी, शिथिल सब बांधनी शिथिल भैलाज, वचन मृदु मरमर कांपिरिछ थर थर, शिहरे तनु जर जर कुसुम बनमाज, मलय मृदु कलई छे चरण नहीं चलई छे, वचन मोह खलई छे अंचल लुटाय।
Spring is the season of new beginnings. It is a time of rebirth, renewal, and awakening. Many trees blossoming, early flowers blooming. The first blooms of spring always make my heart sing.
বাতাসে বহিছে প্রেম, নয়নে লাগিল নেশা, কারা যে ডাকিল পিছে, বসন্ত এসে গেছে। মধুর অমৃত বাণী বেলা গেল সহজেই, মরমে উঠিল বাজি, বসন্ত এসে গেছে। থাক তবু ভুবনে ধুলি মাখা চরণে, মাথা নত করে রব, বসন্ত এসে গেছে, বসন্ত এসে গেছে। পূর্ণিমা সন্ধ্যায় তোমার রজনীগন্ধায়, রূপসাগরের পারের পানে উদাসী মন্ধায়। পূর্ণিমা সন্ধ্যায় তোমার রজনীগন্ধায়, রূপসাগরের পারের পানে উদাসী মন্ধায়। তোমার প্রজাপতির পাখা আমার আকাশ চাওয়া, মুগ্ধ চোখের রঙিন স্বপ্ন মাখা। তোমার চাঁদের আলো নিলয় আমার দুঃখ সুখের সকল অবসান। ফাগুন হাওয়ায় হাওয়ায় করেছি যে দান, তোমার হাওয়ায় হাওয়ায় করেছি যে দান।
মাঠের বাঁশি শুনে শুনে আকাশ খুশি হলো, ঘরেতে আজকে রবে গো, খোলো 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 দুয়ার খোলো। আলোর হাসি উঠলো জেগে ধানের শীষে শিশির লেগে, ধরার খুশি ধরে না গো, ধরার খুশি ধরে না গো, ওই যে উথলি মরি হায় হায় হায়। তোদের ডাক দিয়েছে আয় রে চলে আয় আয় আয়, ডালা যে তার পড়েছে আজ পাকা ফসলে মরি হায় হায় হায়। তোদের ডাক দিয়েছে আয় রে চলে।
fresh and unfiltered beauty that captivates us. There is something magical about rain. They not just calm the parching earth, but also symbolize life and creation. Rain makes things grow. Rain encourages fun. Rain inspires creativity. Weather, ecology, and hours of sunlight changes differentiates the seasons apart. But no matter which one we dearly love, they are all unique in their own ways. Till now, all the performances beautifully portrayed the diversity and characteristics of each season. Next, we move on to a completely new genre. A comedy drama will be presented before us by the PG students, titled Climax, written by Rishabh Mukherjee. জীবনে যা কিছু করব বা করতে চাই তার মান বজায় রাখাটা অত্যন্ত জরুরি। এ কথা চোর থেকে চৌকিদার প্রত্যেকের ক্ষেত্রেই প্রযোজ্য। আমাদের আজকের পরিবেশনায়ও আরও একবার সেই সত্যকে সকলের কাছে তুলে ধরবে। সঙ্গে থাকবে হাসি ও মজা। দেখতে দেখতে মনে পড়বে মনোজ মিত্রের নিশি কুটুম্বের কথা।
হ্যাঁ হ্যালো, আপনি লোকেশনটা দেখতে পেয়েছেন তো? আমি সেভ ড্রাইভ পোস্টারটা নিচে দাঁড়িয়ে আছি। হ্যাঁ, আপনি লোকেশন থেকে চলে আসুন। কি, আপনি কোথায়? দিদিকে বলো সামনে? না বসে, এখানে কোনো দিদিকে দেখতে পাচ্ছি না আমি। আপনি লোকেশনটা দেখ। হ্যাঁ, সেই রাইট ক্যান্সেল করে দিচ্ছেন কেন? আরে আপনি দিদির সামনে দাঁড়িয়ে লাইট ক্যান্সেল করে দিচ্ছেন, আপনার তো সাহস কম না? হ্যালো হ্যালো
দাদা, এটা কি শ্রদ্ধানন্দ পার্ক?
না, এটা গোল পার্ক।
না, পার্কটা তো গোল সেটা বুঝতে পারছি, কিন্তু এটা কি শ্রদ্ধানন্দ পার্ক?
আচ্ছা মানুষ তো আপনি। আরে এটা গোল পার্কই, নামটাই গোল পার্ক, ওইদিকে পূর্ণদাস রোড।
কি আশ্চর্য ব্যাপার! আমি তাহলে কোথায় এসে পড়লাম? আমার বন্ধু দাঁড়িয়ে আছে শ্রদ্ধানন্দ পার্কে, আর আমি এই গোলে। কি মহা গন্ডগোল!
আচ্ছা,
মানে দাদা, কেন যে যাব সেটাই ভুলে গেছি। আজকাল যেন কিছুই মনে রাখতে পারছি না।
সে কি?
হ্যাঁ, আজকাল আমার এটা খুবই হচ্ছে, আমি সবকিছুই ভুলে যাচ্ছি, এমনকি নিজের নামটাও।
হ্যাঁ, এও এক ধরনের রোগ বটে।
ধুস, পোড়া কপাল আমার। আচ্ছা দাদা, মেট্রো স্টেশনটা কোন দিকে?
সে তো অনেক দূর, কিন্তু এক মিনিট, আপনি মেট্রো করে তো শ্রদ্ধানন্দ পার্কে যেতে পারবেন না।
দূর, ওখানে কে যাচ্ছে আর? আমি আর ওসব ভাবছি না, আমি মেট্রোর লাইনে ঝাঁপ দিতে যাচ্ছি।
এই মরেছে। এই না 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 দাদা, শুনুন শুনুন, এখন অফিস টাইম, বুঝলেন তো? লাইনে আপনি যদি পড়ে না মরতে পারেন, পাবলিক আপনাকে এমনিতেই পিটিয়ে ছাতু বানিয়ে দেবে, তারপর আবার পুলিশ কেস টেস।
উফ, এই হতভাগ্য জীবনটা আমি আর রাখবো না। আপনি বলুন তো কিভাবে মরা যায়? আমি এভাবে আর থাকতে পারছি না।
সে কি, আপনাকে একা ছাড়া ঠিক হবে না। আপনি এক কাজ করুন, আপনি ঠান্ডা হয়ে দাঁড়ান দাঁড়ান, আপনি দাঁড়ান। হ্যালো, বলছি তুমি আজকে একটুখানি ছেলেকে টিউশন থেকে নিয়ে আসবে। হ্যাঁ, আমি একটা খুব ইম্পর্টেন্ট কাজ ফেসে গেছি। হ্যাঁ, কি বলছিলেন বলুন।
আপনি আমার জন্য এতটা?
আরে না 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 না, ওটা কোনো ব্যাপার না। আপনি শুনুন, আপনি ভালো করে মনে করার চেষ্টা করুন। আপনার নাম, আপনার ঠিকানা।
পার্কটার নামটা কি যেন?
গোল পার্ক।
হ্যাঁ 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 হ্যাঁ, মনে পড়েছে।
কি?
গোলক পার্ক।
এই তো মনে পড়েছে। আচ্ছা, এবার আপনার বাসস্থান।
হ্যাঁ হ্যাঁ, মনে পড়েছে জায়গা। বালগেরিয়া।
আপনি বুলগেরিয়ায় থাকেন?
হ্যাঁ, এরকমই তো নামটা ছিল।
সে তো অনেক দূর।
কই, বাসে করেই তো এলাম।
বাসে করে বুলগেরিয়া?
না না, আমি ঠিক জায়গায় বলছি।
না 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 না। আচ্ছা, জায়গাটার সম্পর্কে আরো কিছু বলুন তো দেখি, মনে করার চেষ্টা করুন দেখি।
ওখানে একটা রেল লাইন আছে আর ওসব, কিন্তু আমার কেন জানি না শুধু কদবেলের কথাই মনে পড়ছে।
সাবাস তো, সে বুঝেছি, বেলঘরিয়া।
আরে বাস, চমৎকার! আপনি তো পাকা গোয়েন্দার মতো কথাবার্তা বলছেন।
গোয়েন্দা বলুন, সত্যান্বেষ। যাকে আপনার যখন এতটা মনে পড়ে গেছে, তখন বেলঘরিয়া যে কোনো বাসে উঠতে পারলে আপনি নিশ্চিন্ত। আমার বিশ্বাস ওখানে গেলেই আপনার বাড়িটা কোথায় সেটাও মনে পড়ে যাবে।
বলছেন?
200 বার। চলুন, আপনাকে এখন একটা বাসে উঠিয়ে দিই।
কিন্তু দাদা, আমার যে বন্ধু দাঁড়িয়ে।
এতক্ষণে আপনার বন্ধু রোদে দাঁড়িয়ে হয় অজ্ঞান হয়ে গেছে, আর নয়তো বাড়ি চলে গেছে।
ও, তাও বটে, ঠিকই বলছেন।
আর শুনুন, আপনি না এই নিজের এই ভুলো মনের চিকিৎসাটা করাবেন, বুঝলেন?
আচ্ছা,
ওই যে বাস আসছে, আপনি উঠে যান। হ্যাঁ, উঠে যান।
থ্যাংক ইউ দাদা, টাটা, ধন্যবাদ।
আসলে কি হয়েছে জানো?
হ্যাঁ বলো, শুনছি, সব শুনছি আমি, বলো।
না মানে ব্যাপারটা আসলে কি বলতো?
কি হয়েছে আমি বলছি। আমাকে পড়ে কি মরে করে ছুটতে হয়েছে আজকে ঘরের সব কাজকর্ম ফেলে। ছেলেটা স্কুল ছুটি হওয়ার পর 8 ঘন্টা বাইরে দাঁড়িয়েছিল। তুমি তো বলেই খালাস, কি মনে করো তুমি আমি মানুষ নই নাকি? যাচ্ছে তাই একেবারে একদম। তুমি একাই বাইরের সব কাজ করো, আর কেউ কি?
আস্তে আস্তে, দাড়ি কমা ফুল স্টপ কিছুই তো করছে না। শোনো, ব্যাপারটা কি ঘটেছে?
এখন যত রকমের এক্সকিউজ দেবে তুমি, যেন সব দায় আমার। এদিকে পান থেকে চুন খসলেই কি ভেবেছোটা কি তুমি? এভাবে হয় না।
আমার তুমি তো রেকারিং নাম্বারের মতো শুরু হয়ে গেলে। বলি আমি এবারে।
বলো।
জানো কত বড় সমাজসেবা আজকে করেছি? একজন মৃত্যু পথগামী মানুষকে আমি প্রাণ বাঁচিয়েছি।
একজন মরে শান্তি পেতে চাইলো, তাকে শান্তি পেতে দিলে না, আর এদিকে আমার অশান্তি আরো বাড়ালে তুমি।
আরে, লোকটা মেট্রো স্টেশনে ঝাঁপ দিতে যাচ্ছে, আমি ছিলাম একদম পেছনে। যেই লোকটা ঝাঁপ দিতে যাচ্ছে অমনি ওনার হাতটা ধরে ফেলি। আমি না ধরলে তো ওর ভবলীলা সাঙ্গ হয়ে যেত।
এক মিনিট, তুমি তো মেট্রোতে যাও না, তোমার তো কার্ডই নেই, তাহলে?
না মানে মেট্রো প্ল্যাটফর্মে না, স্টেশনে একটু দাঁড়াও। হ্যালো
হ্যালো দাদা, চিনতে পারছেন? নমস্কার, আমি গোলপার্ক। এই ভুল বললাম, গোলকপাল।
হ্যাঁ হ্যালো, আরে আপনি বাড়ি খুঁজে পেয়েছিলেন তো? কোনো অসুবিধা হয়নি?
দাদা, আর আপনার কাছে আমার কৃতজ্ঞতার কোনো শেষ নেই। আপনার তো কোনো জবাবই নেই।
কি বা করলাম? এক মিনিট, কিন্তু আপনি আমার নাম্বার পেলেন কি করে?
হ্যাঁ, আসলে সেই কথাটাই আপনাকে জানাবার জন্য ফোন করেছি। কবি বলেছেন, দিবে আর নিবে, মিলিবে আর মেলাবে, কিন্তু যাবে না ফিরে। আর দাদা, আমি এই কথাটা এত বিশ্বাস করি, আর আপনি আমাকে যা টাকার আত্মত্যাগ দিয়ে যা দিলেন, এ আর আলাদা করে আপনাকে কি আর বলবো বলুন।
টাকার আত্মত্যাগ মানে?
হ্যাঁ, আসলে ছোট ভাইয়ের মতো আপনার পকেট থেকে আপনার মানি ব্যাগটা নিয়ে আমি সেটা পকেটস্থ করেছি, আপনি সেটা টেরই পাননি। আসলে উচ্চ দরের হস্তশিল্পী কিনা।
আপনি চুরি করেন?
এ বাবা, এসব আবার কি ধরনের কথাবার্তা? এসব আমাদের বলবেন না, আমরা কি সমাজের কেউ নই? এইসব বাবা চোর টোর অপবাদ এসব বাজে ভাবে দেবেন না, বরং বলুন আমরা উচ্চদরের হস্তশিল্পী। একটু বেশ ব্যাপারটা অন্যরকম হবে, তাই না? আচ্ছা, আমার মোটামুটি চলে যাবে, আপনি ভালো থাকুন, একটু সামলে নিন, ঠিক আছে? টাটা, ভালো থাকবেন।
ফোন করেছে।
গোলপার্কের হস্তশিল্পী।
We have now come to an end to our second day cultural program of modern trends in microbiology. I thank you all on behalf of St. Xavier's College Autonomous Kolkata for spending your valuable time to encourage these young minds. Stay healthy, stay safe. Neelatra.
Thank you for the nice program. So good night, all of you. See you tomorrow.
Good night, sir.